right? If a population of a single species becomes separated by a geographical barrier, point number 2, obviously the population will split, isn't it? Yeah, it will split into two, provided that that geographical barrier is separating the population into two, right? So if a population of a single species becomes separated by a geographical barrier, the population will split into two. Point number 3, there is now no gene flow between the two populations. Point number 4, since the two populations are now exposed to different environmental conditions, natural selection will take place independently, such that they become different from each other genotypically and phenotypically. And even if they were to mix again, they would not interbreed and produce fertile offsprings. All right. Okay, let's go back. Right, so we are saying that if they say to you, explain speciation using geographic isolation, so you are going to say, now if a population of a single species becomes separated by a geographical barrier, number 1, if a population of a single species becomes separated by a geographical barrier, number 2, the population will split into two. Number 3, there is no gene flow between the two populations. Number 4, since the two populations are now exposed to different environmental conditions, natural selection will take place independently in each population, such that the two populations become different from each other genotypically and phenotypically. And even if these two populations were to mix again, they would not interbreed and produce fertile offsprings. All right, now let's explain it using the application of theory. All right, so let's say now we have a pond or a dam. Right, so in this dam, there's species of fish, isn't it? And then there's variation there, because we know that there's always variation, isn't it? Right, there are fishes which are blue, and there are ones that are black. Look at the fishes there. Right, so we have two types of fish. We have blue fish and then we have black fish. Right, so if this population becomes separated by a strip of land, right, so this is the original population. So this population is separated by a strip of land. Separated by a strip of land. Of water or of land, ne? Right. Now we have these fishes here. All right. So now let's say this one is a dark river, maybe dark pond, or here it's a blue. Right. So now, so if they say to you, use the diagram above to explain the formation. Let's give these fishes names. What name can we give this one? Yeah? Naled. And then this one, Sihle. Alright. So now they are asking you explain using speciation the formation of a species called Sihle and another one called Naledi in the fishes. Right. So can we pay attention? Right. So we are going to say, right, the population of fishes was separated by a strip of land. The population split into two. There was no gene flow between the two populations. Since the two populations were now exposed to different environmental conditions, then you explain that different environmental condition. Some they were found in a blue dam and others they were found in a dark dam. Right? Since the two populations are exposed to different environmental conditions, some are found in a blue dam and some are found in a dark dam. Natural selection took place independently in each population such that they became different. The fish became different from each other genotypically and phenotypically. And even if they were to mix again, they would not interbreed and produce fertile offsprings. Hey, right. So you can see that here in a dark dam, the ones that are dark colored, they survive, right? Why? Because they are camouflaged. And the ones that are blue, they die because they are easily exposed. Can you see it? Right? Did you get that one? Or should we do another example? The last example. Right. So if they show you, right, now, let's say this is Africa. And then here we are going to have Australia. Right. And then we have dogs. Here we have what you call jackals. And then here we have coyote. Right. And they ask you, explain the formation of jackals and the coyote in the dog family. Right. Using speciation. Right. Explain the formation of the jackal and the coyote in the dog family using speciation. So you are going to say the population of a dog family was separated by the ocean. The population split into two. There was no gene flow between the two populations. Since the two populations were exposed to different environmental conditions, natural selection took place independently in each population, such that the two populations became different from each other genotypically and phenotypically. And even if they were to mix again, they would not interbreed and produce fertile offsprings. Right. Now let's do mechanisms of reproductive isolation. Mechanisms of reproductive isolation. Oh yeah. All right. So we have 5 mechanisms of reproductive isolation. What are those? Those are mechanisms that keep organisms which are not of the same species separate. Right. Mechanisms. If I was you, I was going to write notes instead of checking the book. It helps. Of reproductive. Mechanisms of reproductive isolation. Those are mechanisms that keep species separate or keeps organisms of different species separate. Right, we have 5. The first one is species-specific courtship behavior. Species-specific courtship behavior. Species-specific courtship behavior. Yeah, species-specific courtship behavior. That if different organisms have different courtship behaviors, then they won't be able to interbreed. Right. Number 2, it's being adapted to different pollinating agents. Being adapted to different pollinating agents. Right. So flowers reproduce by a process called pollination, which is a transfer of pollen from an anther of one plant to the stigma of another plant. That's called cross-pollination. Right. But this pollination is made possible by the agents which are called pollinating agent. It can be bees, it can be wind, it can be any flies. So if different flowers are adapted to different pollinating agents, then they won't be able to interbreed. Are we together? Right. And then number 3. Hey, there's noise there. That disturbs me. Right. Number 1, species-specific courtship behavior being adapted to different pollinating agent. Number 3, infertile offsprings. Infertile offsprings. That during artificial selection, if you interbreed organisms of different species, then the offspring will be infertile, which means the generation ends there. It won't reproduce any further. Right, number 4, it's breeding at different times of the year. Breeding at different times of the year. Breeding at different times of the year. Right, and then the last one, it's prevention of fertilization. Prevention of fertilization. Prevention of fertilization. Right, what causes that one? Right, who can tell me what causes prevention of fertilization? You can't have a huge animal interbreeding with a small animal because a huge animal has big genitals. Yeah, so those genitals really that they cannot. In other words, let me just put it blunt: the penis cannot deposit sperms inside the vagina. So we call that prevention of fertilization because it's too big and the vagina is small. Yeah. All right. Now we go to human evolution. Right. Human evolution. Human evolution. Right. Hey, shh. We are doing human evolution now. Right. But before we go to human evolution, please just know present day evolution. When we are talking about present day evolution, that's when we are talking about uh, resistance, having resistance. So they are going to ask you, maybe a question will come and say, right, explain the development of resistance of bacteria to antibiotics. Explain the development of resistance of bacteria to antibiotics. So you're going to say, now there's a great deal of variation among bacteria. Some bacteria are resistant to antibiotics and some bacteria are not resistant to antibiotics. When the antibiotics are introduced to their environment, the bacteria that are resistant to antibiotics, they survive. And the ones that are not resistant to antibiotics, they die. And the bacteria that survive, they reproduce and pass on the allele of resistance to their next generation. And eventually, there's a great proportion of bacteria which are resistant to antibiotics. Hmm? Right. The question is, explain the development of resistance of bacteria to antibiotics. Explain the development of resistance of bacteria to antibiotics. So we are going to say there's a great deal of variation amongst bacteria. Some bacteria are resistant to antibiotics and some bacteria are not resistant to antibiotics. When the antibiotics is introduced to their environment, the bacteria that are resistant, they survive. And the bacteria that are not resistant, they die. And the bacteria that survive reproduce and pass on the allele of resistance to their offsprings or to the next generation. Then eventually, there's a great proportion of bacteria that are resistant to antibiotics. Now we are doing human evolution. Human evolution. Right, human evolution. Human evolution. All right. Are you ready for human evolution? Yes. All right. Um. Right. Human evolution. Human evolution. All right. So for you to master human evolution, number one, you must know the classification of humans. Classification of humans, number 1. Right. Classification of humans. Right. Number 1, just know that humans belong to kingdom Animalia. Classification. Number 1, kingdom Animalia. Number 2, phylum Chordata. Number 3, class Mammalia. Number 4, order primates. Number 5, to what family do human beings belong to? Hominidae. Right, so it's kingdom Animalia, phylum Chordata, class Mammalia, order primates, family Hominidae. Right, and then number 6 we have genus. The genus for human beings, it's Homo. And then number 7, it's species. To what species do we belong? We belong to Homo sapien. Underlined. So when you write Homo sapien, you must ensure that you underline it. Right, it's kingdom Animalia, phylum Chordata, class Mammalia, order primates, family Hominidae, genus Homo, and then species Homo sapien, underlined. Right, then from there you need to know the similarities that human beings share with African apes. Right, you underline the entire thing. Right, everything. Right. Now, we need to know the similarities that human beings share with African apes. Okay, I'm trying to draw a human being. Right. So similarities that human beings share with African apes. Number 1, we have large brain size. Number 2, we have 2 front eyes. Number 3, we have freely rotating arm. Number 4, we have long upper arm. Number 5, we have rotation at the elbow joint. Number 6, we have opposable thumb. Opposable thumb. And then number 7, we have flat nails instead of claws. Right. So these are similarities that human beings share with African apes. So we have large brain size, 2 front eyes, freely rotating arm, long upper arm, rotation at the elbow joint, opposable thumb, and then flat nails instead of claws. So this is the upper limb. The hand is the upper limb. So if a question says, name the similarities between human beings and African apes in their upper limbs. So obviously it's freely rotating arm, long upper arm, rotation at the elbow joint, opposable thumb, flat nails instead of claws. Right. Those are similarities between human beings and African apes. similarities between human beings and African apes. Right. Now let's go to differences between human beings and African apes. Are we still together? Are we still together now? Yes. All right. Now the differences between human beings and African apes. So we'll have a human, and then you're going to have an African ape. Nice drawing, eh? All right. So this is a human, and then this one is an African ape. Right. Now they ask you what is the difference between the human beings and the African ape, or they say tabulate visible differences between human beings and African apes. Right. Number one, African apes have a small cranium size. Humans have a big cranium size. That's number one difference. So there's a small cranium size compared to this one. African apes have a small cranium size. Humans have a large cranium size. Number one. Number two is that African apes have pronounced eyebrow ridges. African apes have pronounced eyebrow ridges. Humans have less pronounced eyebrow ridges. Number three is that African apes have a sloping face. And then humans have a flat face. And then number four, African apes are prognanthous. African apes are prognanthous. And then humans are non-prognanthous, not less. They are non-prognanthous. The giraffe is appearing there. Lying again corn. Right. So, just a review of visible differences. African apes have small cranium size. Human beings have a large cranium size. African apes have pronounced eyebrow ridges. Humans have less pronounced eyebrow ridges. African apes have a sloping face, whereas humans have a flat face. And then number four, African apes are prognanthous, whereas humans are non-prognanthous. And then number five, African apes have large canines, whereas humans have small canines. Then number six, African apes have a rectangular shaped palate, and humans have a C-shaped palate. Then number seven, oh wait, right, number seven, the foramen magnum of African apes is pushed to a more backward position, whereas the foramen magnum of humans is pushed to a more forward position. Right, so those are differences between human beings and African apes. Right, then now we go to bipedalism. 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 Right. Bipedalism is the ability to walk on two lower limbs. Right, your hands, they are called your upper limbs. Your legs are called your lower limbs. So bipedalism is the ability to walk on two lower limbs. That is bipedalism. The ability to walk on two lower limbs. Right, there are three factors that affect bipedalism. There are three factors that affect bipedalism. In other words, there are three things that makes organisms who are bipedal to be bipedal. Right. Number one is that the foramen magnum is pushed to a more forward position. So foramen magnum is pushed to a more forward position so that the spinal cord can join in vertically. The foramen magnum is pushed to a more forward position so that the spinal cord can join in vertically. That's number one. Number two, the spine is S-shaped. The spine, so we don't say the spinal cord. We say the spine is S-shaped to allow for flexibility. The spine is S-shaped to allow for flexibility. And then number three, the pelvis is short and wide to support the upper body mass. The pelvis is short and wide to support the upper body mass. So those are three factors that affect bipedalism. Foramen magnum is pushed to a more forward position. The spine is S-shaped. The pelvis is short and wide. Right. Now, we need to know the differences between quadrupedalism and bipedalism. When they say tabulate the differences between quadrupedalism and bipedalism, right? Quadrupedalism is the ability to walk on four lower limbs. The ability to walk on four limbs. That is quadrupedalism. Right, so we have bipedalism here, and then we have quadrupedalism. Right, so in bipedalism, the foramen magnum is pushed to a more forward position, whereas in quadrupedalism, the foramen magnum is pushed to a more backward position. And then in bipedalism, the spine is S-shaped, and in quadrupedalism, the spine is C-shaped. And then last one, in quadrupedalism, the pelvis is short and wide, whereas in quadrupedalism, the pelvis is long and narrow. It's long and narrow. So those are differences between bipedalism and quadrupedalism. Right. Then they are going to ask you about, they might ask you about the advantages of bipedalism. What are advantages? What are advantages? Number one is that you have a better field of view. Number one, you have a better field of view. You can see far. You can spot predators from afar. That's number one. And then number two is that movement is easy and efficient. You can move easily and efficiently. Right, number 3 is that your hands are free to carry tools. You can use your hands. Your hands are free to carry tools. Right, they won't ask you more than 3 advantages. Right, now let's come to the evidence of human evolution. If human evolution is really a theory, then there should be evidence of human evolution. Right, evidence of human evolution. Evidence of human evolution. Right, so we have 3 evidence of human evolution. Number 1, we have fossil evidence. Fossil evidence. When we are talking of fossil evidence, we are talking about the transitional fossils. Transitional fossils. Fossils that have characteristics which are common to the ancestor species and the species that follows after. Right. And then number 2, we talk of cultural evidence. When we speak about cultural evidence, we are talking about the use of tools. Then the last one is genetic evidence. The last one is genetic evidence. Genetic evidence. So here we are talking about mitochondrial DNA. So these are 3 evidence of human evolution. It's fossil evidence. We are talking about the transitional species or fossils. And remember, who was this one? Karab. Right? Australopithecus sediba. But we'll go there, it's fine. Right, and then cultural evidence, we are talking about the use of tools. Here we are talking about Homo habilis, handyman, the first one to use tools. And then we speak of mitochondrial DNA when we are talking about genetic evidence. Right, let's explain this evidence further, and then we do out of Africa hypothesis. We look at the phylogenetic trees, and then we are done. All right. Now, when we talk of fossil evidence, we are looking at the fossil record. We are looking at the fossil record. All right. Do you remember the fossil record? Yes. Let's see if you can remember it. See, people, you must close your books. Sure. Can we close distinction material? Thank you. Hey, these ones, they want monetary value now. They want man. There is, it's fine. Yeah. All right. So remember, on the fossil record, you must know the scientific name. Nah? You must know the scientific name. Right. You must know the scientific name, and then you must know the common name, and then you must know who discovered it. Who discovered it, and then you must know where. Where did they discover it? Right. What was the oldest fossil? Right. So the first one was Ardipithecus ramadas. Right. So what was the common name for Ardipithecus ramadas? It was Adi. Right. Who discovered Ardipithecus ramadas? It was Tim White. Where? In Ethiopia. Right. So the oldest fossil, it was Ardipithecus ramadas. Common name was Adi, and then Adi was discovered by Tim White, and then in Ethiopia. Right after Ardipithecus ramadas, we had who? Australopithecus afarensis. Right, Australopithecus afarensis. The common name was Lucy. Who discovered Lucy? Johansen. Where? In Ethiopia. Right. After Australopithecus afarensis, we had what? Australopithecus africanus. So it's Australopithecus africanus. Ne? Africanus. Australopithecus africanus. Africanus. So there are three, isn't it? Right. The first one is Daung child. Right. So who discovered Daung child? It was discovered by Raymond Dutz. Right. Where? In Daung. Right. And then the second one was, okay, Mrs. Bless. Right. Mrs. Bless was discovered by who? Roberts. Who? Where? In the Steekfontein, right? And then the last one was Littlefoot. So Littlefoot was discovered by who? Ron Clark. Where? In the Steekfontein. Steekfontein. Is it jealous or something? Right. Now after these species, Australopithecus africanus, we had a transitional fossil. Right. Who was a transitional fossil? Australopithecus Sidiba. Right. What was the common name for Australopithecus Sidiba? Karabo. Hey, now, why is it doing this? Karabo. Right. Who discovered Karabo? It's Prof. Nipega. Ne? Right. Where? Malapa. Cradle of humankind. Yeah, it's a fossil site. Right. And then after Australopithecus Sidiba, we had who? Homo habilis. So we had Homo habilis. Right. What was the common name for Homo habilis? Handyman. So this one was handyman. Why was he called handyman? Yeah, so the first one to use tools, eh? Right, who discovered Homo habilis? Yeah, it's Mary and Louis Leakey. Right, where?
Tanzania.
In Tanzania. So you need to know all of this. All of it, you need to know it. And I'm glad you do. Right. And then we had Homo erectus. Homo erectus. Right. What was the common name for Homo erectus?
Right man.
Upright man. Right. Who discovered upright man?
Eugene.
It was Eugene Du Bois. Right. Where did Eugene Du Bois discover this fossil?
Indonesia.
In Indonesia. Right. And then the last one was
Sapien.
Homo sapien. Right. Common name for Homo sapien? It was wise man. Right. Where did they discover? Oh, who discovered wise men? Tim White. Where? In Limpopo. Right. That is fossil evidence. So now we are done with fossil evidence. Actually, we are done with paper 2. We've moved. Ngiyadlala. Hayi. Sebashaya izandla bafuna ukulala. All right. Now we come to cultural evidence. But cultural evidence and genetic evidence is explained better in the out of Africa hypothesis. So let's just go straight to out of Africa hypothesis. Right, out of Africa hypothesis. All right, you can tell me, what does the out of Africa hypothesis state? All present day. All modern life forms originated from Africa and migrated to other parts of the world. That is the out of Africa hypothesis. You're free 2 marks. All modern humans originated from Africa and migrated to other parts of the world. Out of Africa. Hypothesis. Right. It states that all modern humans, all the keyword, it's all modern humans originated from Africa. And migrated to other parts of the world. That's out of Africa hypothesis. All modern humans originated from Africa and migrated. All modern humans originated from Africa and migrated to other parts of the world. That is the out of Africa hypothesis. Right. Now, what is the evidence for out of Africa hypothesis? People, this one is very important. It will be there. I feel it. Okay. They will ask you, explain how fossil evidence.
GPS signal lost.
Okay, sorry. All right. Explain how fossil evidence shows the evidence of out of Africa hypothesis. Right. Number 1. Fossil evidence. Fossil. You are going to say fossils of Ardipithecus were found in Africa only. Ardipithecus found in Africa. Only. Right, that is 2 marks. Fossils of Ardipithecus were found in Africa only. Right, number 2. Fossils of Australopithecus and Homo habilis. Australopithecus and Homo habilis were found in Africa only. For marks. Number 3, you are going to say the oldest fossil of Homo sapien. Homo sapien was found in Africa. So this is fossil evidence. Fossils of Ardipithecus were found in Africa only. Fossils of Australopithecus and Homo habilis were found in Africa only. And then the oldest fossil of Homo sapien was found in Africa. So you need to know this fossil evidence by heart. Right, and then now we come to cultural evidence. We are trying to explain that all modern humans originated from Africa and migrated to other parts of the world. Right, so now we have cultural evidence. All right, cultural evidence. Cultural evidence. Right, so now in cultural evidence, we are going to explain how does cultural evidence serve or supports the out of Africa hypothesis. Right, now the use of tools improved human nutrition. And when you look at evolution, the development of tools became more sophisticated as hominids evolve. Right, so the first fossil to ever use tools was found in Africa, and his name was Homo habilis. Right, when we talk about culture, we are talking about the way of life. Right, you are talking about the way of life. Right, so the first man to ever use tools was found in Africa. That is cultural evidence, which means all modern humans really originated from Africa because now tools are used everywhere. Right, then the last one is genetic evidence. Genetic evidence. Right, when we are talking about genetic evidence, we are talking about mitochondrial Eve. Right, so here you are going to say the analysis of the mitochondrial DNA shows that the oldest female was found in Africa, and her name was mitochondrial Eve. Right, so Mitochondrial DNA comes from the maternal lineage, right? Right. So when they were analyzing the mutation on the mitochondrial DNA, they found out that the oldest female was found in Africa, and her name was mitochondrial Eve. Right. That is genetic evidence. Right. And so we are done. We are done. Oh, phylogenetic tree. Aye, aye, aye. We must finish now. Right. Shh. Now let's look at few questions and then, yeah. Remember, the warmer the blankets, the colder the future. Yadlala personal. All right. All right. Now let's look at evolution. Question number 1. Question number 1. Tabulate 3 differences between Lamarckism and Darwinism. Right. Let's look at the differences between Lamarckism and Darwinism. People, are we together?
Yes.
Right. So we have Lamarck and then we have Darwin. Right. So the first difference is that when we look at Lamarck, in Lamarck there is no variation. Whereas in Darwin, there is variation. That's number 1. What is the second difference? Or should I give you 5 minutes to do it and then we do corrections? Aye, nah. What's another difference between Lamarck and Darwin? Yeah, so in Lamarck, no, in Lamarck, not all offsprings are affected there. It's only those ones that are determined to change that change. Yeah, so in Lamarck, individual changes, whereas in Darwin, the whole population changes.
So, can you also say Lamarck says that um, organisms evolve because they want to, and then Darwin says the environment makes them evolve.
Not necessarily, but what we can say is that in Lamarck, characteristics are acquired, whereas in Darwin, characteristics are inherited. Right? Very important. Right? So this is a difference between Lamarckism and Darwinism. Right? Again, I'm recapping. The first difference is that in Lamarck there's no variation, in Darwin there's variation. And then in Lamarck, individuals change, whereas in Darwin the whole population changes. And then in Lamarck, characteristics are acquired, and then in Darwin, characteristics are inherited. All right. So we move on to another question. Sorry. Where is this food? All right. Shh. Now the question is, um, are we reading the question? Right. Ancestors of duck did not have webbed feet. Are you seeing it? So the ancestors did not have webbed feet. In terms of natural selection, explain how the webbed feet could have evolved. Explain how the ancestors of ducks did not have webbed feet. In terms of natural selection, explain how the webbed feet could have evolved. All right, let's do it. There's a great deal of variation amongst the ancestors of ducks. Some ducks had webbed feet, and some ducks did not have webbed feet. When there was a change in the environment, the ducks that had webbed feet survived, and the ones that did not have webbed feet, they died. Ne? Right? The ones that survived reproduced and passed on the allele of having webbed feet to their offsprings. Ne? Then eventually, there's a great proportion of ducks that has webbed feet. Yes, yes. Yeah. So here the competition is not stated, ne? But you can state it. You can say maybe the ducks had to swim because these webbed feet they are used to swim. But since here it's not mentioned, I don't think it's compulsory. Right, and then question number 3. Describe how speciation occurs when a population becomes separated by a geographical barrier. So we've done this one, isn't it? Right, if a population of a single species becomes separated by a geographical barrier, the population will split into two. There is now no gene flow between the two populations. Since the two populations are exposed to different environmental conditions, natural selection will take place independently in each population, such that the two populations become different from each other genotypically and phenotypically. Even if they were to mix again, they would not interbreed and produce fertile offspring. All right, question number 4. Study 3 diagrams, diagram A, B, C, and C, below that shows the mechanism used to explain evolution. Right. Okay. Okay, we've seen the diagram. Let's just memorize the diagram. Here's a diagram. Right, here's a diagram. So we have roots. These are roots. Some they are long and some they are short. And then in diagram B, we can see that the short roots, all of them, the trees with short roots or plants with short roots, what happened to them? They died. Are you seeing it? Yeah, they died. All of them, they died. And then in diagram number C, we can see that the new offsprings are developing what? Long roots. This is obviously natural selection, right? So you can see that there's a great deal of variation among the roots of the plants, right? Some plants have long roots and some plants have short roots, right? When there's a change in the environment, can be droughts, the plants with long roots survive because they could reach groundwater or water table, and the plants with short roots, they die because they cannot reach groundwater. They are no longer there. These plants with long roots, they reproduce and pass on the allele of long roots to their offsprings. And then eventually, there's a great proportion of plants that has long roots. They are called cutty plants. All right. Okay, so it says from a comparison, picture A and B, describe the feature of the cats that have enabled them to survive long periods of hot and dry weathers. The answer there is long roots. Right. And then 4.2, name the mechanism put forward by Darwin to explain his theory of evolution that illustrates in the diagram. What is that? Natural selection. Then they say use the three diagrams to explain the mechanism that you've mentioned. Right, I've already explained it, isn't it?
Yes.
Right, now let's come to question number 5. People, are you following? Or ngiyadlalela nje la? Right, they are saying study the diagram below that shows the change in the number of black and white mice living in an environment with light sand. Okay. Shh. Right, study the diagram below that shows the change in the number of black and white mice living in an environment with light colored sand.
Oh, yes.
So the land is light colored. The mice are white, others
Yeah.
are black. Obviously, the white ones are going to be camouflaged, isn't it?
Oh, yes.
The black ones will be exposed. Mm. Right. 5.1. What phenomena is illustrated in the diagram? Natural selection. Describe how the sequence of events A, B, and C represent the phenomenon. Right. Let's go back. Right, Stini, there was a great deal of variation amongst the mice. Okay? Some mice were white and some mice were black. Are we together? Right, when the predators came, the white mice survived because they were camouflaged by the light colored sand, and the black mice died because they were exposed by the light colored sand. The mice that survived reproduced and passed on the allele of being white to their next generation. Then eventually, all the mice, or no, there was a great proportion of mice that was white, isn't it?
Yeah.
Right. 5.3. Will the next generation of mice population more likely be all white or all black? The answer is all white. Right, question number 6. Study the diagram below showing a process of evolution. Diagram 1, 2, and 3 shows the sequence of events that occurred in the rabbit population. The rabbit population. Right. Okay. Okay, so now there's a river here. We know a river is a geographical barrier, isn't it? Right. This river was found on the side. People A. And then there was gene flow, which means these rabbits were able to interbreed, isn't it? And pass on their genes. Right, in diagram 2, I don't know whether that is called river capture or what. Active. Right, or stream pirates. Alright, now listen game. This river started bending downwards. Are you seeing it? Yeah, so it started bending downward and it separated the populations. Now we have population A and population B. Right, in diagram number 3. Right, in diagram number 3, there is now no gene flow. Right, and since there's no gene flow, obviously they cannot interbreed anymore, and they are exposed to different environmental conditions. Natural selection took place independently and they became different from each other. Right, so we ended up with species X and species Y. Right, 6.1, name the evolutionary process represented by the sequence of events shown in diagram 1, 2 and 3. It's speciation. Speciation. Right, describe the process stated using the diagrams above. So we are going to say right, there was once one population of rabbits, né? Right, this population of rabbits was separated by the river. The population split into two. We had population A and population B. Since these two populations are exposed to different environmental conditions, since these two populations are exposed to different environmental conditions, did you mention gene flow? No. All right, let's start afresh. Right, we said that there was once one population of rabbits, né? Then this population was separated by the river. The population split into two. We had population A and population B. There was no gene flow between the two populations. And since the two populations were exposed to different environmental conditions, natural selection took place independently in each population, such that the two populations became different from each other genotypically and phenotypically. Hence the formation of species X and species Y. Even if these species can mix again, they would not interbreed and produce a fertile offspring. Right, question number 7. Question number 7. Right, the diagram below represent the evolution of horse compiled from a fossil record. Right, so we have a modern horse, and now let's look at their legs and their toes. Now 7.1. How many times is the modern horse taller than the earliest ancestor? Now this is an earliest ancestor. Né? They are saying how many times is the modern horse taller than the earliest ancestor. So you are going to say 1.6 divided by 0.4. Then you are going to get an answer. What is the answer there? Yeah. No, it's 1.6 divided by 0.4. It's
It's full. It's full. It's full.
It's 4. Okay, which means the answer is 4 times. So we said 1.6 divided by 0.4. Then you see the answer is 4 times. Right. And then 7.2. What happened to the appearance of the feet of the horses over time? What happened to the appearance of the feet? Right. So now we can see there's feet here. Toes. Eh? And then over time, they started doing what? Shrinking. Isn't it? Right. Paleontologists suggest that the ancestor horses lived in forest, but modern horses lived in open grass. Explain one way in which the modern horse is adapted to open grassland. One way in which the modern horse is adapted to live in a grassland. Here's a sorry. It is used. Eh? What is she saying? Yes, good. It has wolves. Right. Now, question number 8. The last one. Say amen. All right. So the red-bellied snakes and the green tree snakes are predators that sometimes feed on the cantods, no totmos, frogs, that contain a toxin that may kill them. So these snakes, they feed on the toads that has toxin which may kill them. Right? So the snakes consume the toads by swallowing them whole. So they just eat them whole. A decrease in the average jaw size of the snake has been observed over a period of 70 years. Right? Which means these snakes, the red-bellied ones and the green ones, they had a larger jaw size. So, or rather, maybe there was variation. There were some with a larger jaw size and there were some with a smaller jaw size. Right? The ones with a larger jaw size, they are the ones that swallowed the thoughts that had toxin. So all those that had a larger jaw size, they did what? They died. All right, let's look at the questions. Okay, the question is define punctuated equilibrium. Can you see how many marks? 3 marks. Define punctuated equilibrium. Right. So we are going to say punctuated equilibrium. It's a hypothesis that explains the speed at which evolution takes place. Then you further explain that it states that evolution involves a long period of time where there is no change and a short period of time where there's rapid change through natural selection. Right. And then 8.2. What characteristic of toad species protect it from the predators? Toxin. Contains toxin. Right. Now 8.3. Explain how the change in jaw size helped the snakes to survive. So they are saying that the snakes consume the toads by swallowing them whole. A decrease. So there was a decrease in the average jaw size of snakes. So how did that save them? The smaller the jaw size, So as the jaw size decreased, it meant that the snakes could no longer swallow the thorns, isn't it? And that made them, or that protected them from the toxin that is produced by the thorns. Yeah, because they were swallowing the thorns as wholes. They were killed by the poison. This is sport. Right now, 8.4. How would Lamarck have explained the development of small jaws in the snakes? All right. So I think Lamarck could have said, okay, so the snakes saw that the thoughts are toxic or they have toxin. Ne? So they did not use, or rather, originally, snakes had a larger or a bigger jaw size. They did not use their jaw size more often to swallow the thoughts. As a result, their jaw size decreased and became smaller. And the characteristics of small jaws was then passed on to the next generation. I just want something that has to do with human evolution, then we are done. Okay, let's do this one. 29, the last one. Last one, ne? Last one. All right. Question 29. It says, Professor Liebega and his colleagues studied the fossils they found in the cave at the Cradle of Humankind. The skeletons from the cave are ranked among the most complete finds to date. The adult female Australopithecus sediba was remarkably well preserved, and some of the following characteristics were identified. Right, so Australopithecus sediba had a small brain size, was bipedal, smaller canines, projecting nose, and a smaller body size. Right. Question 29.1. Write down 3 characteristics from
ladder, then you just know that steps, you know the steps, yes, you just know that that is called punctuated equilibrium. Then the phase of meiosis, during which homologous chromosomes separate and start moving towards the opposite poles. Homologous chromosomes are being separated and then they move to opposite poles. So they are looking at the next phase after this, meaning that they have to separate, they move to the opposite poles. So it is anaphase 1 because they are saying homologous. Don't just say anaphase, it must be anaphase 1. The defect in the cell division that lead to Down syndrome. So how do we call that defect in the cell that leads to Down syndrome? We call it non-disjunction. The structure that is made up of 2 chromatids joined together by the centromere. Structure that make up 2 chromatids joined by the centromere. So it means that structure made up of 2 chromatids. These are 2 chromatids and then joined by the centromere. How do you call this? It is a chromosome. Then they are saying that an explanation of something or for something that has been observed in nature and which can be supported. When they say supported by facts, laws, tested hypothesis, then automatically it can be hypothesis. It is a theory. Then they are saying that, remember when they're asking you this, it is either theory or a hypothesis. So if they say tested hypothesis, definitely the answer can be hypothesis. The answer is the theory. We go to the next question. Guys, if there is anything related to the sound and the sound is not clear, you let us know. Maybe there is echo, maybe you don't hear me properly, something like that. Or I'm too fast, you let me know. Yeah. But because you want to finish this in 2 minutes at least, yeah, the similar structure of different organisms that suggest they have a common ancestor. Similar structures. Whenever you say similar, it is homo. starts with the word homo. So structures that structures means logos. When you say homo, similar, and then structures is logos. When you say logos means structure, so it is homologous structures. Then they're saying that complete set of chromosome in a cell. How do you call that complete set of chromosome? Is a genome. So you guys, you confused, sorry, did I say genotype? You confuse three things: genome, karyotype, and a genotype. Genotype is a description of organism in terms of genetic composition or in terms of gene. And then when you talk about the complete set of the chromosome in a cell is a genome. And then when you talk about the arrangement, it must have the word arrangement. The arrangement of chromosomes, we talk about size and talk about the structure. That's when you talk about the karyotype. I think I'm clear there. A barcode pattern formed from DNA is DNA profile. Then the bond formed between two amino acids. Guys, this bond is called peptide bond. Please take note of this. We don't have what you call polypeptide bond. It's peptide bond. If there are many bonds, we call them peptide bonds. We only add the word polypeptide if we are talking about a chain. So if you don't add the word chain, then you can talk about poly. So it's polypeptide chain, peptide bond. Is it clear? Yes, please, if you add the word polypeptide bond, is wrong. You will get a zero with big eyes, and those eyes are looking at you. So make sure that you don't say polypeptide bond. It's peptide bond. Okay, we continue. The representation of the number shape, here is the answer. Representation of the number, shape, and arrangement of chromosomes in the nucleus of a somatic cell. So is what you call the karyotype. Opening in the nucleus, the nuclear membrane that allow messenger RNA to leave the nucleus, nuclear pore. So we have got it. I'm not writing the words because we want to finish up this so that we go to the major section. They're saying that a tentative explanation of a phenomena that can be tested. A tentative explanation of a phenomena that can be tested. You see now, it's a tentative. So it becomes a what? A hypothesis. Yes. Distribution of species in different parts of the world. Different parts of the world is geography. Different parts of the world, but distribution of species, so it must be linked to bio. So it becomes biogeography. Then they're saying that chromosomes that carry same set of genes. Chromosomes that carry same set of genes. How do you call those chromosomes? Homologous chromosomes, meaning that when they look at it, they look like that. They're saying two or more alternative form of a gene at the same locus. Two or more alternative form of a gene at the same locus. What does it mean? Two or more alternative form of a gene. It means that either this or this, for example, or capital R, small r, something like that. We call them alleles. So they call them what? Alleles. The structure responsible for pulling the chromosome to opposite poles of animal cell. So opposite poles. You remember when you are like this, how do you call that structure which pulls the chromosomes to opposite poles? So we call it spindle fibers are the one which pull. Yes. Then they're saying that a phase in the cell cycle that occur before cell division, interphase. Cell cycle, a phase in a cell cycle. Yes, it's not. They're not saying the a phase in meiosis. They're saying cell cycle. So it means that you talk about interphase. So now let me ask you a question. Who has got all of them? 26 out of 26. I hope you are getting 26 out of 26. Yes. I'm seeing people are getting answers correct here. Thank you very much. Thank you very much. Sir, the sound is making it hard to concentrate. Let me see. I don't know the sound. Which sound? Is there any other sound behind it or something like that? I don't know. Let me know if there is something like that. All right. Let's continue, guys. Let me know if there is any awkward sound behind my what my speakers or something like that. Yes, so that I can try to work upon it. All right, let's continue, guys. They're saying that let me see how many people got it. Or let me check how many people got all of them. Someone is saying that it's gone now. It's gone now. Oh, thank you. All right. Let's continue. They're saying that the grammatic representation showing the possible evolutionary relationship among organisms. The grammatic representation, yes, possible evolutionary relationship. Here we are talking about phylogenetic tree. Yes, yes. The type of vision shared by two humans that allow for deep perception. Stereoscopic vision, yes. We call it stereoscopic vision. A genetic cross involving two characteristics. We call it dihybrid cross. They are saying that because two means die. Yes. A genetic disorder characterized by absence of blood clotting factor, hemophilia. Present day distribution of organisms. Present day. How do we call about the present day distribution? How do organisms being distributed? So we can call it biodiversity. Yes, we can call it what? Biodiversity. Then they're saying that the nucleic acid that the nucleic acids that carry amino acid 2, the ribosome, transfer RNA. Then they're saying that the natural shape of DNA, double helix. Guys, don't say double helical or helical. We will cross it. It's double helix. It can't be something else. That's it. Double helix. Section of DNA that code for a specific protein. Section of DNA that carry for a specific protein. Okay, let me read again. Section of DNA that carry for a specific protein. How do you call that section of DNA? Check. They're looking at something like this. But this section, how do we call those sections? We call them genes. Yes. And then I'm saying evolution with a long period of no change followed by short periods of rapid change. Don't forget that. It is drawing it ugly. Remember, it is long periods of no change, rapid periods of change. Long periods of no change, rapid periods of change, like that. So, sex chromosome of organisms. How do you call that? Gonosomes. The descent with modification. How do you call descent with the modification? Yes, how do we call descent with the modification? Let me see in the comment below. I want to know number 37. Yes. I want to know this answer. Then in the comment, please put it in the comment. Just write 37 and then you put the answer there. I want to see if you're gonna go get it correct. A group of organisms of the same species in specific habitat. Group of organisms the same species, yes, but they live in a specific habitat, becomes a population. The family to which humans belong, hominidae, not Homo sapiens, hominidae. Human disorder, human disorder caused by non-disjunction, Down syndrome. The stage of protein synthesis during which messenger RNA is formed. How do you call that stage of protein synthesis during which DNA is messenger RNA is formed from DNA is transcription. Then they are saying that the type of variation in humans with no intermediate, we call it continuous variation. Guys, let me just explain this just a little bit. Whenever they talk about examples of continuous variation, anything which we are born and it keeps on changing, we call it continuous variation. Skin color, size, body size, size of the shoe. Check, when you are born, you are putting on size 1, if not -1. I don't know if there is such, or 0. whatever. But now you are putting on size maybe 47. Big, such a way that is bigger than you. So it means that it keeps on increasing. So it changes. What about your blood group? If you are born blood group A, you cannot change to blood group B. Never. So blood group becomes discontinuous, while the size of the body becomes what? Continuous. The size of the color, continuous. Yes. Oh, I'm just emphasizing this because I might not give you examples when you reach there, when you're trying to do highlights. The type of inheritance where dominant allele masks the expression of a recessive allele. Complete dominance, meaning that if it's capital T small t, this one is completely dominant over this. Therefore, complete dominance. Variety of living life forms that exist on Earth is biodiversity. Let me check if there is any comment. There's someone is saying discontinuous. Darwinism. 37 is saying Darwinism. Thank you. J punctuated curriculum. I don't see people telling me the answer. A discontinuous. It's only one person who did J. Yes, J. Since you have tried to answer, you must remind me after the live. Yes, I'll give you 20 rand airtime, ne? Yeah. However, that's not a correct answer, but you have tried, ne? Yes, I'll give you 20 rand air time. Yes, but I know how I'm gonna find out that it's you. Don't worry about it, ne? I'll find out. Yes, so 20 rand air time, Jace, is for yours. Congratulations for that. All right. Let's continue. So the type, if you go back here, we're saying the modification by descent J. The answer is supposed to be let me leave it. Let me leave it. Let me see if I can have some more. Okay, someone is telling me, well, leave it to see that if there are some people who are going to participate. But I'm not saying that I'm going to give you a gift. All right. They're saying that a variety of life forms that exist on Earth, that is called biodiversity. They're saying opening in the skull through which spinal cord enters. We call it foramen magnum. The testable statement that can be rejected or accepted, it is a hypothesis. Total disappearance of species from the Earth, we call it what? We call it extinction. They are saying that a segment of chromosome that code for a particular characteristic, should I leave it also? A segment of chromosome that code for a particular characteristic, we call it a gene. We talked about it. Ability to walk on two limbs, we call it bipedalism. The organelles in the cytoplasm, which is a site for protein synthesis, is a ribosome. The name of the bond that formed between two amino acids, I explained this. It is peptide bond. Then we are saying that the process by which DNA molecule makes identical copy of itself. Then process by which DNA molecule makes identical copy of itself. Yeah, we call it, should I leave it there? So it's DNA replication. Name the phase, the process where homologous chromosomes pair fail to separate during meiosis. Homologous pair fails to separate during meiosis. How do we call that? We call it, they're saying the process is non-disjunction, fail to separate during meiosis, but which phase? Is anaphase, ne? Yes. So we call it what? Non-disjunction. Permanent disappearance of species from the Earth is extinction. They're saying that testable statement that may be rejected or accepted. I don't know why they repeated it, but is a hypothesis. The type of nucleic acid that carry amino acids, we call it transferrin A. The selection of breeding, the selection and the breeding of organisms with the desirable characteristics by human. How do we call it? You select the breeding and then desirable characteristics. Since this selection, then therefore we can call it selective breeding. And then they are saying that an allele that is not shown in or expressed in the genotype, in the phenotype when found in heterozygous state. For example, capital T small t, this allele that we are talking about. So you call it a recessive allele. Sudden change in the sequence or the nitrogenous base of the nucleic acid, we call it gene mutation. Explanation of an observation that is supported. That's what is supported by facts, models, and so here they didn't talk about tested hypothesis. But the moment they are supported with this, then it becomes a theory. They are saying that the breeding of organism over many generations in order to achieve desirable phenotype. The breeding, when you breed, yes, so it is basically you are doing artificial selection or artificial selection. Then they are saying that the type of sugar found in messenger RNA, in RNA, is ribose sugar. They're saying that the type of evolution with long periods of time when species do not change and short periods of time when there is a rapid change. So we call it that type, we call it punctuated equilibrium. We talked about it. And then he's saying that the mineralized remains of organisms that have lived in the past. How do you call that? We call them fossils. And then the hypothesis which support the migration of ancestor from point of origin. We call it out of Africa hypothesis. Yes. So here is how do we say this? We say that this is comparative anatomy. Let me just show you just something small before we go there. Yeah, this is what they are. Another comparative anatomy. Modification by descent. If you look at our distinction material, yeah, it gives you the perfect answer there. Yeah, sometimes they ask you modification by descent, which means comparative anatomy. Sometimes they ask you these structures, whereby you're supposed to tell us that these structures are called homologous structures. So if they ask the process, then it's going to be modification by descent or comparative anatomy. But if they ask you the structures, which is under here, we call it homologous structures. That's why I said, let me know the answers people are giving out. All right, 62 is a ribosome out of Africa hypothesis. Thank you, biotechnology. Lucia, okay, thank you. Yeah, guys, I think now we have got it. Now, let's go to the section whereby what to focus on DNA, what to focus on other topics, so that, guys, we don't lose it. We have to look for a distinction today. Yes, the distinction must click. Yes, must click. Yes. I'm going to make it a little bit 2 hours. So we are starting our 2 hours because the previous we are just going through, yeah, and then some technicalities it came up, and then yeah. So now let's start our 2-hour journey story. All right, guys, on DNA, what are you supposed to focus on when you go to DNA? Let me just remove this. Sorry. Yes. So, yes. Hi. Sorry, guys. Let me just do this. Yes. Yeah. We have that. We have that. I have that. Yeah. Now I think we are done. Let's start now. Guys, on DNA, don't look at that diagram. I'm not talking about the diagram. Guys, on DNA, there are some things which you're going to ask you, and then if you don't know them, you are gone. Yes. Okay, now let's see. Guys, if they ask you describe the structure of DNA, what do you say? Describe the structure of DNA. Structure of DNA. Guys, when talk about the structure of DNA, look at the characteristics of DNA. So I think that's done. The characteristics of DNA is double helix is made up of 2 strands. It's made up of the nucleotide. It's made up of nucleotide, yes, which are made up of phosphate, the oxiribose sugar, and nitrogenous bases. You talk about the nitrogenous bases, talk about all of that. Yes. Challenge. Don't go to the paper without knowing DNA replication. Yes, don't go to the paper without knowing translation. Don't go to the paper without knowing transcription. And then describe the description of the structure of DNA. So one will come. I won't tell you which one, but one will come. You'll come. We're gonna discuss the paper after. You'll know. And then now, if they ask you a question that give the difference between DNA and RNA, that one is simple. You can talk about everything you know. Make sure that you draw the table. Don't leave it hanging like this. You want a complete table. You mentioning this, you writing like this, and you say DNA, and the other side you say RNA. Guys, put DNA, put RNA, close the table. If you don't know anything, at least you got a mark. Yes. If they ask you to tabulate, draw a table, put the title, close the table, leave. If you don't know anything, yes. If you know something, then write something, yes. I'm done with that. But now the challenge comes in when they ask you that give the difference between DNA nucleotide, nucleotide, and RNA nucleotide. Here, you only look at the nucleotide of DNA, the nucleotide of DNA and the nucleotide of RNA. And there are only two differences in there. DNA has, don't talk about the phosphate because it's the same. DNA has deoxyribose sugar and RNA has ribose sugar. And then DNA has thymine, RNA has uracil. Done. If they ask you about what? About the nucleotide. Is it clear? If they ask you the difference between DNA replication and transcription, you have to know the differences. DNA and transcription. We're going to spend a few minutes here. I'm only going to focus on the things which you fail. Which you fail is what I'm going to focus on. Yes, too much. Then we are saying that DNA, you talk about DNA, in this case, in DNA replication, two strands act as a template. The other side, you talk about one strand transcription, one strand act as a template. Guys, only know at least 3, but we want 2. In most cases, we want 2. We won't ask you more than 2, the differences. Don't go to the paper. If there's a lot, just know few things. Don't on each, at least go have a dose on each, whatever I'm going to talk about. Yes, then you'll be able to make it. Then we are saying that yes. Then another thing which you go off is about the mutations on DNA. About the mutations on DNA. So when they ask you about the mutation, let me just use my book here so that you understand. Excel in life science, distinction material. Here, let me just, I don't want the structures. Okay, I don't want the structures. What I want is this. What I want is at the end here. Yes, at the end, the mutations. Yes. Guys, let's answer this question. That what will happen if a mutation changes triplet number 1? Triplet number 1, it's this, from this, the same thing. From this to this, it means that this has been changed. Is it clear? You changed G to A. So now what you do when they ask you such a question, you first identify what is here. It is a codon. What is given here is DNA. Therefore, I have to convert this into messenger. Remember, we say that. Let me do like this. Yeah. Remember, we say that when you talk about code, it means DNA. DNA. When you talk about codon, it means messenger RNA. 3 bases on messenger RNA. When we talk about anticodon, we mean 3 bases on transfer RNA. So now the question is asking it means that it's changing from DNA to codon. It means that it's talking about messenger RNA. Is it clear? Yes. So now what you do is to change this into messenger RNA. So change this into messenger RNA. So when you change into messenger RNA, this is gonna be A goes with U, T goes with A, G goes with what? G goes with the C. So I'm gonna CUA. I come here, look for AUC, AUC, where is it? US is triosine. So this is triosine. UCA, isn't it? Then what about this? Now I change it again. Also is U. It's gonna be A. It's gonna be U. UAU. What does it mean? UAU is also triosine. So it means that a change from this to this, yes, will lead to this USC, from USC to UAU. That's the first mark. It will, you converting this into codons. You understand? Into codons. Is it clear? Which will code for the same amino acid, triosine. Therefore, there will be no change in the protein. Why? Because it's the same amino acid which is being coded for. Can I repeat it? I said, when they bring such a question, I know when they bring it, and I know it will be there. When they bring such a question, students lose these marks. Change in A, this first find out changing, because here it might be tRNA and here is codon. So you change into the codon, which is messenger RNA. Is it clear? So change from ATG basically result in this, and the change in this result into this. Is it clear? And these two, they code for the same amino acid. Therefore, it does not code for a different protein. It's like me calling you, let me check here. It's like me calling you Lerato, Lo, Lailo, whatever. Ne? Sorry if I pronounced you wrongly. Ne? I can call you Lerato, or I can call you whatever, ne? The second name. I don't want to pronounce it badly. Ne? Yes. So if I call you the other way around, you say yes, sir. When I call you with another name, you also say yes, sir. You understand? So it means that they are coding for the same amino acid. However much the codes are different. Is it clear there? Yes. Then, what about base triplet number 4 from, yes, base triplet number 4. So this is the first instance. The second instance, what will happen to a protein if the base triplet number 4, ATA, is changed to ATT? So ATA, I have to convert it because the DNA, I need them to be in messenger RNA. ATA is going to be, T goes with A, U, and then this one is going to be U. So now I say that this will change into this. Okay, this will code for this, and this will code for this. Cool. That's point number 1. But what does this code for? AAU. AAU is valine. What about UUU? UUU, is it valine? No. Where is it? Hey, do you have UUU? No, man. It's not UU. Yeah. What am I doing? It's AAA, ne? It's AAA because T goes with A, T goes with A. So it's AAA. So what am I looking for? Is the AA is proline. So it means that if you look here, this one code for valine. While this one code for proline. You see? So it means that these two, they are different. Is it clear? So it means that even the protein will change. It's like calling you Carabo, and then I call another. I call again Cabero, and Cabero is different from Carabo. You see? Yes. So it means that now this will code for this. This will code for this. Of which this codes for valine, and then this you write this in full, that AAU, ne, which code for proline. Therefore, a different protein will be formed because of the different amino acid. That's the scenario number 2. Scenario number 3. Yes, one scenario will come. Ne? Explain what will happen to a protein if DNA base triplet, or if DNA base has changed. Yes. There, they have not talked about you have to interpret it automatically. If the DNA sequence has changed, one base has removed or has been replaced, whatever, it will lead to a different sequence of messenger, which will lead to a different sequence of transferrin A. yes, which means a different sequence of amino acid. You understand? Hence a different protein. Different sequence of DNA will lead to a different sequence of messenger RNA, which leads to a different sequence of transfer RNA with its amino acids, which leads to a different protein. That's the third scenario. The first scenario, it might not change the protein, therefore the protein is not going to change because the amino acids are being coded by different codes as we saw it. The second scenario, they can code for a different amino acid, meaning that valine and proline, for example, therefore a different protein. Or they can ask you what will happen if DNA base sequence changes or messenger RNA sequence changes. So you start from messenger RNA sequence. If it changes, it will lead to a different sequence of transfer RNA with its amino acid, hence a different protein. Guys, don't go without knowing those things. Lastly, on DNA, it's this. Let me just use this example, but you still have questions, ne? You still have questions we can use. Please don't go to the paper without knowing that, ne? Someone is telling me T, they have given you T or C. Just in mind A and C. Yeah, so guys, make sure that you get the things right. Yes, if you have any challenge, let me know, ne? Yeah. Lucy, Lucia. Yes, you're saying adenine John. Yeah, I'd made a small mistake, but yeah, I saw it when I was trying to. Yeah, I'm also human. I can make a mistake, ne? Yeah, but when I do it, ne, sometimes when you talk, you can talk about something else, and then what in mind is something different, ne? Yeah. All right, but I'm sober today, ne? I'm sober. I don't drink and smoke, ne? So I'm sober. All right. All right, let's continue to the next. Guys, don't go without knowing how to label these things. What is this? It is amino acid. What is this? Is ribosome. What is this? What is this? It's transfer. You see, it is a T. Guys, check. It's a T. tRNA. If you don't know it, try to see if that T will come up. Check. You see? It comes out. It's a T. So it's a T transfer RNA. Guys, this is not a protein. It's a polypeptide chain. Protein is 50 amino acid and above. Is it clear? So this is DNA molecule. This is messenger RNA. This is the nuclear pore. This is the nuclear membrane. Please go when you know how to label these simple things. Is it clear? Yes. This is the transcription. It occurs in the nucleus. This is translation. It occurs in the cytoplasm. Therefore, if they ask you what is this space, is cytoplasm, and what is this space, is the nucleus. Ne? Yes. Lastly, but not the least on DNA, so that we go to meiosis. Two things on here. The first thing is if DNA is 30%, if adenine is 30%, what is the ratio of guanine to cytosine? What's the ratio of guanine to thymine? Let me just say like that. Guys, you have to know how to do the base calculation. A goes with the T, C goes with the G. If this is 30%, automatically also this is 30%. So 30 plus 30 gives me 60. So remember, this is percentage, ne? Percentage means 100. So 100 minus 60, it gives me 40%. So this 40% is for these two. So divide by 2, so it's going to be 20, it's going to be 20. That's not the answer. They're saying the ratio. So it's going to be G to T, whereby G, where's G? G is 20 and then T is 30. So I divide by 10, so I get 2 here and then I get 3 here. You know what you're gonna do? The first objective we're gonna say that 3 to 2. Because when you saw 3 and 2, you just say that it's A. And the last one is gonna be 2 to 3, because you don't want to read everything. Make sure that you read everything, because there is most correct answer. I remember in paper 1, whereby they gave a correct answer halfway correct, and there is the most correct answer just down. You understand? We always try to trick you to see that, yeah, you are off. But I know since you are here, you won't be off. Ne? Yeah, we are here to kill the paper, not the paper to kill us. Ne? Yeah, tomorrow we will make it. We will make it. We'll make it. I know. All right. Yeah, if do you have any question, let me know. Let me know if you have any question. Let me know so that I smash that question without. That's number one. That's number one. The last thing which I need to talk about is about profiling. This profiling. Someone might say that profiling is easy, man. Yeah, I know that profiling is easy, man. But there is a time when they are talking about comparing the paternity test. Ne? Let me see if this question is about paternity test. Identifying the criminal or this. Okay. Sample DNA sample. Okay, let me just read it. Detective investigating the scene of broken window. Suspected blood was found in the criminal identified. No. It won't give me what I want. Blood. Broken. No, it won't give me what I want. Okay, now let me design my question so that now you understand what I'm saying. If they say that two guys they are crying, or they are saying that okay, you have a boy, and then you have uh, two guys, ne? And these guys they are saying that they are the is the each is saying that is the father of this boy. But now the mother doesn't know who, because this mother slept with these guys, ne? These guys are the same day, ne? You understand? So now we don't know who is the father of this boy. So what you need to do is, if this is the DNA profile of the boy, guys, listen until we end this, ne? Because sometimes this question, when it comes, you are off, ne? Okay, if this is the profile of the child, yes, if the profile of the child, I know DNA profile is very easy, but once it comes to this, you lose the marks. Yes, you lose the marks. Okay, this is the boy, ne? And then now you have the profile of the mother. You have the what? The profile of the what? Of the mother. You have the profile of the mother. Is it clear? Yes, you have the profile of the what? Of the mother there. And then now you have the first profile of the guy. You have the first profile of the guy. You have the first profile of the first guy. You have the first profile of the first guy. Ne? You have the first profile of the first guy. Yes. And then you have the second profile of the second guy. You have the first, you have another profile of the second guy. You have another profile of the second guy. Yes. Yeah. So now they ask you, this is the mother, this is X, this is Y. Is it clear? Who is the father of this guy? And after that, they can ask you to explain, and they say 4 marks. Explain 4 marks. That's it. That's what I'm saying. That's where students go off when it comes to this. Listen carefully what you're supposed to say. Why did I divide with the 10 so that I get the simplest ratio? Ne? Yes, I divided by 10 so that I can get the simplest ratio. Yes. To simplify the ratio, yeah. Yes, correct. What you said is correct. I divided by 10 so that I can get the simplest ratio. Equilibrium, I will reach there, don't worry. So, guys, look here critically. What you need to do here is to first, because I know the mother. So take all the bars which correspond with the mother. So this correspond with the mother. Yes. This bar correspond with the mother. Yes. This bar correspond with the mother. So it means that, let me just use a different color so that we see exactly. So this bar is out. This bar is out. This bar is out. So now the remaining bars, not that one. I'm saying this bar is out with the mother, this bar out with the mother, and then this bar is out with the mother. So the remaining bars, these remaining bars, this, and this, it must correspond with one of the guy. If there is one bar which is not corresponding, then that's not a father. Is it clear? Yes. Now I come here. I look for this. What does this one correspond with? This. If there is no one who is corresponding with it, then is out. You understand? Then is what? Is out. So this bar correspond with the with this, ne? Yes. So it corresponds with that. This one is a little bit down. And then this bar corresponds with that. This bar corresponds with this and this. So however much this one has one which is corresponding, but this one and this one did not match with him. So the remaining bars, you see that all the remaining bars, yes, have corresponded with this guy X. Therefore, guy X is the father. Explain your answer. Why are you saying that X is the father? X is the father because the bars of the boy correspond, the remaining bars which of the boy which do not correspond with the mother correspond with this guy X. All the remaining, the word all must be there. All the remaining bars of the boy, yes, after matching them with the mother, correspond with guy X. Is it clear? Because they always ask you this. Yes. And then students tend to go off. These bars must correspond with the mother. The remaining bars must correspond with the father. If there's one bar which is not corresponding, we will ask you, where did that boy get that gene? You understand? Remember, we get these chromosomes as a sperm and ovum. Where did that boy get that gene from? That trying to mean there's a mutation? So that's why all of them must correspond with the father, so that we conclude that that gentleman X is the father of that boy. Yeah, guys, unless you have any question concerning about describe the process of translation according to the messenger RNA, the transfer RNA with the complementary anticodon match according to the messenger RNA, the transfer RNA with the complementary anticodon, yes, bring the required amino acid to the ribosome. The amino acid is joined together by a peptide bond to form a required protein. Guys, those are just processes I'm not going to go through. Maybe I can ask a question. What is DNA replication? When does it occur? When does it occur? When does it occur? It occurs during before the cell divides, during interphase. Where does it occur? In the nucleus. Why does it occur? To double the chromosome number. Yes. How does it occur? That is the process of DNA replication. So there are so many questions. I'm not going to go through them, but make sure that you know those processes. Unless you have a question about DNA replication, about DNA code of life, let's go to meiosis. Yes, when you talk about meiosis, guys, there are some things which you need to talk about. You see, this is a transfer RNA. It makes it. You see, this is amino acid. This is transfer RNA. We said it. This is a ribosome. This is messenger RNA. Sometimes it's up, sometimes it's down. This is what? Codons. Hey guys, how many? This question. Two questions. The first one is how many codons in 12 bases. So remember, 3 bases is equal to 1 codon. So it means that 12 divided by 3, so makes them to be 4 codons. How many bases, how many amino acids in the 4 codons? It's gonna be 4, because each codon code for 1 amino acid. You understand? How many bases code for 5 amino acid? Remember, each amino acid is coded by 3 bases. So I'm gonna say 5 times 3, which gives me 15. You understand? So you have to know how to interpret that. And then let's go to meiosis. Yeah, we try to explain this. If they say left to right, make sure that you translate it left to right. Ne? Yes. The first thing you need to know about meiosis are the phases. But I'm not going to go through the phases. Use IPMAT, you'll be able to know which phase is it. But guys, something is itching me. And that itch makes people to lose marks in meiosis. Number 1, identify phase in diagram 2, diagram 1, diagram 3, and diagram 2. Guys, whenever they ask you about the phase, please turn your paper if these centromere, the centrioles, they are not on the sides. So turn your paper to be like, yes, you see, it must be, I don't know, I think this is east and this is west. If I'm not a geographer, I hope this is the correct. Yeah, but it must move east to west, east to west. It must leave what? East to west. Don't leave these, they must be in east to west, so that you identify the perfect what. If it's not straight with a paper, turn it, because if you turn it, you will be able to identify the actual what? The actual phase. So in this case, because there is space here, it means that is anaphase. But because it is single, therefore is gonna be anaphase 2. So it's anaphase 2. What about here? The chromosome, you see they're in the equator. So it's gonna be metaphase 1. Yes, because the chromosomes are double, you see, therefore it's gonna be metaphase 1. If they're single, then it's gonna be meiosis 2, which is you identify the phase and then you put the word 2. That is the first thing which you need to know before you go for the paper. The second thing which you need to know about, before I tell you what to focus more on this, is the number of chromosomes. How many chromosomes are found in diagram 1? You will find out someone saying 46. Did they say this is a human cell? No. If they don't talk about human cell, don't talk about 46. Don't talk about 23, because they didn't talk about human cell. How many do you see? 1, 2, 3, 4. It's 4. How many chromatids found in diagram 1? It's 1, 2, 3, 4, 5, 6, 7, 8. It's 8 chromatids they're talking about. How many chromosomes will be found at the end of division of diagram 2? Yes, because this is meiosis 1, therefore it's gonna be, since there are 4, therefore it's gonna be 2. Yes, that's the number of chromosomes which will be found at the end of meiosis 1. Now the question comes, how many chromosomes will be found in diagram 2 if this was a human cell? Now they said if this was a human cell, because this is meiosis 1, human cell, therefore there will be 46, or there will be 23 pairs. Yes, I think I'm clear there. That is point number 1 about meiosis. Point number 2 is about the karyotype. This is where also you lose marks. Describe the structure, describe the karyotype. It's fine. You can describe that from this karyotype is for a male. If you see, this is for a male, this is for a female. Because here, the chromosome number 23, they have the same size and shape. While this one is big, one is small. Yes, therefore it means one is X, one is Y. But now, if they ask you to describe the karyotype, from is made up of 46 chromosomes, which are arranged in pairs. From pair number 1 to pair number 22, we call them autosomes. The 23rd pair is called the gonosome. The gonosome has X and Y. If it is a male, has X and X. If it is a female, the X is longer than Y. So you describe what you see. But if they ask you how many autosomes are found in a P, please guys, don't just say that is 23. Sorry, don't just say that is 40, 44, or is 22 pairs. No, count them, because sometimes maybe chromosome number 6 we put them 3, whereby you say 1, 2, 3, 4, and you don't see that, you don't recognize. Look at chromosome number 21, but it's better to count them 1, 2, 3, 4, 5, because you'll find out that you won't just ask you a question, how many chromosomes are there? No, man, they will have to hide something. There must be a reason why they ask you that. Yes, there must be a reason why they ask you that. Maybe there is a chromosome which they have they have done the hiding, and then when you have to identify where they have hide that. Guys, give it or don't panic. Take your time, read the question 3 times exactly. If you feel like you don't understand, skip it. Yes. Why? Right? What? Start. If you see that things are not working out, start with what you think is right. I'm telling you, don't panic. Don't panic because you have not seen the diagram. Yeah, the diagram is known. This time they have brought it in terms of what? So why are you skipping? Skipping what? I'm not skipping anything. Yeah, I'm going to come back. I'm trying to talk about the things. After that, I come back to those questions. Ne? Yes. So guys, make sure that you concentrate on those karyotype. Make sure that you know how to identify them. Give a reason. Give one observable reason why state the gender of P. P, where is P? This one. We say that this P is what? Is a what? Is a male. Why? Because the chromosome number, the pair number 23, it has two different chromosomes. One is X, one is Y, or one is big, one is small, meaning that one is X, one is Y. Yes, that's number 2. That's where people fail. Number 3, non-disjunction. Non-disjunction. You see this? This is a non-disjunction question. Yes. Whereby when they ask you to which phase is it, is anaphase. It could be anaphase 1 or anaphase 2. Yes, but looking at the diagram, you can identify that it's anaphase 1 or anaphase 2. Yes. Describe Down syndrome. How is it? How is non-disjunction lead to Down syndrome? Guys, you just talk about what is extra. That Down syndrome is as a result of non-disjunction, leading to one cell having an extra chromosome. Yes, on chromosome number 21. Yes, meaning that it's gonna have 24 chromosomes instead of 23. Yes. When this cell fertilizes a normal ovum, the resultant individual is gonna have 47 chromosomes instead of 46, leading to Down syndrome. Is it clear? Down syndrome is caused by non-disjunction. Is it clear? Either in anaphase. In anaphase, it could be anaphase 1 or anaphase 2, leading to a cell having an extra chromosome on chromosome number 21. In brackets, you can say 24 chromosomes instead of 23. Then, when this gamete, yes, fuses a normal gamete, meaning that it is having 23, it will result in 46 chromosome, a zygote with the 46 chromosome, 47 chromosome instead of 46, leading to Down syndrome. You see, it's simple. I don't know why people make it difficult and complex and what. No, man, it's simple. Is it clear? Yes. So that is another thing where you lose marks. Number 3, give two. Let me check here. I hope that question is here. Metaphase 1 and anaphase 1. Those ones you can do. Okay. Describe two ways in which this is Down syndrome. Yes, so we have answered that one. Yes. Name the organ in a male that where meiosis occur. Okay, meiosis occurs in the testes for the case of male, and then female, it occurs in the ovary to produce ovum. Yes, but there is something which I want where students fail. Give two causes of variation in meiosis. Guys, they are saying in meiosis. They're not talking about the causes of variation. No, they're saying about in meiosis. So what are you supposed to talk about here? You talk about the the random arrangement of chromosomes during metaphase. Yes, random arrangement of chromosomes during metaphase, and also talking about crossing over during prophase 1. Remember, when you talk about prophase, crossing over, you are specific to prophase 1. Prophase 1, not anything else, is prophase 1. You don't talk about something else. It's supposed to be prophase 1. That is crossing over during prophase 1, which leads to exchange of the genetic material. So please don't go to the paper without knowing the process of crossing over. Don't go to the paper without knowing anaphase. Don't go to the paper without knowing metaphase. Yes, metaphase 1, metaphase 2, anaphase 1, anaphase 2. Those we might likely to bring it. Division of the cytoplasm, what does it mean? Is called cytokinesis. This division of a cytoplasm is called cytokinesis. Lastly, on meiosis, what you can't go without knowing is know how to label. What is this? In most cases, people, they call this nuclear membrane. No, nuclear membrane cannot be seen. You see, the nuclear membrane can only be seen in telophase. Yes, in telophase. And in prophase, early prophase. So this is cell membrane. This is chromatid. This is centriole. The dot is centromere. This is spindle fiber. Ne? This is, what is this? This is a cytoplasm. So now I think you know. The last question whereby students get lost is draw the diagram of a cell at the end of meiosis, or draw the diagram of a cell after crossing over. Let me just show you using this. I think I have the diagram here. Give the collective name, give the gender, name of observable reason, the main result of segregation, describe the character type. Down syndrome will result differ. Okay, sharp. That's not what I want. Yes, here. Look at that diagram. Look at that diagram, guys. If they ask you to draw the cell or this after this process, guys, look at where crossing over took place. In this case, the crossing over took place up, while here is took place here and here. So if they ask you to draw this cell, maybe they are saying chromosome X and chromosome Y, whereby you say structure of chromosome Y after maybe crossing over, then you have to draw it nicely. Yes, you have to draw it nicely. But where did the crossing over take place? Crossing over took place on this chromatid, but halfway up. So it means that when I'm shading all this, remember this is the black thing, so I have to shade all this. I have to shade all this. Yes, I have to shade all this to be black. And then the crossing over did not start from here, it started from here. So this variation of shading will give you a mark. Writing a caption will give you a mark. And then don't forget to label. Even if they have not asked you to label. Even if you don't ask you to label, at least have 2. Because in most cases when they ask you to label, at least 2 points. Yes, they say at least 2 points. You have labeled it at least 2 points. At least then you get those marks. Yeah, please don't forget to label. If they say draw a label diagram, or they'll say draw and then just leave it like that, guys, make sure that you draw and label, because it's not losing you, it's not taking, they won't cross it because it's labeled, but you get a mark if you labeled, just in case, because you are not a memorandum to know which one is not going to be credited or is going to be credited. So you just write. Lastly, before I forget, if they ask you to describe, write everything you know in the head. At least for us, we look for what is correct for you. If they say name, that one you only name. You don't give alternatives. But if they say describe, just write everything you know. Write everything you know, or sometimes I call it write all the rubbish. I'm not saying that write the rubbish, but write everything, whatever you have in the head. Don't leave it, because when you leave it, you come out of the paper and say, I thought about it. No, don't do the regrets, because this is your last time to change this subject, the mark. So just write everything. If the correct answer is there, we look for it and we'll give you a tick. Yes. Yeah. Do you have any question, guys? Comparative anatomy. Someone saying comparative anatomy. Okay. Thank you. Guys, if you have any question, you let me know, because I'm here to make sure that you get a distinction. You get a what? A distinction. Yeah. Make sure that the distinction clicks, because we want it and we will get it. Yes. God willing, me, when in my region we say, Inshallah, we will get a distinction. God willing, we will get a distinction. That's what it means. All right. Check this. It's simple. You guys, I'm winding up now. Ne? Make sure that you know how to label those diagrams. Make sure that you go when you know the process of crossing over. Make sure that you know the karyotype. Make sure that you know the prophase, anaphase, telophase. Make sure that you know when you go there, when they give you a diagram, it must be east to west. Those spindle fibers, they must be east to west, meaning that you have to turn the paper. Turn the paper. No one is going to ask you, why are you turning the paper? It's your paper. No one is going to ask you whether you turn it, whether you write upside down. Doesn't matter. Turn it so that you identify the things correctly. You understand? Yeah, these phases, they are confusing people to identify. Please don't make a mistake. It's the last time for you to identify what is it. Let's try to label this and then we see. How do you call C? Is the centromere. How do you call this? The point where crossing over takes place is chiasma. How do you call this? We call it chromatid. How do you call this? Because they are chromosomes and crossing over, we call it bivalent. Yes, we call it a bivalent or bivalent. Yes. Or you can call them, sometimes you can call them, they can correct you when you say homologous chromosomes. Yeah, but scientifically we call it a bivalent. Or bivalent. Someone might ask me the spelling. Yes. So identify. Okay, we have done that. Name two ways in which structure A and B is similar. Structure A and B. How are they similar? How are these two structures similar? They have the same size. Same size and shape. Cool. Number two, they have the same location of the chromosomes. Not same chromosome. They are not the same chromosome. If they're same chromosome, you'd have looked exactly the same as your father. Yes. So they have the same position, locus, same position of the genes. They have the same position of the genes. Describe the process at D. So they are looking at describe the process of crossing over 5 marks. The homologous chromosome come together. They cross over at a point called chiasma. They exchange the genetic material between two homologous chromatids, and then they separate, and then they form genetically different chromosomes. So basically, that's what you need to explain when it comes to that. I'm not going to use too much of the distinction material, but I know that you guys, you will make it. Lastly, before I go, where does meiosis occur in male? It occurs in the ovary to form ovum, testis to form spermatozoa, then in plants, ovary to form ovules, anthers to form what? Pollen grains. That's it. Hey guys, I feel good when you guys are feeling good. Don't panic, because when you panic, you make me panic. Yes, so make sure that you don't panic. Yes, make sure, I repeat, don't panic, you're gonna make it. Let's go to genetics. What are you supposed to talk about? Genetics. So we have used 1 full hour. Remember, we said that we're gonna discuss this paper for 2 hours. Yes, so we have used 1 full hour. So let's go to the second hour, that is genetics, and 30 minutes, and then 30 minutes for evolution, and then we know that we have smashed the paper. Guys, make sure that you get all the marks in DNA, all the marks in meiosis. Yes, guys, in genetics, when you talk about genetics, terminologies, I'm not going to talk about the terminologies, but go when you know that a circle means a female, and then a box means a male. Is it clear? That's number 1. Number 2, when they give you a question, identify whether it's a sex linked or a non-sex linked. I repeat, identify whether it's a sex linked or a non-sex linked. How do I differentiate between the two? Sex linked, they will mention it that is sex linked disorder. Always read what is on top here. Is a sex linked disorder. Is it clear? Make sure that you read it. If they say sex linked disorder, definitely you have to use X and Y. Male, there will be X and Y. Female, there will be X and X. If it is not a sex-linked disorder, please don't use X and Y. However much they have talked about same and carabo, and you know that this is a male, this is a female. Please don't use X and Y if they have not mentioned that this is a sex-linked disorder. I think I'm clear there. Number 2. Let's do one example on sex-linked recessive allele. One example, sex-linked dominant allele. One example, non-sex-linked. The first one, identify whether it's a sex-linked or non-sex-linked. Okay, now we have identified here that is sex-linked. The second thing you need to know, guys, I repeat, and I'm putting down the pen and the mouse. I wish you could see me the way I'm begging you. Please, let me come closer to the mic. Please identify whether is caused by a dominant allele or a recessive allele. I repeat, identify whether it's caused by a dominant allele or recessive allele. Because if it is caused by a recessive allele, the one who is homozygous recessive is the one who is going to be affected. If it is caused by a dominant allele, the one who is homozygous recessive is going to be normal. Let me show you using here. This diagram is saying that. So it means that it's caused by a recessive allele. Ne? They said it here. Ne? So it means that the one who is shaded, they're saying that is affected with the disease. Therefore, this person is a male because the sex linked is going to be that way. While this is a female, is going to be small d, small d. Is it clear? This one is a male, is going to be capital D, Y. Okay. Question. What about if it's caused by a dominant allele? Let me check here. Perfect. This is caused by a dominant allele. Yes. Okay, let me first finish here so that I don't confuse you. Now, let me go to the questions and see how many generations are shown here. It's 1, it's 2, it's 3. So there are 3 generations. How many kids listen carefully, how many kids are sons of 1 and 2? How many kids are sons of 1 and 2? 0. How many kids are for 1 and 2? 2. Remember that this is not a kid of this. This is just came and married this daughter. And this one came and married that daughter. Therefore, there are how many? There are 2 kids which are coming from these 2. Is it clear? You see that this thing is not linking down. Is it clear? Yes. Sorry. I'm done with that. Let's go to the third question. How many family members are in this phylogenetic tree? Family members. It's 1, it's 2, 3, 4, 5, 6, 7, 8, 9, 10. They are saying family members. Is it clear? They're saying family members. Don't say that this is not a family member unless you're telling me that your father is not among the family. No. He's a family member. But if they say how many kids for this, then I think now it's clear you know what you're supposed to do. Yes. That's the third thing. If they say that this cross with this, produce these kids, do a genetic cross, because we are in the second generation, so it's gonna be P2F2. Is it clear? If they say this, it's gonna be P1F1. I think I'm clear there. Yes. So I will talk about when I go to the crosses. Then we go to the next what you're supposed to know. Yes. Can you please do a live later? Today I join. Can you do the live again later? Today I join. No, what you can do if the live is start from where you have started from, and then after that join, you will play the video, ne? Yes, before. Lindo Kutle, why are you removing? I didn't see what you are saying, sir. The sound at the back. Eh. Someone saying there is a sound at the back. Ah, serious, there's a sound at the back. Okay, let's go to the next question. Okay, how do I find the unknown one? I'm gonna talk about it, my daughter. Uh, yeah, guys, I hope the sound at the back is removed. Ne? Let me know. Let me know. Let me know if the echo sound is removed. I don't want those echo sounds. Ne? Yeah, it must be nice. All right, let's go to the next question. How do I identify? Okay, now someone is asking me. I'm gonna go there so that we finish this. Ne? How do I identify one? To identify one, remember you have to first identify that this is caused by a recessive allele. Yes, if it's caused by a recessive allele, so what you need to do is okay, what you need to do is, okay, this one is XX, but this person is normal, therefore it's gonna be capital D. I don't know whether it's gonna be a capital D, small d, or capital D, capital D. But they produced a kid who is sick. Therefore, this kid is sick with a, don't just say sick, sick with what? With the disease, ne? So automatically, one came from the father, one came from the mother. Therefore, this mother cannot be like this because it gave rise to this. You see? Whenever they ask you to identify, make sure that you look at the kids, ne? Yes, which kind of kids they have produced. Therefore, we have identified that because this kid is small d, therefore one came from the mother, so therefore the mother is like this. Ne? Now what about the father? Remember, this is sex-linked disorder. Yes, if one came from the mother, then definitely another one came from the father. Therefore the second x, the first x is like this, or the x is like that. Therefore the father is affected with that disorder. Don't just say affected. Affected with the what? HIV, sickle cell, flu, corona. Is affected with what? With the condition they have talked about. Don't leave the answer hanging. Ne? Please, 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 please. Don't leave the answer hanging. Then the second one. Okay, someone saying, ish. Hey guys, just give me 2 minutes, ne? Let me check why is it like. Ahem. I hope the sound is gone. I don't know whether. Okay. I hope the sound is gone. Yeah, you let me know. Please slow down. I hope. I hope it's okay now. All right, let's continue, guys. Let me know if the sound is fine. I don't know because I've tried to see that everything is right. I've tried to check. Let me check. Let me check here. I've tried to see that. Yes. Let's continue and see. Hey guys, I think that sound, I don't know because we changed the system and the system I'm using is a different one from the main one. Yes, so I had to change the stations. All right, let me just continue. I hope at least you get something. Yes, okay. I think now you have got it correctly. How do we identify this? Yes. Now let's go to the question, the next question. And then we are finding out. Identify the phenotype of individual 2. These ones I'm going to do the questions and then we see. The phenotype of individual 2. So we have said that this person is normal. Yes, is normal. A normal male or female is a normal female. Or female without C. Yeah, that disorder. Then the phenotype of 1, we have identified that this person, because they have produced a kid who is sick and the sick is like this, therefore one recessive allele came from here, another recessive allele came from here, and remember this is a male. This is a male, therefore this individual is affected with seed. And then they're saying that identify the genotype of number 3. Yes, remember this person is sick, therefore is gonna give remember is a female, is gonna give 1 chromosome from there, but because it's normal, therefore the mother gave her this, therefore is gonna be that way because they say the genotype. They say explain how individual 5 inherited disorder. Individual 5 is small d, therefore we're gonna say that individual 5 got 1 recessive allele from the mother and then 1 recessive allele from the father. No. Individual 5 is a male, is a male. Can't be from the father. Father is normal. The individual is male. So you're gonna say that the individual 5 is a male who is sick and then therefore obtained a recessive allele from the mother. Where is these 2 marks? You have to tell us that individual 5 is affected with this disorder and is received 1 recessive allele from the mother who is number 4. Therefore, I think that's how you can try to explain. But now let's go to, hey guys, the problem comes in when it is caused by a dominant allele. Okay, let's look at it. When it's caused by a dominant allele, whatever is syndrome sex linked. You see, they have talked about it, but it's caused by a dominant allele, isn't it? The diagram below shows this syndrome in the family. Okay, now we are stating how many generations are there? There are 3. 1, 2, 3. Sharp. How many females are in this phylogenetic tree or in this family? Females. 1, 2, 3, 4, 5. So 1, 2, 3, 4, 5. Is it clear? How many females were produced by Patty and Gabby? It's gonna be females 1, 2. This one is not family member, not a kid of this. It's a family member, but it's not a kid of this. So it's only these ones who are the kids. I think I'm clear there. Then now we go to the next question, whereby they're asking you. They're asking you. They're saying that. They're saying that. They're saying that. Okay. Name the. All right. Let's find out. Remember now they're saying that this is affected, né? And then this is also affected. This is unaffected. But don't forget that this syndrome is caused by a dominant allele. Therefore, these people who are affected, they are going to be dominant. Capital D, capital D. Can it be capital D, or capital D? Sorry, X, capital D, X. Is it capital D? No. They used G. Sorry, guys. They used G. Please use the letter they have given you. Don't just use what you think is correct. Therefore, it's gonna be X, capital G, X, capital G, X, small g, no, capital G, and X, small g. Yes, but now I need to find out, do we have, what about this one? This one is normal. So if it's normal, therefore it's going to be small g. It's going to be small g. Therefore, this person can't be this, because this person, the kid, has a small g. Therefore, came from here. Therefore, I have to say that this individual is like this. Capital, small g. No. Capital G, small g. So, that one is done. What about the female? The male. Remember, this is normal. If it's normal, therefore automatically is having small g. That's why this and this, they combine to produce a kid who is a female with the two recessive alleles. We are done with this. What about this? Is a female who is sick. Who is sick. Therefore, this female can't be homozygous dominant. It's going to be capital G and then small g. Why? Because one X came from here, then another G came from there. Do you understand? Yes. Now, how many, name the type of diagram is phylogenetic tree. They're saying how many female in this family? We talked about them. 1, 2, 3, 4, 5, 6. Male in this generation have, how many male in this generation? Generation 1 have what is syndrome? Generation 1. Where is the generation? The first generation. The first generation. Remember, this is a generation 3. 1, 2, 3. Because they are looking for male in generation 1. So this 1, 2, 3. They're talking about generation 1. They're saying with the disease. 0. The answer is 0. Because it's caused by a dominant allele. And then they're saying that give the male in generation 1 have what is disease. Give the genotype of Gabi. Genotype of Gabi. We talked about it because they produce a kid who is not sick. Guys, please take note which one is dominant, caused by a dominant allele, which one is caused by a recessive allele. They will have to specify that is caused by a dominant allele. Is it clear? Males in okay, we're done with that. Aju and Pirusa have another 4th child. Give a phenotype of their sons. And Pirusa. Yes, so of their sons. Remember this is caused by a dominant allele. The moment it's caused by a dominant allele is easier to find. For the case of what? A son. And then, remember this one, son. Ne? It's a Y, it's a Y. Therefore, because this one is normal, remember is recessive, is normal, therefore this one is going to be, both of them are going to be normal. Don't forget it's caused by a dominant allele. If it was a recessive allele, then they would have been affected. But the one who has the recessive allele is the one who is normal. And the one who has the dominant allele is the one who is affected with that disease. Is it clear? Yes, that is the only trick you need to know about that. The one is normal. Yes, the one who is normal is having a recessive allele for the dominant allele if it is caused by a dominant allele. The one who is sick is the one who is having a dominant allele. You understand? Yes. Explain your answer above. Why? Because they are normal. Because Anju, yeah, who is a female, is normal with the two recessive alleles. Therefore, it gives one recessive allele to the males. You understand? Yes. Therefore, they are normal. So basically, that's what you need to talk about. When you go to a cross which involves an autosome, a non-sex linked, ne? Yes, please, even if they talk about same and carao, please don't use x and y, ne? Yeah, if they say that this also is caused by allele r, so you just say r, ne? It is homozygous. If it's heterozygous, is r, small r. You don't put them on x and y. That is the major difference between the two. Yes. Okay, lastly, about blood group. And then after that, we talk about the cloning. How many blood groups, how many genes control blood group? How many genes control blood group? How many genes control blood group? Someone is asking me, sir, it started confusing you. Daily interesting videos. How am I confusing you? Let me know. Let me know. How am I confusing you? Oh, where are you confused? Yeah, in this video description. You're saying I'm confusing you. Let me know where am I confusing you so that I can sort that out. Ne? Yeah, just tell me that maybe this question or this question. Yeah, so that I can sort it out. How many genes? Yeah, correct, guys. Correct. Thank you very much. Yes. How many genes control blood group? It's one. It means that you guys watch my videos. Thank you very much. And I'm seeing live as Mbali as a member. Yeah. Thank you very much, Mbali. Yes, for being a member. And I saw another member here coming. Oh, Lindy Wend Love. Thank you very much also for joining us. So you are on the team, ne? Yes. All right. Never be saying that X and X are. Oh, okay. Okay, let me explain to you what I meant there. Someone is saying that X and XR. I'm saying don't use X and okay. Let me just look for a question here. Yes, here is a question. Ne? Mbali. I think it's Mbali, ne? Yeah. Here they're saying that this syndrome in dog is caused by a dominant allele, capital H. You see? The pedigree diagram below shows the offspring produced by two dogs. You understand? They're not saying that this is sex linked. You understand? Yes. How many offsprings are affected by this disease? How many offsprings? You see now? Is caused by now this one is also caused by a dominant allele. Is caused, meaning that the one who is sick, ne? Check here. Even here they are saying it that dogs with the disease and dogs without the disease. So these ones which have the disease, it means that they are going to be capital H, or can be capital H, small h. Even this one is going to be capital H. You see, is a male, but I'm not using X and Y. Capital H, or capital H, small h. Whenever there is a capital H, because it's caused by a dominant allele, this individual is going to be sick. You understand? Yes. So, however much is a male, I'm not using X and Y. You understand? I don't use that. Why? Because they didn't say it is caused by a sex linked. Is it clear? Yes. So in this case, but I can identify because they say that how many individuals are affected. Okay. That one is simple, but now this one is what is not affected, meaning that this individual is gonna be small h, because it's caused by a dominant allele. Whenever you see a capital H, it means that that individual is what? Is sick. If they produce a kid here who is having these two, then automatically one came from the father and one came from the mother. Therefore, they can't be this, they can't be this. So this is like this, so that one H comes here, and then this one is like this, so that another H comes there. You understand? So when they give you a question and they don't talk about sex linked, please do not use, I repeat, do not use X and Y. Use the letters the way they have given them to you. Yes, you only use X and Y if the statement says that is caused by a recessive allele is sex linked. You see, is sex linked. Even here they said it here is sex linked. Even they gave you what to use, that is carried on X chromosome. Is it clear? Yes. So this situation is caused by a dominant allele, but it can be caused by a recessive allele. So I think now I'm clear when it comes to that difference between phylogenetic tree and pedigree. Phylogenetic tree, guys, phylogenetic tree is explaining the evolutionary relationship among the members. You understand? Evolutionary relationship, like how did you evolve? But a pedigree is describing a character in a family. Is it clear? Is describing a character in a family. Yes. Okay, guys, let's go back to blood group. I think I've got it. You have got it. I'm no longer confusing you. How many genes control blood group, guys? Is one gene. Is one gene. How many genes control blood group? Is one gene. How many alleles control blood group? One gene, and that gene is blood group. Do you have any other gene? Tell me if there's any other gene. This is one gene, blood group. Yes. But how many alleles? There are 3 alleles. Yes, that is allele for A, allele for B, and allele for O. Guys, please don't make a mistake and you say allele for A like this. This is not A. This is a Roman numeral, which means 1. Allele for B is supposed to be like this. Allele for A. Don't put at here. Don't put them, make italics. Don't say allele for O like that. What is this? We will cross it. I is supposed to be like that. Is it clear? Yes. So allele for A, allele for B, and allele for O. So there are 3 alleles. Yes. I don't want to go to the distinction material to show everything. No. We go to the next part, of which the next part they're asking you how many genotypes. Genotypes. Oh, before I go, guys, back, there's something I forgot. Before I go back, I don't know what took me off. Why are males more affected by sex-linked disorders than females? Why? Or they can say, why are males more affected by hemophilia than females? Or they can ask you, why are males affected by color blindness than females? So the answer you give here, if they say 4 marks, you say that males have 1 X. Yes. If a recessive allele, for example, is capital small h, appears on X, yes, the individual is affected with the disease, whatever they have talked about. While females, they have 2X. Yes. If a recessive allele appears, for example, on 1X, it can be masked by a dominant allele on another X. Therefore, the individual is normal. Males have 1X. Yes. If a recessive allele appears on the X, the individual is affected with that disorder they are talking about. While females they have 2X. If a recessive allele appears on 1X, it can be masked by a dominant allele on another X, and the individual is normal. Sometimes they can ask you a question that why males have higher chances of of becoming hemophilic than females. So guys, it's the same thing. Please, you need to know about that. Ne? Yes, you need to know about that. We go to the next question. Because they have 1x. Yes, because they have 1x. This question they can come for 3 marks. Ne? Yes. Is it possible for a person of blood group A marrying a person of blood group B and they produce a child of blood group O? Is it possible? Yes, it is possible. How do I know it's possible? You look at the genetics. Ne? Okay, here the questions A. Before even I go there, let me just answer this question, at least one question. And so that, yeah. A man who is blood group AB and then who is heterozygous married blood group B and then have a children. How many alleles control blood group? They are now talking about the alleles. Yes. Gene 1 is 1. Allele, there are 3. Genotype, there are 6. Phenotype, there are 4. Why am I using 4? You have A, B, O, and AB. A, B, A, B. Do you understand what I'm saying? A, comma, B, comma, A, B, comma, and O. Those are the 4 phenotypes. But genotypes, there are 6 because A is homozygous or heterozygous. Let me show you. The distinction material, because some people, just talking, they might not take it, and they must take it long to understand my ideas, what I'm talking about. Yeah, you see here is what I'm saying from the distinction material. Blood group are as a result of multiple alleles. It's a multiple allele, yes, meaning that it has more alleles, more than 2. There are 3 or more alternative form of yes, for this there are 3. All right, so phenotypes, there are 4. 1, 2, 3, 4. You understand? And then the genotypes, there are 1, 2, 3, 4, 5, 6. That's what I say. Because this is homozygous and heterozygous, codominant, and then homozygous recessive. Yes. Then they are saying that 3 different alleles, which are these, and then 4 blood groups or types, which you call the phenotypes. No, the genotype, the phenotypes. Yes. So it's A, B, O, A, B, and then O. That's what I'm saying. A, comma, B, comma, A, B, comma, and O. So that you understand. Yes. So now we go back to our question. Describe the type of dominance that occur in the inheritance of blood group of woman B. Describe the type of dominance that occurs in the inheritance of blood group B. Now they are saying inheritance of blood group B. Check. They're saying that a man with blood group AB and a woman who is heterozygous for blood group, yes, heterozygous for blood group B, meaning that this lady it looks like this. See? Yeah, meaning that obtained one dominant allele from the father or mother and another recessive allele from the father or the mother. So it can come either from the father or mother for A, and this from either father or mother, because they are not specific in that regard. Yes. Use the genetic cross to show the possible genotypes of the phenotype of their children. So you guys here, this principle works to all of them. Start with P1, F1. Please, F1. Don't just say P1 and then you leave, you finish after you say F1. You will forget this F1 and then you lose a mark. So you say P1, F1, at least we shall come here and we give you a tick. Is it clear? Then you say P1 phenotype, because P means parent phenotype. Ne? Yes, you tell us. The phenotype is always obtained from the question. Check here. You use because these 3 marks, if it is 6 marks, 3 marks are mahala. You understand? Use the genetic cross to show the possible genotype of phenotype of what? But what did they say? They said a man who is blood group. So you say a man of blood group AB times a woman of blood group B. You don't need to tell us heterozygous, because when you look at someone, you can't say that he's heterozygous. We just say heterozygous, homozygous, so that you know what you're gonna cross. So you saying P1, F1, we give you a tick. Now you tell us phenotype. You tell us a man of blood group AB times a lady of blood group B. That's all. Then we give you another tick. Why? Because you have identified it, but you always get it from the question. So that's the second mark for Mahala. Then you talk about genotype. If you don't know the genotype, leave the space. Talk about meiosis. If you don't know the meiosis, leave the space. Talk about gametes. If you don't know the gametes, leave the space. Then you talk about fertilization. If you don't know this fertilization, leave the space. But you shall come here and give you a mark there. So you have got 1 mark, 2 marks, 3 marks. Meiosis and fertilization, they must go together. If you know now, that's where you're going to obtain the other 3 marks. To know the genotype, because most of the cases, you people, you get lost on the genotype. When you get the genotype wrong, the remaining thing here becomes what? Becomes wrong. Someone is asking me, please, I answered that question. You'll have to rewind the video. Yes, I answered that question. Yes, for case of the DNA changing, the triplet. Yes, I answered that question very well. Please, you have to watch it maybe when I'm done with this session. All right, guys, let's go to the next question. That is dihybrid cross. And then I show you what you do or what you focus on dihybrid cross. Dihybrid cross, we are not going to ask you to cross. No. What we might ask you about is identify the genotypes of the parent, the phenotypes, and the gametes. Yes. So guys, you need to know first identify which one is which. But now if they ask you what is the genotype of the parent here, for example, don't repeat the letters before you finish. For example, the genotype of this can be capital P, even capital L. Then another one is capital small p, small L. So the one which are the same, you combine them. Capital P, capital L, small l. So this becomes the genotype of the parent. I'm just trying to show you how do you identify. And if they ask you to identify the gametes from here, then it still is fine. You will say that P goes with L. P, capital L. So this one and this one is done. Now P again with the small l, capital P, small l. So it's done. This with this now, small p, capital L. Then this with this, small p, small l. Sometimes people, what they do is not a cross. It's not a cross, but what they do this. They do this, capital P, small p, capital P, small p, capital L, small l. So they just cross with this, capital P, capital L. They cross this now again with this, capital P, small l. Then now they cross this with this, small p, capital L. And then now this with that, it becomes small p, small l. See, they form the same gametes. You understand? Yes. So at least you have done. Now they can ask you, they have given you the gametes and they want you to write the genotype of the parent. Yeah, that's where the story comes from. So you start with you start with one letter, capital P. Do I have any other capital letter? Yes. So you first leave it. Do I have a small letter of the same type? Small p? Yes. If I have a small p, I write the small p there. So they are supposed to be 2 letters. But if I don't have a small p, then definitely I will have to write a capital letter. Is it clear? I will have to write a capital letter here. But because I have a small p, therefore I will have to write the small p, Then I go to L, capital L. Do I have another capital L? Yes. Do I have a small l? Yes, I have a small l. Therefore, it's going to go with a small l. Therefore, this becomes the genotype of the parent. Check. It's the same thing. Capital P, small p, capital L, small l. And then when you come here, it's capital P, small p, capital large L or capital L, small l. So that's how you can identify that. Difference between transcription and DNA replication, I talked about it, guys. I talked about it. Just wind up. I know that you will find everything you have asked me. You are trying to ask me. Yes. Lindo, yes, I talked about it. Difference between transcription and DNA replication. Yes. And even the similarities also talked about them, that both the DNA, the double helix, unwind, you talk about both there is unzipping. Yes, when you talk about differences, you can talk about DNA is formed, RNA is formed, two templates are formed, one template is formed. So you guys, we talked about it, yes. All right, let's continue to the next one. How many genes control? Once you see the dihybrid, then there are two what? How many genes? There are two genes. Dihybrid is controlled by two genes. Yes. How many alleles? Yeah, in most cases there are 4 alleles. Yes, you'll find out the capital letter and the small letter. So depending on whether you see they are here. It's 1, 2, 3, 4. But there are two genes. Genes for the number of seeds per pod and then the shape of the what? Of the leaf. Yes. So guys, the hybrid is gonna be is simple, It's simple. Just make sure that you do it, Yes. So it's the same thing here. No one is gonna ask you to cross. Before I leave this point, I wanna ask you a question. Is it conclusive to use blood group to identify the paternity, the paternity of the individual? Is it conclusive? I want yes or no. Is it conclusive? Then once I see the answer, then I will give the reason. If the answer is correct, I will give a reason why. Three factors of, okay, the experiment, that's one you must know, guys. Lastly, let's go to, before I go to, please read about the stem cells, read about the cloning. Yes, the cloning, the process of cloning. I designated a full video on cloning. Stem cells, what are stem cells? The cells which have ability to modify and form other body structures. Yeah, basically you just need to read this and the examples of these stem cells. Yes, let me just show you here from Excel in life science distinction material. Yeah, you see sources of bone marrow, umbilical cord, adipose tissues, amniotic fluid. Yeah, uses to generate new or replace the worn out cell or damaged cells, or to generate new cells. Can be used for research purposes. Yeah, so cloning, please know how to use this cloning, but most especially, let me just talk about, it can come like this or it can come as a doll. What? Do you know that this is the name of a person? Is a singer. Dolly is a singer from, I think it's from America. Do you know that? That is a singer from America. You just listen to people's songs, but you don't know that they're asking you questions and you don't know. I hope he's a singer, because I'm not a fan of that much music. All right. Don't ask me what are you fan of. Okay. Is it conclusive? Okay, thank you very much, guys. Everyone is saying, sir, can you please touch on stem cells? Okay, sharp. Someone is yes. I'm saying that. No. Yes, it's no. It's not conclusive because many people share the same type of blood group. Therefore, it's not conclusive. It's not conclusive. Yes, it's not conclusive. Someone say, yes, I'm going to talk about them. Let me just talk about stem cells. Then we shall go to, not stem cells. I'm done with stem cells. Let me talk about the cloning. Guys, this is what I want to clone is this. I want to clone. Let me just do like this so that it gives me the full version. I want to clone this. Therefore, I have these two. I get ship A. If I want to clone this, I have to keep its nucleus. Is it clear? Why? Because the nucleus contains the genetic material. So I keep the nucleus, I throw away the shell. Then this one, I remove the nucleus because I don't want its genetic material, but I use the gamete, the ovum. But here I just get any cell from anywhere else apart from a gamete. So I throw away the nucleus because I don't want it, but now I keep this. But why do I need to have, why do I have to be with a casing of the cell? Why should I be with the casing? Yes, it's like you drinking water in a bottle of alcohol. What are we going to say? We shall say that you're drinking alcohol, Baba. Yes, that's what you're gonna say, that you are drinking alcohol. Yes. Why? Because you are putting water in a bottle of alcohol. So when I put this nucleus into a cell, yes, this cell, it will look like a gamete. Remember, this is a diploid cell from anything else. So if I put this nucleus to this, then this one is going to look like a zygote, because this is a casing of an ovum, and I've put a nucleus which is 2n. You understand? Therefore, it's going to look like 2n. So if they ask you how many chromosomes are here, these are, if they say the sheep has 24, therefore it's going to be 24, or it's 2n. Yes, you understand? Yes. So now, we are saying that which method do we use? We also ask you about the method. Is mild electric shock. Then you have here the foster mother here, and then we bring this, we put it in here, and then after putting it here, then this one starts to develop, to develop that embryo, and then it forms the baby. This baby is exactly the same as this. They can ask you a question: why do we remove the nucleus? Why do we keep the nucleus? Because we wanted the genetic information from sheep 1. Why did we throw the nucleus away? Because we wanted the casing of the ovum. We are not interested in the genetics here. Yes. So which method we use? Mild electric shock. And then which sheep will this, which of these two sheep will this one resemble? It's sheep 1. Give a reason for your answer, because we used its the nucleus. We used its nucleus or the genetic material to produce the sheep. Yes. So when they ask you such questions, they're not that difficult. You'll be able to answer and get it right. Paternity test, blood group. So it's the same thing we are saying. This is the question I was asking. Is it conclusive? It's not conclusive, because many men have the same type of blood group. So it's not conclusive to confirm or to determine that this is, yes, the parent of this child. Guys, now we are going to evolution. Yes. Guys, before I start to talk about evolution, the way now we are setting is a little bit different. We used to have a lot of marks in human evolution, and we are specific that human evolution takes this amount of marks. But now it's not the case. Why? We can have more marks in evolution by natural selection, and that's what they usually do now. I don't know why. And the more they bring more questions in evolution by natural selection, the paper becomes harder. Because they talk about, we only have few sections to set in evolution by natural selection, but they try to tilt the thing. So today I want to tell you that, listen to what I'm going to say. Be ready. Show them that you are not here to joke. You're gonna get a distinction. You're gonna get what? A distinction, and you're gonna be set free to this world. All right, let's start. Why don't we use the whole genetic material? Why? So why don't we use the whole genetic material of sheep 1? So why don't we use the whole genetic material of ship 1? Oh, why don't we, maybe you are asking me, why don't we use the whole cell of ship 1? Ne? Yeah, it's because this casing is not for ovum. This casing, the casing is not for ovum. You understand? And so therefore it cannot undergo meiosis. It cannot not undergo mitosis too. It won't look like, ne? It won't look like a zygote. Yes, yeah, because it is coming from this other body cell. It's not coming from the gamete. That's why we have to bring a nucleus, its genetic material, and then we put it in the casing of the ovum so that this thing looks like a zygote. You understand? It must look like a zygote so that when we put it in the womb doesn't know what is inside the what? This zygote-like structure. Yes, what it knows is this, né? So that now it can start undergoing mitosis and then forms those finger-like projection, and then done, implantation takes place. So that part of implantation is paper 1, eh? So we are done with that. So that's why we don't use the whole thing. If you bring this and then you put it in, it won't be recognized as a zygote. It won't be recognized. It will be recognized as any body cell. Eh? Yes. Scientist. Let's just do 2, 3, 4 questions here, and then we go to human evolution, and then I wind up. Scientist, let me see how many minutes do we have. We have 30 minutes. We have 30 minutes. We are saying that scientists hypothesize that these species of flighted birds arose from a common ancestor. Guys, when they bring a question of evolution, make sure that you relate to the question they have asked you. Don't just write. Don't use, now you'll find out that someone is using giraffes here. Why should you use giraffes when they said flighted birds? Don't use giraffes. Use what is. Ne? Make sure that you use what is given in the question. Ne? Yes. All right. Let's start. The same flighted birds are currently distributed across different continents, and then okay, the picture. So you see it. Now, if they ask you to use Lamarck's theory, whenever they ask you to use Lamarck's theory, you talk about 2 laws: the law of use and disuse. Then number 2, the law of inheritance of acquired characteristic. Yes. So these 2 cannot be used at the same time concurrently. Never. We will either do the use or we will do the disuse. So you have to pick one depending on what they have asked you. So let's look at this. So I hypothesize that species of flighted birds arose from single common ancestor, which was able to fly. It means that he was able to fly, but now they are flightless. It means that they lost that ability of flying. Therefore, was it use or did they want for disuse? Definitely it's gonna be disuse. Is it clear? So now I'm gonna say that maybe at the beginning, all birds were able to fly, because remember, when you are talking about Lamarck's theory, you talk about there is no variation. All of them are the same. You understand? All the birds were able to fly. So now you talk about the disuse. They reduced how frequently they were using these birds to fly. You understand? And then the wings maybe became smaller. Is it clear? They became smaller and weaker maybe. And then they couldn't fly. You see? They passed this allele. They passed this character, not allele, because this one doesn't talk about alleles. They passed this character. Which character? Of not able to fly to the next generation. They passed this character of not be able to fly to the next generation. Then leading to these birds which are not able to fly. So that's how you're supposed to explain this. In this case, they are bringing about disuse. What if they ask you a question that how did they, maybe they have, how did the elephant came up with the long trunks? At first, all elephants had short trunks. The elephants started to use the trunks to get water, yes, and defend themselves frequently. So you see now, in this case, they are using it. They are not misusing it. You see, frequently, the trunks became bigger. You understand? And then they passed this character of big trunks, big long trunks to the next generation. That's now the inheritance of the acquired character. Don't tell me that they passed the character to the next generation. Which character? They passed the character of big trunks. In this case, they passed the character of not able to fly to the next generation. You see now? The reason why people lose marks here is because they don't apply what is being asked. What they do, they just write. When they write, they think that they have killed the paper. Can't the paper has killed them? So tomorrow, you have to kill the paper. The paper can't kill you. So make sure that you apply. When you apply, you'll be able to smash the paper. Is it clear? Yes. So make sure that you guys, you apply. Why is Lamarck's theory rejected? Organisms don't evolve because they want to evolve. But now you have to tell us. In this case, you tell us that the flightless birds did not evolve because they want to evolve. You see, you are linking the question to what you know. There is not enough evidence to show that the flightless birds acquired this character, or they pass their genes, they pass this character to the next what generation, like from being flying birds to flightless birds. There is not enough evidence to support that. There is not enough evidence to support that these flying birds, they became flightless, by not using this character. You understand? By not using this character of flying, so they became flightless, just like that. There is not enough evidence to show that. Don't just say that there is not enough evidence to support the theory. How? Which evidence? It's too broad. Ne? Yes. So it's too broad, so we won't give you a tick. But when we are writing it in the book, we say that organisms don't evolve because they want to. Yes. But now which organisms? In this case, they are talking about the flightless birds. They are talking about maybe the elephants I talked about. Elephants did not evolve. There is not enough evidence to show that elephants acquired long trunks through using it frequently. You see? Yes. And then the elephants did not acquire the long trunks because they wanted to acquire it. You see? So you have to apply so that you are able to obtain a mark. Is it clear, guys? Distinction material, ne? Someone saying this. Sir, if use both of it, will it happen? I'm going to be wrong. Yeah, how are you going to use the two laws? There is no way. There is no way you're gonna say that. Sir, can you please give us a tip on how to remember the names of scientists? The scientists, you mean the scientists, the other ones, ne? Okay, I'll reach there, ne? I'll reach there, and then yeah. Now, what is expensive? I don't understand. I don't understand what you are saying. Ne? Leah, I don't understand, but it's expensive. What is expensive? I don't know. Hey, guys, let's continue, guys, so that we finish in our time. And someone is telling me there's something which I said. Sir, please, sir, describe how the high-flying similar ancestral birds evolved into different species of large flightless birds. Serious. Why, sir, why did they reject Lamaka's theory? That's what I'm talking about. Someone is saying distinction material is expensive. Is it 20 rand? Is it 10 rand? Oh my god. You're gonna get a salary of thousands and thousands. We're talking about 19 rand is too expensive. Yeah. Guys, that money is helping us to buy data, to do this, so that at least, the channel can continue. You have to support the channel. 20 rand. Yeah, to support the channel. How will it be supported? Data, systems and stuff. Yeah. Sometimes you just do like donation. Yeah, donation. 20 rand, guys. It's not that a big deal, ne? Just donate and then get the resource, ne? And actually, you don't only receive that, you receive a lot. You ask questions, we answer questions and stuff. Okay, guys, someone is asking me a question. Let me see if I have that question here. If I have that question, I'll try to answer it. If I don't have that question, unfortunately, but I had it. Let me see the one for flightlet birds. How did they become? I think. Do you mean this one? I think I don't have the question. I have to look at the question. Who is this asking me? Who is this asking me? Who is this asking me? Olwetu. Magula. Or Mahula, I don't know. Yes, I don't have the question here. I would have explained it, ne? But guys, make sure that you apply using that, ne? Make sure that you apply, ne? Yes. Number 2, let's go to natural selection. We go to punctuated equilibrium, ne? We go to natural selection. We go to punctuated equilibrium. We go to speciation. We go to present day evolution, just to show you how to master it. Yes. You don't go astray. Give differences between Lamarck's theory and punctuated equilibrium. Here you talk about punctuated. No. Lamarck's theory and the Darwin's theory. Lamarck's theory: organisms evolve because they want to evolve. If I want to be tall, guys, I will just stretch my head and then I become tall. Ne? Organisms evolve because they want to evolve. That's what Lamarck says. Don't say no. You're looking for marks. Then Darwinism say that no, organisms evolve because of natural selection. Is that clear? Then Darwin's theory, you can talk about the whole population change. Ne? While Lamarck's theory, individual in the population change. Ne? Just mention 2. Go when you know 2, not more than that. Describe how Lamarck could maybe explain how did the elephants came up with the long trunks. Let me just use this example this girl is asking me. I hope I can design a small question. Please describe how the high flying small ancestral bird evolved into different species of large ancestral flying birds evolved into large. Okay, it means that if they started with the large small flying birds into large flighted birds. Is it the same question? I think it's the question of, please describe. Okay, it's the same thing, ne? Let me use Darwin's theory to explain this, guys, ne? Yes, it could be, it's the same thing which we use, ne? But now let me just use Darwin's theory, ne? And I also link to what you are asking me. Describe how Lamarck, okay, now describe how will Darwin would have explained this. But remember, they say that flight birds currently distributed across this one. Eh? So now if they had an ancestor, a common ancestor, yes? Let me check the question you said. A common high-flying small ancestral bird. You tell us that at first there was one population of a common ancestor with the high-flying, yes, which are high-flying, small-flying birds. You understand? There was one population, but you describe as which population is that? A population of a common ancestor which are able to fly. Is it clear? There was one population of, yes, that's number one. Then now, but now that is, you can explain it using speciation. But now here, let's just using natural selection or Darwin's theory. How did they become like that? One population, okay. Now, that is the speciation. Okay, now let's start with this. Darwin's theory. There was variation among, when you talk about explain Darwin's theory, there was variation. The first point is variation. There was variation among these birds, those which are able to fly and those which are not able to fly. When you talk about the variation, you tell us the first statement must be there was variation among these flying or flightless birds. Flightless birds arose from single common ancestor of flying birds. There was one population, there is variation among these birds, those which are able to fly and those which are not able to fly. Is it clear? Those which are able to fly, yes, maybe they were not favored, maybe because they were being able to be detected by the predators up there. You understand? Therefore, they were being killed by the predators. While those which are not able to fly, they were able to survive. You understand? They were able to survive and they hide from predators. This is called natural selection. Those which are able to fly, they produced in large numbers. Is it clear? And then now you talk about they pass their genes to the next generation. After many generations, all birds were not able to fly, or they became flightless birds. Is it clear? That's how you can explain. But still, you can use this and ask you to describe how did they become new species. That's the challenge now. You know what makes the students to lose marks? When they bring this question, they don't know whether you're going to explain speciation or they are going to explain natural selection. That is the problem. Describe how Lamarck, describe how Darwin explained this. Guys, that is the natural selection. You talk about the first point, you talk about variation. There was variation among these organisms. Which one? These birds. Those which are able to fly and those which are not able to fly. Those which are able to fly, they were being detected maybe by the predators, and therefore they were killed by the predators. Then those which are not able to fly, they are able to hide from predators, and then they survived. They reproduced in large numbers. This is called natural selection. Those which are not able to fly, they pass their genes of not able to fly. You tell us which gene, which character of not able to fly to the next generation. After many generations, all the birds were able to fly. That's number one. Now, if they ask you the same question, they have brought the same question like this, and then now you don't know whether they're asking you whether you are going to explain speciation or what. Now, here they will ask you, how did these species form? Now you are talking about speciation. Is it clear? Now that's where you talk about there was one population of flying birds. There was one population of flying birds. Yes, with a common ancestor. Yes, population of common ancestor that were able to fly. There was one population of a common ancestor or a single ancestor that were able to fly. Now we are describing speciation. Yes, the common this population was split into different continents by continental drift or by the water. Yes, by the water. Yes, mass. These populations or subpopulation or individuals of birds, they experienced different environmental conditions. Natural selection occurred independently. You understand? There was no gene flow between or among these birds. Is it clear? Therefore, they became genotypically and phenotypically different. If you remove this barrier of aura and you make them mix, they are not able to interbreed to produce fertile offspring. Therefore, the ram, the ostrich, the im, and the quickie, they became new species. Do you see now how you explain? When they ask you to explain how did a species form, think about speciation. When they ask you describe using natural selection, you describe Darwinism, you explain what? You explain natural selection. Natural selection, you start with the variation. You tell us the variation between the two. While speciation, you start, there was one population. Yes, and then it split. You tell us what split them. Don't just say it split by geographical barrier. What is geographical barrier? What do you mean by geographical barrier? We need to know which geographical barrier split them. Is it clear? I think now I'm clear there, ne? Hey, someone is someone is asking me which predators can eat the flying birds. Hey, are you trying to mean that the eagle can't eat those small birds? Yes, it can eat the small birds, Baba. Yes, it can eat. There are so many. Are you trying to mean that if you are the birds which are flying up, they are not being eaten by predators? They also have predators, ne? Yes, they also have predators. They have, yes, which can, yes. Yes. Guys, let's go to the next question so that at least, I think, guys, I'm clear there. Natural selection is covered, and then speciation is also covered. Guys, don't make a mistake to describe speciation without telling us what made them separate. Don't go to the paper without knowing. Let me just show you here the different isolation mechanisms. Different where am I? Different isolation mechanism. Let me just talk about them here. This is the difference between Lamarck and the Darwinism. Yeah, the different isolation mechanism, artificial selection. Yes, these ones. Yeah, even artificial selection. No, that's not what I want. Yes, these ones. Yeah, breeding at different time of the year. Species courtship behavior. Adaptation to different pollinating agent. Prevention of fertilization leading to infertility of the offspring. Don't go there without knowing these different isolation mechanisms. So you guys, I know you're gonna make it. Don't stress. Read the question. Remember what I've said. Have enough time to sleep so that you can remember paper 2. I'm telling you, you come out of the paper when you are happy. When you come back tomorrow here to discuss what we have talked about, you know that yes, you will make it and you will pass it, and I'm sure you will be grateful for this lesson we have. Alright, guys, let's wind up with this so that we finish up with natural selection, and I show you how to remember the names of the scientists who discovered those. Yes. Guys, present evolution can be brought into threes. Use of DDT, that's the mosquito, use of the bacteria, or use of the antiretrovirus, ARVs. But all of them, they work together. They work the same way. It's the same way, natural selection. So when you talk about this, evolution in present time, that evolution is taking place, it's talking about the resistance. So it's about the resistance. So how did the mosquitoes become resistant, or any example they have brought, and then they are talking about resistance? You talk about there is there is variation among the mosquitoes, there is variation among the bacteria, there is variation among the virus. Those which are able to survive, those which are resistant and those which are not resistant. When the application of DDT is done, yes, those which are not resistant are killed in large numbers, while those which are resistant, they survive in large numbers. Yes, those which survive, yes, they reproduce in large numbers and they increase their population of resistance. The continued use of this application you have talked about, RVs, DDT, whatever the question is asking you about, yes, will have little effect on those organisms you are talking about. In this case, they are mosquitoes. It could be bacteria, it could be virus. Hence, the resistance of mosquito has increased. So basically, that's what you talk about. So it's not that a big deal, but please know how to apply. Variation, you tell me which variation is it. Those which are able to survive and those which are not able to survive the DDT. Those which are able to survive the antibiotics and those which are not able to survive the antibiotics. You see? Those which are able to survive the RVs and those which are not able to survive the RVs. Those which survive the RVs, they reproduce in large numbers. While those which are not able to survive, then they die in large numbers. Then those which are able to survive, reproduce in large numbers, they pass their gene of resistance to the next generation. You understand? Continued use of these ARVs or antibiotics or DDT will have no effect or little effect on these organisms. Therefore, the resistance has increased. So that's what you talk about. Let's go to yeah, guys, let's go to the next question, which is the next not next part, the last part now, the last part of human evolution, the last part of human evolution. In human evolution, what you need to talk about is you need to talk about the family to which humans belong. So this one is cool. The family to which humans belong. Did I say family, not species? Family, hominidae. And then the species is Homo sapiens. So this is basically what we usually ask you about the human. Writing the scientific name, capital letter Homo, and then small letter sapien. Yes. Phylogenetic tree, we will talk about it at last. At least, guys, you know these characteristics, but don't forget that sometimes we concentrate only on the head, only the forelimb, the characteristics there. So you have to know here. You can't just say that, let me just know. I just know two. But these are the things you have on your body. So you just think about, when they ask you about the hand, look at your hand and then you start describing. Yes, the fingers, opposable thumb, you have the nails instead of claws. You just look at your hand and then you describe your hand. When you talk about the head, you talk about the head. Ne? Look at your head and then start large cranium or large brain, eyes in front. So you look at your head and then you describe exactly what you have. Then you get the marks. Ne? It's not a big deal there. Ne? Then this one, guys, this is the difference. How does the cranium lead to intelligence? We say that large cranium means a large number of brain cells, which means that you higher intelligence. So if you have a large cranium, it means that it encloses a large number of brain cells, and the more brain cells, the larger the intelligence, or the higher the intelligence, or more brain cells suggest a high intelligence. So basically, that's how you can talk about how is the cranium linked to intelligence. So large cranium, more brain cells, large intelligence. Difference between the skull of human and the other primates, please know them: sloping face, this flat face, the canines. These ones, guys, I'm not going to talk about too much, but there's something which I need to talk about here. Position of foramen magnum, we say that is in forward position. Foramen magnum is in forward position. We don't use any other word. Foramen magnum is in backward position. That's it. Ne? Yes. Then prognanthus, the protruding jaws. Look at the jaws here. The jaws are what? Are protruding out. If I do like the line here, you see that here the protrusion is too much, you see. So here the protruding jaws, yes, well here is just straight. Yes, so this is the protrusion of these jaws. Check this black thing, ne? It shows you exactly what happens, ne? Check this black color, you see that it protrudes what? That color, yeah, it protrudes out. Check. Yes. And then what is this? Broridge. This is what called the broridge. Pronounced broridge. While here, broridge is not pronounced. So you guys, there's something which I need to talk about here. Don't say that les prognanthus. You see this word? This word was is no. You say that less protruding jaws. You can say less protruding jaws, but you don't say this one. This part, ne? Yeah, is out, ne? Because we updated this after the book is out, ne? Yeah, so we have to update then next year. Yeah. So this is, don't say less prognanthus. We say not prognanthus for the case of human, and then here you say prognanthus. Don't say more prognanthus. Or you can say less protruding jaws is correct, and then more protruding jaws is also correct. But don't say less prognanthus. You say not prognanthus. Here you say more. You say prognanthus. Foramen magnum in backward position is shape of the spine. That's all cool. Then here, walking on two legs. Walking on two legs, or walking on two hind. The word hind must be there. Walking on two hind limbs. Yes. Or lower limbs. Lower limbs. You can say lower limbs, or you can talk about hind limbs. So make sure that when you are defining what is bipedalism, so that is also a small update so that you don't mess it up. Factors that affect bipedalism, we say that the first one is, please know them: position of foramen magnum, the shape of the pelvic girdle, and then the shape of the spine. S shape allow flexibility. I explained this very well. Shape is short and wider to support the upper weight. The position of foramen magnum is in forward position to allow the spine connect vertically. Yes, I'm not reading here. You might be thinking, but still when you read, not supposed to be like cramming. You just need to know. You just need to what? To know. Sir, this lesson is recorded. They're saying, can you explain why most predators are less likely to feed on... Now, where is that question? Guys, don't ask questions which is in the passage. I don't have the paper. Unless you're asking a question which is an open question, yes. But now, if you ask me a question which is in the passage, I don't know the question you are asking me. So is this lesson recorded? Yes. It will be, you can only play it offline, maybe after a certain time. Or I will do a copy of this so that the copy can be downloaded offline. Yes, so that you can watch it offline. But you have to come back maybe after like an hour. Yeah, maybe around 7 o'clock, then you will be able to play it, yeah, or to download it. Yes. All right. Let's continue to the next question. Yes, so you guys make sure that you know. Then advantages of bipedalism. The difference between bipedalism and quadrupedalism. This one is obvious also. For position of foramen magnum in forward position. This one is in backward position. This one is S shape. This one is C shape. Short and wider, long and narrow. So you guys, you have to know that. I want to talk about the advantages. At least go when you know 3 of them. Free hands so that you can pick food. The standing upright. The eyes are high. The ground, so it's better view. You understand? You see, you think why ladies like to put on high heels, so they can see far, man. They can see far. They want to see far. Why? Because you're off the ground, so that you can see exactly what is happening there. I don't know what you're looking for, but those who put on the high heels, you must tell us what you're looking for. Anyway, paper 1 is done. There's no reproduction here, so I won't talk about it. Yes, but anyway, we're talking about this. Yeah, bipedalism, so that you show us. They are trying to show us. Have you ever seen... Let me leave it. Because people that will say that, you guys, hey, this guy is doing this. Okay, let me leave it. But basically, when you walk, you show us, you display the 6 organs, so that we see that this is my feet or this is not my feet. something like that. Yes. Here are some questions. This tabulate the observer difference between skull 1 and 2. Yeah, guys, I think you know. You talk about this. You talk about the canine. Guys, if they bring it like this, don't talk about the position of foramen magnum. Do you see any foramen magnum here? No. You only talk about foramen magnum if you see that, yes, I can see it. For characteristics of the upper limb, you see what I talked about. They're talking about the upper limb. Explain how increase in the brain volume lead to intelligence. I talked about that. That increase in the cranium increase means that more brain cells, which more brain cell leads to a high intelligence. So what is bipedalism? Walking on two hind limbs or lower limbs. Explaining how each of the following skeletal have continued bipedalism. Position of foramen magnum is in forward position to allow the spine connect vertically. Pelvic girdle is short and wider to support the upper weight. S shape to allow flexibility. Yes, allow the flexibility of the organism. You see, so it's a simple thing. Lastly, but not the least, the evidence of hominids. Fossil evidence, genetic evidence, and cultural evidence. Guys, there is a difference between don't say when they ask you about the evidences, you don't say the mitochondrial DNA. You don't talk about tools. The evidence is genetic evidence. Example of genetic evidence is mitochondrial DNA. The evidence is cultural evidence. Example of cultural evidence is use of tools. Is it clear? Yes. So fossils remain fossils. Yes. Don't talk about Australopithecus. Those are examples. This one, it kills people. Kills people. A transitional fossil. What is a transitional fossil? A transitional fossil is that fossil which have both characteristics of the present species and the species before it. Don't say that it has the characteristic of present species. No. It has the characteristics of present species and the species before it. You understand? Yes. So you must link the two. How? Or they can bring two skulls and they ask you, give a reason why this is classified as a transitional fossil. Let me see if I can find a skull here, and then I show you what I'm trying to say. I think there are no skulls here. Explain how colonial drift relate to common ancestors, how speciation lead to portals and lemurs become different species. So it's the same thing which we was trying to explain. Yes. So you see now, here you can talk about the position of foramen magnum because now you can see it. Here you can talk about the C shape. You can talk about the rectangular shape of the jaws. Here you can talk about the position of foramen magnum. You can see it. Here you can talk about flat face. Here you can talk about the sloping face. Talk about the flat face because you talk about what you see. Yes. Here you talk about the jaws. You see exactly that you can see them. What is a transitional fossil? You see. Now, what shows that this is a transitional fossil? You can talk about, maybe they can say that this is X, this is Y, and this is Z. Y is a transitional fossil. Give a reason for your answer. Because it has both characteristics of this and this. So, but now you can talk about that because now they have given you a scenario what to use. Pick up the character. Look at the canine. The canine of Z is not as big as this, but is not as small as Z. The canine of Y is not as big as X and is not as small as Z, meaning that it's just there in between. You see? Yes. The shape, this one, is not as rectangular as X and is not as C-shape as Z. You see? Yes. So basically, you have to link the two. When they ask you, please know how to apply. Don't just use what you know from the head. Use what you see in the diagram. Is it clear? Yes. Let's go to the fossils and then we finish them. How do I remember this? This is the table where you're supposed to remember. This is the table where you're supposed to remember. Adipithecus. The first one and the last one is discovered by the same person. Ardipithecus, Ramidus, Homo sapiens. There is a small video I created which shows this. It's just like it's like a song, ne? Yeah, but you are remembering, ne? Yes. So so that is the first step. So basically, Ardipithecus is team white in Ethiopia, and then Lucy, who discovered Lucy? Now, Lucy is Johansson, is in Ethiopia. You understand? Johansson, Ethiopia. Actually, the first 1, 2, where were they discovered? In the country where there was no colonization, a country where the one which was not colonized. You think about that country, and is Ethiopia. Yes. Number 2. Now you have 3 Australopithecus africanus species. All of them who discovered them, they start with the R. Raymond Dart, Robert Broom, and Ron Clark. All of them are starting with R. Is it clear? So they cannot ask you home child, date of food, Mrs. Place, and then you start thinking about Lady Bagger. All of them are starting with R. Is it clear? Yes. Taum child, Raymond Dart. It has the A, it has the A. Is it clear? Yes. So now you know it. So now the rest you can know. Ron Clark, Littlefoot. And then Mrs. Place, actually it's called Mr. Place, but I don't know if they credit if you say Mr. Place. But the recent one is supposed to be Mr. Place. But because the books, but when you are trying to do the discussion, they credited it that is Mr. Place, but it is Mrs. Place until we are told to remove it, to update it. But we will not update it until they tell us to do so. So when you say Mrs. Place, it's cool. When you say Mr. Place, I think it's also cool because lastly, they credited it. Yes. So think about it. Then Australopithecus sediba. Sediba is Karabo. That one is Lady Bagger. Please, you need to know that. Homo sapien had a man, and then Homo erectus. Please watch that small video. We will be able to know. Yes. But now, how do we use this as genetic evidence? These 6 marks, these 5 marks. Please. Evidence to support the out of Africa hypothesis. How do I use this? Let's go to the next question so that we wind up. And they're saying that fossil, guys, you need to know this. Let me just go here straight. Here. If they ask you to explain the out of Africa hypothesis, guys, you need to know that this is modern humans originated from Africa and migrated to the rest of the world. That is the first point. You need to know that. But now, when they talk about evidence to support that, is fossil evidence, genetic evidence, and cultural evidence. Fossil evidence, you talk about the oldest fossil, Ardipithecus. Ne? The Ardipithecus, Australopithecus, and the Homo habilis. All of them, they were discovered in Africa only. Ardipithecus, Australopithecus, and Homo habilis were only discovered in Africa. But now when you talk about oldest fossil of Homo erectus and Homo sapiens were discovered in Africa. The oldest Homo erectus and Homo sapiens, you have to add that word oldest. This is what I say. Also, fossils of Ardipithecus were only found in Africa. Fossils of Australopithecus Rabo, little foot, child, you can talk about too. And Homo habilis were only found. You see, this word only is very important. But once you come to Homo erectus, you talk about the word oldest. The oldest fossil of Homo erectus and Homo sapiens were found in Africa. So these two, we want the word oldest. If you don't put it, you cross it. And then this one, you don't add the word oldest because we will ask you what about the newest? So we cross it. So don't add here the word oldest. Guys, here we are too strict. Here, you only talk about Adipithecus, Australopithecus, and Homo habilis, only found in Africa. But when you talk about Homo erectus and Homo sapiens, you talk about the oldest fossil of Homo erectus and Homo sapiens were found in Africa. Mitochondrial DNA, that's genetic evidence, mitochondrial DNA. Now here you just talk about where the oldest female was discovered. Is it clear? Yes. Then cultural evidence, you talk about the oldest tool, which is the one were discovered in Africa. Yes. So basically that's what you need to talk about. Yes. Lastly is the phylogenetic tree. I'm gonna show you the questions in this booklet. The questions they can ask you, they won't ask you apart from these questions I've given you. This book was designed to make everyone pass, and not just a pass, but you pass with a distinction. Eh? Yes, we pass with a distinction. Lastly, you guys, are you listening or you are busy asking? You are busy asking numbers? Muzanzi. All right, guys, make sure that you make Muzanzi shine. Eh? Yeah. All right. Here are the questions. These are the questions we're going to talk about. What is the name of the diagram given above? It's called the phylogenetic tree. How many genera are shown? When they talk about genera, you are looking at is one. Adipithecus is the first genera. Australopithecus the second. The first name, Australopithecus. Second is one. This is two. Then 3, Paranthropus. Do you have Homo? 4. Do you have another one? Homo? I think there are 4. Unless there is something I've not seen, but you look at the first name and you don't repeat it. So we are done with the genera. What is the common ancestor of Homo sapien and Homo erectus? Common ancestor of Homo sapien, Homo erectus, and Homo sapien. So you move back until where they join, Homo ergaster. So Homo ergaster is the common ancestor of Homo sapien, Homo sapiens, and Homo sapiens and Homo erectus. Then the next one is, what is the common ancestor of all species? Here you just look at it. Is the common ancestor is Adipithecus remidus. It's the common ancestor of all species. So it is easy that way. Then the next one is they're asking you, give give 2 pairs of species that competed for resources. When they say 2 pairs of species that competed for resources, they're talking about 2 species which lived at the same time. Sorry. Yeah. So for example, you just choose. If I draw a line here, I will see that this species, they existed at the same time. So you just look at the species which existed at the same time. Yes. Then they're asking you, so then name 2 species that competed. Okay. Which species first used tools? Guys, this question depends on the question they have asked you. The species which first use tools is Homo habilis. But if they give you a diagram and then they show you that, okay, this is the other one tools. These are the species, okay, this is other one tools. If this is, where's the tool? The tools are here. So it's 2.5 billion years. It's 2.5 billion years ago. So which species first used tools? I have to draw a line and then I find out which species first used tools. So here is showing you is Paranthropus ethiopicus and also Australopithecus afarensis. Afarensis africanus, something like that, based on the diagram. You understand? Yes, but if there is no diagram, then it is Homo habilis, the handyman. Then they are asking another question. Species to walk on two limbs. The species to walk on two limbs is Homo erectus. But if they give you a diagram, you have to look at it. Bipedalism is here. So which species first walked on two limbs is the Australopithecus anamensis. So Australopithecus anamensis is the one which first used the two limbs. So then they're asking you another question. How many million years ago did Australopithecus africanus appeared? Did Australopithecus africanus appeared? Yeah, when you talk about Australopithecus africanus, Australopithecus africanus, they're saying that which species first walked? Okay, how many million years ago did Australopithecus africanus appeared? So you come back here on the diagram and then you see where did it get appeared. Australopithecus africanus, where is it? Africanus is here. Ne? When did it? Which one? Yeah, it's here. So you find where did it get appeared? Maybe is 3.2. 3.2. Remember the 0 is this side, so you count going downwards. Ne? You count from 0. So 0. Maybe is 3.2. Yes. Million years ago. Million years ago. Then they are saying that when did the Homo habilis became extinct? Now you look at where it got extinct. Homo habilis. Where is Homo habilis? Homo habilis is here. When did it get extinct? So you draw a line touching here, and then maybe is 1.6 because 1.5 is around there. Maybe it's 1.6 there. You understand? That's when it got extinct. Then they are saying that how many million years did the Australopithecus africanus existed? Here now we don't say million years ago. You just tell us how many million years, because it's like asking you how old are you. You're telling me 20 years ago. No. Australopithecus africanus is here. So now I have to come where it started, maybe 3.2, and where it ended, maybe 2.5. So I'll say 3.2 minus 2.5. Maybe it's going to become 0.7 million years. Not million years ago, because they're talking about how long did it exist. Yes. Then they are saying that how does the brain size show evolution trend? We talked about that the brain size means that the higher the brain size, the higher the number of cells which lead to more intelligence. Give one example of transitional fossil. An example of transitional fossil is Carabo. We can talk about Carabo, Australopithecus sediba. How does dentition show the diary of the organism? Dentition shows the diary. How does it show? If you have sharp canines, it means that you are feeding on raw or raw food. Yeah, food which is raw. You understand? Yes. Raw food. And this food is tough, therefore you have to use sharp canines like this. Sharp canines, yes. I think that's the question. Then let me just go here just lastly. How name the family to which the organisms belong? Hominidae. And then describe the cultural evidence. We talked about it. How long did it, the most recent, how long ago? So we talk about how many years ago. Yes. And then explain the possible reason why Homo ergaster was placed between Australopithecus afarensis and Homo. Then it means that it's a transitional fossil, therefore it has characteristics of both this and this. Yes. Explain why fossil of organisms that existed from 4 million years ago to present time are used to support out of Africa hypothesis. How do they explain how? So from 4 million years ago, Therefore, you talk about the oldest fossil of, you talk about the fossil of Australopithecus afarensis, Australopithecus, just say Australopithecus, and the Homo habilis were only found in Africa, while the fossils of Homo sapiens and the Homo erectus, the oldest, were found in Africa. Basically, that's what you need to talk about. Guys, I think I'm going to stop here. I wish you the best. I wish you the best. I repeat, I wish you the best, and I bless you with the distinctions. I know that you'll make it. Don't let me down. Remember what I've said. Watch this video again, so that, because when you watch it, basically you are revising. You understand? You are revising, and I know that you're gonna make it. May God bless you, bless your hands, and bless your future. I stay as MSID. See you again at the university courses. And tell your brothers and sisters to join the YouTube, so that those guys can be helped the same way you are helped. Thank you very much. Until we meet again. Welcome back to our class, guys. Today we're gonna be talking about the memorandum of paper 2, so that we see how did we manage to score high marks on this paper. All right, without wasting time, I hope you guys enjoyed the paper. Yes, you enjoyed the paper. If we look at our predictions and the discussion which we did, we really covered 99% of the paper, meaning that whatever was set in the paper, we covered it. And if you go back and play the video which we did our last minute, you'll find out that yes, we have covered each and everything. Let's go to the paper without wasting time. It is MSID. Thank you so much, guys, for subscribing, for liking, everything. Yes, let me know if you have a question. It is a live class. I will be able to sort out that question you are talking about. All right, the first question is saying that they are saying that which one of the following is a characteristic of African apes and human in common, meaning that which of the two is in common. We say it is opposable thumb, meaning that we said it, you remember that. So 2 marks there. Meiosis is biologically important. We say that to produce haploid gametes. We said it, we look for it. Where is it? Is here. So we get 2 marks there. We're saying that reproductive isolation characterized by animal displaying specific action to attract the mating pattern. Remember, even I gave you an example that except the humans, they like to copy, and then they say that, okay, we call it species specific courtship behavior. And I said that the way how you do your dating is not the same way how frogs, how dogs, how cats, and I said that except but humans, they like to copy. Yes, all right, so this one becomes the most correct answer. So we go to the next one. Then they're saying that which one of the following genotype is homozygous, dominant, homozygous. So I need to look for letters which are the same, but they say the dominant. So this one is dominant, this one is recessive. So it is C. So C becomes the most correct answer. They're saying that how many alleles control blood groups have been singing this song. We say there are 3: allele for A, allele for B, and allele for I, allele for O. So we said it, 2 marks there. Guys, the paper was flowing. Okay, which one of the following is an example of discontinuous variation? We say that anything which you are born with and it's exactly the same way it is. We say the blood group. We say the height is different, the skin color. You are born yellow bones, now you are dark skinned like me. Color, the hair color keep on changing. You understand? So the answer here is B only. If you are blood group A, you are born blood group A, you will die blood group A. So that becomes the answer. So 2 marks there. Don't get disappointed if you see that your answer they are not like the same. Yeah, still at least if we get maybe 80 out of 8 out of 100, maybe 80%, that's a distinction. Yes, so to fall in a distinction doesn't mean that you have to get 100%. No, it's 80%. All right, we continue. Which one of the following is a correct description of stem cells? Stem cells, remember that we say that these are cells which are unspecialized. They have ability to divide and form specialized cells, meaning that they have specific function. So we have to look for that. So we have to look for cells, specialized cell that divide by mitosis, divide by mitosis to produce cells that become specialized for specific function. So B becomes the most correct answer. We go the next question. We are saying that a list of different lines of evidence to support the evolution given by we say that the 3 evidences, we say that fossil evidence, genetic evidence, cultural evidence. I don't need even to read the rest. Those are the 3 we talked about. So we look for it. 2, 3, 4 is C only. So we go to the next question. Say that the police arrested 2 suspects accused of housebreaking. Their DNA profile was analyzed and compared to the profile DNA, okay, that's collected from the crime scene. So they're saying that what conclusion can be made from the DNA analysis conducted by the police? So we need to look at, are they corresponding? No. They're not corresponding. No, they're not corresponding. No, they're not corresponding. No, they're not corresponding. It looks, but still this one is a little bit up. No, they're not corresponding. No, they're not corresponding. What corresponds here? Nothing. Only this one corresponds, but this one no. This one corresponds, but this one no. The answer is no, meaning that there is no suspect. The suspect here, no one who did that. So the answer here we have to look for neither of the suspect are positively linked to the crime. So D becomes the most correct answer, because there's nothing. If they were matching, they're not matching. Therefore, the answer becomes none there. So those 2 marks. So we got the next question, the terminologies. I hope guys you got 18 out of 18. If you got 18 out of 18 or 14 out of 18, congratulations. Clap your hands. All right, we got the next question. Alternative form of a gene, we say this allele. Allele, DNA containing organelles found only in plants, chloroplast. Plast. Yes, they are saying that the diagram representation showing the possible evolutionary relationship among the different species. We call it what? Call it phylogenetic tree. Yeah, we call it phylogenetic tree. Allele that is masked in a phenotype, masked meaning that if we say that if the vice president is there, if the president is there, the vice president cannot talk, meaning that there is that allele which is being masked. So we call it a recessive allele. So we say recessive allele. They're saying that the permanent disappearance of species, we call it extinction. The deliberate breeding of organisms for desirable characteristic, we call it artificial selection or selective breeding. Artificial selection. They're saying that genetic disorder characterized by absence of blood clotting factor. We said it. Then we did a lot of examples here. Hemophilia. Someone is gonna say, does spelling count? It counts. A group of organisms of the same species, same species that habit, a group of organisms of the same species in a specific habitat at a particular time. How do you call that? We say that it is called a population. Population. We say that, hey guys, give me happiness. I told you that when I come back here, I need happiness. I need happiness. Yes. Chromosomes pair in 1 to 2. We call it autosomes. There's nothing we didn't do, guys. There is nothing we didn't do. A representation of number, shape, and the arrangement of all chromosomes in the nucleus. The moment you see the nucleus, that a nucleus we said is called a karyo. So it's a karyotype. We talked about it. All right, they're saying DNA containing material in the nucleus of a non-living DNA. How do we call that? We say that we call it chromatin network. Chromatin network. Before the DNA divides, before the cell divides, it is chromatin network. Yes. Because that's why they said a non-dividing cell. So it's B only. They're saying that location of DNA. Where do we find DNA? We find in a nucleus, in the chloroplast, and the mitochondrion. So the answer is both A and B. We continue. They're saying that the first Yo, alhamdulillah. People saying 3 out of 10. No, don't. Others saying 10 out of 10. Don't cry, my man. Who is there? Zanele. And then who is that? Guys, Kahiso. All right, guys. Tabom. All right. We continue. They're saying that. By the way, alhamdulillah means praise to God. Yes. They're saying that the phase between two consecutive cells, there is nothing here. Remember, it's called a growth phase. Inter in between the two. You understand? That's why I said use IPMET. Interphase means in between the two phases, the two growth, the first growth and the second growth. That's it, interphase. So the answer here is none. 2 marks. We go. We kill the paper. We said we're gonna smash the paper. The diagram below represents two biological processes. So if you look at this, we see this is transcription. Why? Because you are forming a messenger. Check U here and then check T here. So that is transcription. And then if you look at this is T, this is T, T meaning this is DNA replication. So okay, there are some questions I ask you to do. Diagram 1, transcription, we said. Diagram 2 is DNA replication. Identify X. What is X? If you look at X, where is X? X is a messenger RNA. They are looking for this molecule. It's messenger RNA. This is messenger RNA. So correct. Someone saying that we'll try next year. For us, we will be here always to help you. We will be here, God willing, helping you so that you get what you want. Yes. So keep yourself on Thundereduc and other channels, Facebook, TikTok. Yes, we are always there. Yes, but there are some people who are having different channels of Thundereduc on Facebook. Those people, there are some people who have opened. So guys, please watch out. They're asking money from people. Please make sure that you don't become a victim of sending money to people who are requesting you. They pretend to be Thundereduc. Yes. So we continue. If you are going to send the money to Thundereduc, for example, you are buying a book, it must be an account which is linked to Thundereduc, no e-wallet. Yes. All right. Thank you. We continue. We are saying that identify the molecule X. So we done that with that. Molecule X. What is molecule X? Is messenger RNA. We say this is the messenger RNA. Natural shape of Y. We talked about it, and I said only one answer. natural shape of this DNA is double helix. Double helix, nothing else. They're saying that the phase of cell cycle during which the process in diagram 2 occurs. Diagram 2, that is, these are the questions I asked you. We said it's interphase. When does it occur? It occurs during interphase. Full name of the nitrogenous base that is found on molecule X, not Y. Molecule X, meaning that it is found on messenger RNA, but is not found in the so the answer here is uracil. They're saying name the sugar found on X. Sugar found on X is the RNA, therefore it is ribose sugar. It's ribose sugar. Someone is telling me that, hey sir, write nicely. Sometimes also when we are marking you is the same thing, you don't write nice. All right, bond Z. Obviously when they ask you bond Z is this one, is this one, so it is weak hydrogen bonds. Weak, I told you in the word weak must be there. Hydrogen bond. Yes, please make sure that is there. They're saying state the number of codons. We say that codons are three bases on X, so we're gonna count them. How many codons are there? This is 1, this is 2, and that is 3. So I see, did I say on X? No man, that is not X. Let's check properly. On X, we are seeing it here. It is 1, it is 2, it is 3, it is 4. So it's 4 bonds. So the answer here is 4. They're saying that not 4 bonds, 4 codons. Then we continue to the next question. 1.5. We go to 1.5. In a plant species, gene coding for plant height has 2 alleles. The allele for tall is capital T, the dominant over allele for dwarf, which is small t. The gene for flower color has 2 alleles, where allele for red, okay, and then allele for is dominant over allele for white, which is small r. Dwarf and white plant is crossed with dwarf and white, okay, crossed with homozygous tall, meaning is like that, crossed with the heterozygous red, meaning that red, remember, is heterozygous. Okay, state the characteristics that is being investigated in this. So we have height. First of all, we have that, and then someone is gonna say height. Okay, cool. And then we have height, and then we have, they said flower color. Cool. They're saying that give the genotype of the plant that is homozygous tall and heterozygous red. Give the genotype homozygous tall. They said homozygous tall and heterozygous red. So it's like that. So meaning that you get a mark there, you get a mark there. Below is incomplete Punnett square. Okay, we say that what is the genotype of X? I will just say, remember this goes with that, so I'm gonna say capital T, small t, capital R, small r. Get that 2 marks. A is 1 mark. Then they are saying that the phenotype for Y, meaning that if the phenotype for Y, I will need to know what is Y. So it's gonna be capital T, small t, small r, small r. Capital T is tall and white, because small r is white and white. So it's gonna be tall and white. This is the answer. I was just using this to identify what is it. What is this Y? So it is tall and white, and then this one you get that mark. That is dihybrid cross. I told you it won't be a big problem. This one, the way we set it, the way we stitch it is different from the way we set it. It's simple. That question is done. We go to the next question. Don't tell me that you didn't do this, because we repeated it several times. Diagram 1 and 2 show the cells of the same organisms in different phases. they are not on scale. I told you when you see it, these spindles must be in, look, in these sides, facing that side. And I told you that when this diagram comes, what should you do? Just rotate your paper, ne? Just rotate your paper to look like this, so that now these things, these spindle fibers and the centrioles, they are here. So don't you see that there is a proper space in between, which shows that this is the anaphase. And then even this one, you see that there is a proper space. This is also anaphase. So if you are confusing it, now at least you know what you're supposed to do, for those people who are using this paper as a revision. Okay. Now, we continue. We are saying that state the phase of the state one place in human body where meiosis occurs. Meiosis occurs, we said the testis. We said in humans, we said it is testis. This is singular, this is plural. Or ovary. They said in human. They didn't say a male or female. So we go. Structure P is spindle fibers, or spindle threads. Structure Q, we say it is centrioles. Yes, a centriole. Identify the phase of cell division that is being represented. I showed you, meaning that they are being represented by what? Diagram 2. Diagram 2, remember there is a space here. I told you to rotate it. So meaning that it is, since these are chromosomes, they are still together. Chromosomes, we talk about chromosomes. Therefore, it is, they are still 2. Meaning that they are separating these homologous chromosomes. They are separating. It means that separating apart. So it means that is anaphase. Since they are still 2, meaning that it is anaphase 1. Anaphase 1. Remember we said if they are still double, then we say 1. If they are single, then it is 2. You understand? We do the opposite, ne? Like this one, ne? They are chromatids. The chromosome is the one which separates. So it's single. Therefore it is anaphase 2 in diagram 2. In diagram 1. All right. So in diagram 2 is anaphase anaphase 2, anaphase 1, because their chromosomes are still double. And they're saying that how many chromosomes will be found in a somatic cell, of this organism. They didn't say that this is a human cell. That's what I told you guys. Don't talk about human cell 46 chromosome when they say that. This diagram. So how many are they? 1, 2, 3, 4. In meiosis 1, those cells, those chromosomes are going to be 4. Why should you talk about 46? Did I say that this is a human cell? No, I told you that. I hope you guys didn't disappoint me. So you deserve a trophy. Thank you so much, Mona Lisa. All right. Oh, I like that name, ne? Mona Lisa. Yeah, you know that Mona Lisa. All right. State one difference between the process occurring in diagram 1 and diagram 2. State one difference between the process occurring in diagram 1. Diagram 1, meaning that you just gonna talk about here the chromosomes separate, they move to opposite poles. You understand? While here the chromatids move to opposite poles. That's it. Simple. This year, May, June, they did how do I say it? They need a trophy for you guys. You need to be getting good marks. At least you continue with your life. Most especially people who are writing May, June exam, these final exams. There are those people who are, maybe they have gone back, or they are upgrading. Maybe something did come up right, or they missed up the exam. They are not fresh. Those are people, it could be their first time, but their age is a little bit higher than those ones who are writing November, October, November. So you see, so thank you so much, the person who said. However much, yes, there are some questions which are not good for those who didn't prepare. But if you prepared with us, it means that you got it. All right. Chromosomes in diagram are genetically different due to they are genetically different. Chromosome in diagram are genetically different due to crossing over. All right. I told you don't go to the paper without knowing. without knowing the process of crossing over, and we discussed it. Describe the process of crossing over. Talk about when does it occur. In prophase 1, what happens? Homologous chromosomes, they come together or they align. They exchange their chromatids. Yes, homologous chromatids at a point called chiasma. Yes, and then we say that after that, what happens? They, after exchange their genetic material, they separate in such a way that new genetic material from both is being shared. So basically this process, even I brought a diagram to explain to you. So guys, I hope you got this process right. You see such process, and then you get your mark. So yes, those people are doing the revision. You can take a screenshot. You pause it, take a screenshot. Now you know what you're supposed to write when they bring such. They're saying describe the non-disjunction. I explained this. How does it lead to Down syndrome? And I said, hey, when these are some of things which you might not miss out. Yes. So I said it here. Where are you? Yeah, after this. Yes, I explained it here. First of all, you need to explain to us non-disjunction. When does it occur? What happens? So basically, we say that you explain that it occurs in anaphase. Yes, in anaphase. It can be anaphase 1 or anaphase 2, meaning that the failure of the chromosomes to separate. It tells us that the chromosomes fail to separate. Yes, meaning that one result in one cell having an extra chromosome, meaning that it's going to have an extra chromosome. It's going to have 3 instead of what? Instead of 2. Or another one is going to have 1 instead of 2, meaning that one cell is going to have an extra chromosome, and then another one is going to have less of chromosome, meaning that it's going to have 23. Another one is going to have 24, because it's supposed to be 23. Another one is going to have 22. So if this 24 fertilizes a normal ovum or a normal gamete, yes, because we don't know what is this, a sperm or ovum, a normal gamete of which is 23, is going to result in a cell. Is going to result in a cell having, or an individual having, 47 instead of 46. And if this non-disjunction occurred on chromosome number 21, therefore it becomes trisomy 21, which we call Down's syndrome. Yes, so that's what you're supposed to explain. I explained it. I can't repeat it now because take a screenshot. Because we're just discussing what are you supposed to be linked in. Yes, I hope you guys, you got 10 out of 10. There we go. The next question 2, question 2.3. The diagram below represents the stage of protein synthesis. Name the stage of protein synthesis there in diagram 1. In the diagram, we say this is the moment you see these boxes. Remember I said that amino acids, they either circles or they are boxes. They brought them as boxes. So cool. State the process. We say this is the translation. Name the stage of protein synthesis. So we say this translation. Then they are saying that name the process. Name the stage of process. Okay. State the site where the process named above occurs. It occurs on the ribosome in the cytoplasm. In most cases, they want to see the word ribosome. Sometimes they want to see cytoplasm. So that's why I told you that I prefer you to say that, for you to say that ribosome in the cytoplasm. Definitely they will give you. If they want this one, they will give you. We have to be there. You said diagram 1. Someone was okay. I was just reading one of the comment. The diagram 1 opposite chromatid put opposite pole. Diagram 2 chromosomes are put opposite pole. Yeah, that's it. That's what I said. Yeah, that's what I said. Tabo, Mona Lisa. That's what I said. Cool. It's cool. All right, we go to the next question. Which question is that? Describe the role of X in protein synthesis. Role of X. What is the function of this in protein synthesis? In actual sense, they're asking you, what is the function of transferrin A in protein synthesis? It brings the required amino acid or the specific amino acid to the ribosome. Yes. So if you want to add, yes, you when the anticodon of it matches with the codon of the messenger. When the codon of the messenger matches with anticodon of molecule X, yes, molecule X will bring the required amino acid to the ribosome. If they're asking you to describe that part, just to remove that part of what? Of translation. Yes, that's what they are looking for. So basically, this is what I'm talking about from our distinction material book. This is what I'm talking about. I told you that this book, I don't want to show you everything, but if I want to show you each step by step, you would have seen. So you see, this is what I'm saying. Transferrin A carries specific amino acid when the anticodon matches the codon of the messenger. Therefore, that molecule X will bring the required amino acid to the ribosome. So this is what I was trying to say. You see how many marks are there? There are a lot. All right, we continue. This identify the complementary base from left to right, representing anticodon 2. Let me first clear it up, and then we see anticodon 2, anticodon 2, anticodon 2. So anticodon 2 is anticodon 2. We are seeing that CAU. They're saying identify the complementary base. Identify the complementary base from left to right, representing anticodon 2. So it's gonna be C goes with the G, A goes with the U, and U goes with A. So you get the mark there. They're saying on DNA codon 1. So I go to codon 1 is, if I pick this one, it's gonna be codon 1 AUA AUA AUA. So I need to convert it into DNA. A goes with T. Remember, this is DNA, and then U goes with A, and A goes with the T. Third. Table below represents the anticodon for different amino acid. The mutation, I told you this one is coming. Make sure that you do. Then they're saying that Maria, we have not finished the paper. Why do you say that you didn't? You failed it. Ayanda, I see you. All right. They're saying that a mutation occurred during replication. The mutation, I know that this one is gonna kill some people, and that's why I emphasized it. Make sure if you don't do it, you are gone, man. All right. They're saying that use the table to explain how this mutation will affect the protein. So we want to know whether the protein remains the same or is gonna change. But remember, you need to show us where did it change, which codon number or which anticodon, which code, all those things. Yes, but now they have given you, this time they have given you what we are supposed to get. So what they say is the mutation, what happened to the mutation? The mutation changed this to this. All right, let's find out. Prorine to arginine. So this one, what does it code? I'm just doing this so that you guys are able to get it right, ne? And then what is arginine is CGU. What is this one doing? This one is gonna go back to CCU. So C goes with the G, and then C goes with the G, U goes with A, and then so this is the anticodon, ne? So this is codon. Because they say that use this explain how the mutation will affect the protein. The mutation occurred during replication. So replication, it means that it occurred on DNA. That's why you want to find out which code from DNA where the problem came from. Then this one, C goes with the G, G goes with the C, U goes with the A. So this is codon, meaning this is messenger. And then now let's go to DNA so that we know exactly what happened. They're saying that C, A goes with T because it is DNA. So it's gonna be G, C, it's gonna be C, G, T. You understand? So what happened if you look at it? The second base C was replaced with G on DNA during replication. So that's the mark. You have identified what happened. You understand? But they say 7. So it means that you have to show which is transcribed as GGA and GGA respectively on what? On messenger. And then CCU and G and CGU respectively on the transferrin A. That's anticodon. Hence proline and arginine, which are 2 different amino acids resulting in different protein. You see? So they wanted you to identify what happened here. They will give you a mark there. You translate it to messenger. They will give you a mark there. They will give you a mark there. And then, yes, that codes for that. They will give you a mark there. And then they will give you a mark there. So they will keep giving you marks there. So you explain what happened. So that mutation, you always, if you see that the amino acids are different, it results in the same protein. Sorry, in different protein. If the amino acids are the same at the end, result in different protein. All right, I hope you guys got it, yeah. We got the next question. That is question 2.4. They're saying that in some chicken, feather colors are controlled by alleles of alleles feather W and black. This is white, and then this is black. But look at the letters are both the same, meaning that they are dominant. So it might be related to incomplete dominance or codominance. If the heterozygous white chicken is crossed with the homozygous black chicken, offspring have the feather that is both black and white. And then they are saying that known as so when we are using the example, we use that one of flowers. Yes, we use that one of flowers. And then we explain it. Explain why this inheritance is an example of codominance, because it results in offspring having both colors, that is white and black and white, meaning that it's a mixture of the 2 colors. So I told you that is a mixture. It's a mixture of 2 colors, that is black and white. Then we got the next question. They're saying that explain why this the inheritance of blood group in human shows the same type of dominance. Explain. Yes, because when A is crossed, homozygous is crossed with B homozygous, they will result in AB, which is the codominance. That's it, meaning that A is dominant, B is dominant, and then you form AB, which is, it looks the same like this one, because white and black, then you produce a mixture of the 2. You see that this looks like that. White chicken is crossed with the eminent chicken. Using the genetic cross, meaning the eminent is this, is this, yeah, cool. Use the genetic cross to show the phenotypes. This one is simple, guys. Remember I told you that when you have this, you have 3 marks free. So P1, F1. So you got, I think you got that mark before you go to the paper. You went to the paper, they said phenotype, we said genotype, we said meiosis, we said gametes, we said fertilization. So we say that you got this mark also. So those are 2 marks. And I say that for phenotype, you go to the question, they say that white chicken. So I said white chicken crossed with eminent chicken. That's another mark. You see, 3 marks. That's what I said. So you got it. So now they're saying that genotype, we say that white chicken, they said white is W, isn't it? Is W, so it's gonna be capital W, and eminent is capital W, capital B, because it's a mixture of the two. Then they say that meiosis, we say that capital W, capital B, then we say that capital W, capital B, then these are gametes. Then we say this cross with this, capital W, capital W. You can use the Punnett square, capital W, capital B, correct, capital W, and then this with this, capital W, capital B. These are offsprings. What do they want? They say the phenotypes. So if they were looking for the phenotype, so I will come back here and I will say that if they're looking for phenotype, I have 1, 2 white chicken, and then 2 eminent chicken. Yes, 2 eminent chicken. So basically that's it. You got it. So they start to mark, but you went to that paper when you know they wanted to see this could be a compulsory mark, that if you didn't get it, instead of getting because there is a lot of marks here, there is a mark there, there is a mark there, there is a mark there, so there is a lot of marks there. All right. Then we got the next question, which is question 2.5. Question 2.5. They are saying that hypophosphatemia is a sex linked. They said it's a sex linked. You see, I said whenever they say sex linked, you use X and Y, inherited condition that is caused by dominant allele capital X and on X chromosome. They said dominant. You see, dominant. Yes, it is as a result of abnormal 2 level of phosphate. It's as a result of abnormal low levels of phosphate in the blood, which can cause rickets. They're saying that the recessive allele small h code for normal, meaning that if you have to be with both like this to be normal, or you have to be with this to be normal if you're a male. All right, they're saying that male without. Okay, they give you give the type of diagram shown. How do you call this? We call it a pedigree diagram. They're saying gonozome found in male, gonozome found in male is Y. Yes, it's gonozome, meaning that the chromosome which is found in male. Yes, yeah, male only. They don't want you like this. They said male only, meaning they are looking for that chromosome which is Y. State 2 effect of this, they said it here. It will cause rickets and also causes abnormal low level of phosphate in the blood. So they say that how many individuals in F1 generation have this condition. We say that the one which is shaded, remember this is shaded, the one which is shaded is suffering from the disease. They said in first F1 generation. This is F1 generation, but this got married, so meaning that it is this one, and this one, meaning there are 3 individuals in there. So 3 individuals. They're saying that all possible genotypes of individual 3. All possible genotypes of individual 3. Where is individual 3? Let me clear it up. We say that individual 3 is normal, and normal, they said that you're supposed to have the recessive allele, and this person is normal, so this is a male, so it's gonna be like that because he's a male. And then individual 4 is having the condition, meaning that is gonna be X capital H and then X small h. Why not X capital H? Because they produced a person who is a female and is normal, meaning that this one is like that. So meaning that one came from the father, one came from the mother, so the mother can't be like this. That's why this is the only option which is there. So you get 1 mark there and you get 1 mark there. Explain why all the daughters in one will have will have this condition, and they say the 6 marks. So do you know what they want? They want you to cross, do a genetic cross. Dora, yeah. So we are going to show you how are you going to explain. We say that the male, who is the male? The male is here. We say the male is what? Is sick. Meaning that if the male is sick, it's gonna be capital H. Meaning that the female is like this, isn't it? And then the female is like that. So when I cross, okay, after that you're gonna explain. When I cross, yes, okay, I have that and I have that, and then the female is gonna be small h, yes, you understand? So now we want to find out if all the Doras are going to be sick. Then from there we're gonna explain why. Then this gonna go like that, capital H, small h, okay, that is a Dora, is sick because it has a capital H. Then this gonna go with that, so it's gonna be capital H, yes, and then small h, okay, this is also a Dora. Then now this with that is gonna be capital H, small h, and then y, this is normal. Then this one with that also gonna be capital H, small h, and then y. So you see the boys are normal while the girls are sick. Not sick, they have that condition, you understand? Now how are you going to put this in wordings? You understand? So when we come back here, we see that the boys are normal, you see, and then the females, all of them are sick. So what does it mean? It means that the males have a dominant allele, you understand? And then the females, they have 2 recessive allele, you understand? So the male will only transfer the Y chromosome to the males, you see? They will only transfer the Y chromosome, these Y chromosomes, to the males. Are we together? Meaning that all the X chromosome which are dominant, causing the individual to be sick, are going to be transferred to the females, you see? The ladies, the female, the mother, will only transfer the recessive allele, which will be masked by this dominant allele, causing all the females to be what? To be sick with that condition. Which condition is that? They said hypophosphatemia. Yes, I think now you get it what I'm trying to say. Yes, I wrote 15 line, yo. Yes, it's fine, because you were just supposed to explain to us that the male have the dominant allele in this case, and then the female has 2 recessive alleles. The male, the Y chromosome is going to only be taken to the males, meaning that the dominant allele on the X is going to be transferred, or is going to be inherited to the female offsprings. You understand? The Doras. You understand? Yes. The recessive allele from the mother would be masked by the dominant allele, which was transferred, which was inherited from the father, making all the females to be having that condition of hypophosphatemia. Hey. All right. So someone saying, yeah, I get it. All right. Thank you. Since you got it, thank you. We go to the next question. Is just it was a dominant allele, and then I said that when they bring dominant allele, people go off. All right, we continue, guys. Thank you so much if you got it. And thank you for not if you get it. If you have a challenge with the question, you tell me. All right, they're saying that describe the describe. They are saying describe the process of cloning. So what do you say when it comes to cloning? We talked about the process of cloning, and let me just use the distinction material book, and then just to show you what you are supposed to talk about when it comes to the process of what? The process of what? Cloning. So we said that we use a sheep, which we call the dolly sheep, but it's still the same process. Use the same what? You see the same process of cloning. It is. So it means that you have to first identify which cell are you going to use. You understand? Yes, you have to first identify the somatic cell. Get a somatic cell. We said get a somatic cell from an individual whose characters are needed. Yes, step number 2. Then remove the nucleus and then keep the nucleus. Then step number 3, get another cell, a mature ovum from another sheep. Yes, and then after that, basically remove the nucleus and keep the cytoplasm, yes, or the casing. And then after that, combine the nucleus of this and this and the cytoplasm of the cell from the other sheep, then with the mild electric shock. Then after that, it will form a zygote like cell, which is allowed to undergo mitosis to form a ball of cell. So those ball of cell is gonna be split into small embryos, which are going to be inserted into the what we call the surrogate sheep, so that now the surrogate sheep can develop that embryo into a mature or a baby of that sheep. So basically that's what you're supposed to explain, link to this, and explain that this is what we want, nothing else. Yes. And H, there's someone who said that H, you talked about it, but I neglected it because I knew that it's gonna come in objective. Hey guys, sorry. And we go to the next question. Name 3 benefits of cloning. Basically, the benefits of cloning, we can use it to get medicine. Yes, like this. Where is that? Yeah, to get medicine like this. Basically, they are looking for this. To get medicine. Where is it? Yeah, medicine. Yes, medication. Basically this is what I wanted to say. This cloning of insulin. Yes. We can also be used for producing identical organisms, which can be used for research purposes. Yes, process of cloning. Yes. And then we can also use basically what else can we use for cloning? We can use it for research. We can use to do the medication. Yes. And then we can use it to produce. There is this scenario also which occurs, like preserving the endangered species. Like if the species is about to get extinct or is about to get extinct, we can bring that genes and then we clone them, and then we come back, we bring them back to life, like so that we multiply them in large numbers instead of getting extinct, like one organism remaining or 2 organisms. So we can do the cloning so that we multiply them to be many. Yes. All right. Someone said doesn't know the process of cloning. I did a full video about cloning. So just go to YouTube, type cloning process, and with the word Thundereduc, it will come. Yes. Those people who are using the distinction material book, this book, then it is being explained based on this distinction material book, meaning that at least, yes, you'll be able to use the same information. Let's go to question number 3. What are they saying? Question number 3, read the extract. Sickle back fish are small fish found in the ocean of the freshwater lake. The one in the ocean have spike fin for protection against predators until about 100 years ago. Most of the stickback fish live in the ocean, but the last ice age ended. The melting ice created new lakes and some population swam into the lake. The connection between the ocean and the lake eventually died up, trapping the stickleback population in the small. lakes for thousands of years. Over time, the stickleback fish population in the lake lost the spike fin. Okay, how will Lamarck explain this? Remember, this is absence of this. How did they become with absence? Remember, Lamarck uses law of use and disuse and the inheritance of acquired characteristic. So what are they saying? You need to explain based on Lamarck. So you're gonna explain to us that there was no variation among these fish. All of them had what? Had the stick back or spike fin. All of them they had the spiky fins. Okay, that's step one, meaning that there was no variation. All the fish they had the spike fins. So number two, these fish they stopped using their fins because there was no need for them to use. Remember, they were using them for what? They are using them spike fins for protection. They are using them for what? For protection. But where they went, there were no predators which were against them. So they stopped using their fins because there were no predators acting on them. So eventually the spike fins started to decrease and then eventually disappear. You understand? Yes. Then they passed this character of no fin to the next generation, and then all of a sudden, all fish in the fresh water they had no spike fins. Yes. So you explain based on what is being asked in that regard. Yes. So you link. You don't use giraffe. You can use the same concept, but you in this case you talk about the spike. And I said that last time, I said that they might bring it in the disuse. You have to use one. Either use it and then they develop, or you don't use it and disappears. Here you don't use it, then it disappeared. Why? Because there was no need for them to use the spike fins to protect themselves from predators, because where they were no what? They were no predators. Therefore they stopped using them, and then all of a sudden the fins started to decrease in size, and then they disappeared completely. They passed this character to the next generation. The next generations, they were born without the spike fins. That's Lamarck's theory. The diagram below represents the skull of pelvis of human and chimpanzee. No particular order. Cool. They're saying the name three structure differently. We did this several times. Okay, we say that draw a table, a complete table. Some of you did it just a T, 0. They're saying that is A and that is B. So they're saying A, you tell us. We use the same example. You people. We say that they have large canines, small canines, rectangular palates, U-shaped palates. Then you see the foramen magnum is in backward position. The foramen magnum is in forward position. What else do you want? You are sure? Someone saying that I'm shocked I didn't see this question. Sorry. They're saying state Y pelvis C is regarded to humans. Remember, we said that the one for human is short and wider. Short and wider. That's all. Guys, this is the things. I don't want to go there. This is the thing we said and they emphasized. Say that, hey, when I don't make mistake to go there. Yes, I gave you that actual yes. Okay, we use this, but still I use the same thing. No, this is not what I want. This, yes, it is short and wider. It's the human. Remember, I say, see, it's a human. This is the bipedalism for human. Because we walk on two limbs. You see, it's a human. So it is the answer here they wanted is short and wide. That's all. Nothing else. Short and wide. Question 3.4. they are saying that malaria parasite, I talked about this also. Malaria parasite is transmitted to human blood by mosquitoes. People living in malaria affected areas take anti-malarial drugs to prevent the infection. If the malarial drugs are effective, there should be no parasite in their blood. The presence of malaria even after treatment indicates they have evolved to be drug resistant. Explain. The research wanted to determine if the use of anti-malarial parasite could causes the resistance in malaria. Okay, they gave you blood sample was collected from people of this affected area. Okay, the collection of blood sample was done in several villages. Okay, and the same blood kept in the laboratory and tested. Then the number of sample having the malaria parasite was counted. They're saying investigation was repeated 3 times. That is good. They're saying that in total, okay, that is good. They're saying that was collected, okay. They're saying how did malaria parasite transmitted? They said through blood by the mosquitoes. So that is the answer. They're saying what is the independent variable? The independent variable is, they said it here. They say that the researcher wanted to determine the use of anti-malarial drug caused resistance, meaning that the independent is anti-malarial drug. So that is the answer there. And then what depends is the drug resistance. Drug resistance will depend on which type of anti-malarial drug they are using, or you can also say presence or absence of malaria. It can be the presence or absence of malaria. They can also credit you, but you saying that resistance is a key point. Calculate the average drug resistance in the blood sample taken. Show your working around of your answer to. Okay, here you are saying that the percentage of drug resistance. They're saying that the sample were collected, 511 had malaria parasite, meaning that they were able to resist. This one they didn't have the parasite, meaning that I'm gonna say that 551 divided by 1485. 1485 times 100, which gives me 37.1. They said 1.1%. Name 2 ways in which you can increase or researcher ensured the following. Validity was done. Basically they use the same blood testing blood sample. They actually tested blood sample from the same individuals in the village. Yes, they said it here. The same area. Yes, meaning that the same village. Okay, cool. Then they also use this the same testing method. They didn't change the methods they used. They said they tested it, so they used the same method. And then how did they ensure, because it is past tense, how did they ensure the reliability? They used the large sample size. Don't just say large sample size. They used 1,485 individuals or samples. 1,485 samples. Yes. Then number 2, that's the large sample size. They repeated the experiment. They repeated it three times in the same same village. That was reliability. That was validity. Yes, they repeated it. They repeated the investigation 3 times. Yes, that is also reliability. So you tell us what they have done. Yes. Then they are saying that describe how malaria parasite developed drug resistance. In this case, during our example, we used what you call DDT. Yes, we use this. With this DDT. How? So the first step you need to talk about variation. The second step you need to talk about selective pressure. The third step you need to talk about the interference. The fourth step you need to talk inheritance. The fourth step you need to talk about the adaptation. Meaning that in this case you tell us that there was variation among the parasite to the there was variation among the malaria parasite, those which are resistant to the drug, and then those which are not resistant to the drug. Yes. when the first application is done, yes, what happened? Those which are non-resistant, they died in large numbers, while those which were resistant, they survived, but those were few numbers. You understand? Yes. Then those which survived, they pass their genes to the next generation. You understand? After many generation, yes, the resistant one, they increased in the population. You understand? Resulting in the resistant parasite. Therefore, the drug will become ineffective, hence resistance. So basically, you explain exactly this. Instead of talking about DDT, you talk about that malaria parasite. Instead of talking about DDT, you talk about the malaria parasite. That's it. That's what we call the application. The question, when the question comes, you just replace it. So that's what they wanted about these mosquitoes. Yes. So yes, then the last question, this one we did it. I don't know how many times we did it, but we did it. And I told you that don't make a mistake to go to the paper when you don't have this. This is what I told you. Don't go to the paper without knowing these questions. And I went through them, and you'll find out that all the questions they said, they are there. And they're saying that number one, how many genera are there? Yes, how many genera are there? How many genera are shown? You see, that's the question. And then they're saying how many genera are there. So you look at Adipithecus and Homo. So there are two. So basically two. Because this is Homo erectus, Homo neanderthalensis, this is Homo sapiens. All of them are Homo. When did the Australopithecus africanus became extinct? The question is there. When did the Homo habilis became extinct? You see, they're saying that. So Homo habilis, they say now Homo Australopithecus africanus. So it's the same thing. So it stopped here, meaning it's 2.0 million years ago. Then they are saying that name 2 Australopithecus africanus species in South Africa. Name 2 Australopithecus africanus species in South Africa. So this is what I told you, that we always ask you that when they come to the fossil, they're going to ask you name 2 Australopithecus africanus species, you know. So you can talk about Taumchild, Mrs. Place, or Littlefoot. They said africanus, Australopithecus africanus. Yes. So when you come back to that, yes, is this thing Australopithecus africanus species. That's when I was explaining it, I said this. They're saying that they use the only the species in the diagram describe the fossil evidence, meaning the species only. Remember we said that Australopithecus africanus, you give example, yes, and Homo habilis. Australopithecus africanus species only were discovered only in South Africa. And then here Australopithecus africanus species only in South Africa. And then Homo erectus, even this one, Homo neanderthalensis and the Homo sapiens, the oldest was discovered in Africa. This is what I'm trying to say. Yes, because based on the question, they only want, you see, this Adipithecus, they didn't talk about it. They started from here. Australopithecus africanus, yes, and Homo habilis only found in Africa. Yes. And then the oldest fossil of Homo erectus and Homo sapien were found in Africa. I said that this one, you must use the word oldest. If you don't use the word oldest, you are gone. Yes, so, and I repeated it, guys, and I said make sure you write the word only, because if you don't say the word only, then you are gone. They're gonna cross it. So, and this word oldest, it must be there. Don't make a mistake to do that. That's what I emphasized you, and I begged you. I said, guys, this question, when it comes, make sure that you answer it properly. So guys, those who are revising with this paper, make sure that you use it now the next time they bring it. But still, in the next paper, we'll also predict for you, and then we think what is gonna be coming in that regard. Explain how the influence of brain volume on the complexity of the tools made by Homo habilis and Homo sapiens. How is the brain size? This question is linked to the question which I asked you, that how is the brain size? This one, the last one, how is the brain size? Describe how the brain size evolution trend. How does the brain size show the evolutionary trend? This is the question which I said that make sure you answer those questions, guys. Yes. Explain how is the brain volume, meaning that the bigger the brain volume, the bigger the brain, yes, the higher the number of brain cells, meaning that the higher the intelligence. You understand? They're saying that the complexity of the tool, you understand? Basically, it shows that, as actually I showed you this diagram when I was explaining it. I showed you this diagram. Where is it? Yes, this diagram. And I say that as the brain size increases, yes, the number of brain cells also increases, meaning that the intelligence increases, meaning that as also the tools also became more advanced. So the brain size, as the brain size increases, the advancement of tools also increases. So as the brain size increases, okay, the advancement of the tools also increases. Then why? Because these tools require thought to be developed. You understand? Yes, that's what we said. And then, yeah. And after that, I told you guys, I wish you the best. And then it's the same conclusion. See that, yeah, it was 99% success prediction. Yes, and we will always predict for you, and we want you to pass. Stay with us, study from the beginning until the end. I promise you, you will never regret you studying with us. There are some people who try to study with us, they put some pressure or say, no, we know what we are doing. Ne? Yeah, we know what we are doing. Yes, and we know that step by step. You don't need to rush for something before you grasp and understand the concept. Thank you so much, guys. Thank you. Thank you all the people. Sabelo, I see you. No cross night for all of it. Saying that geography paper 2 was released already. I didn't do a lot of useless mistakes. I'm sorry. So thank you guys. Thank you. I see you. Ne? And I wish you the best in your life, and if things don't go right the way you wanted them, you can register again. You can register again by, I think, by what? By what? By August, yes. And then you never know, you might make it again. Thank you so much. See you again in the next class. Line for our September exam. Yes. However, we will try to highlight some of the areas where you can focus more related regarding to these upcoming papers. Also, especially for those provinces which have not written, and yeah, we can use it for final exams. And then we can also use it any exam as long as it has paper 2. Anyway, let's start without wasting time. His M-Side as usual. Thank you so much for subscribing, liking, sharing, taking care of this channel. This channel wouldn't have been there without you and without your input for following us. Yes, let's go to question number 1, and then we see what is exactly they are talking about. Question number 1. Which organelle represents the location of extra nuclear DNA in animal cell? When you talk about extra nuclear DNA, remember that we have two kinds
three kinds of DNA. The first one is the nuclear DNA. The second one is the mitochondrial DNA. The third one is the chloroplastic DNA. So now, if you have the nuclear DNA, DNA code of life, basically here we are looking at the nuclear DNA. But we have other areas where you can find DNA in the cell. So if it is a plant cell, we can find it in the chloroplast. So we call it chloroplastic DNA. If it is animal cell, then we're going to find it in the mitochondrion. But because the mitochondrion can also be found in plant cell, then it means that in the plant cell, we can have the nuclear DNA, chloroplastic DNA, and the mitochondrial DNA. But animal cells only have the nuclear DNA and then the mitochondrial DNA. So what does it mean? What does it mean? It means that they said animal cell. If you look at it here, we see that they're talking only about the animal cell. So our focus is going to be on the cytoplasm out, nucleus is out, the mitochondrion, it can be found in plant cell and animal cell. Chloroplast is out. So because this is plant, this is no, this one they said extra nucleus. So the nucleus, they have talked about it. So the only answer which is much more correct is what? Is C. So C becomes the most correct answer. So you get close to marks. We go to question 1.2. The main purpose of the genetic cross in the genetic crosstalk diagram is to measure the DNA sequence, not really determine the genetic mutation, not really predict the possibility of the offspring's characteristics, and then solve the what? Solve the crime. So if you look at it here, we see that one of the function, not sequence, we see that when you do the genetic cross, you find what we call the offspring. So the offspring's characteristics can be obtained. So when you do a genetic cross or a genetic diagram, you're able to identify the possibilities of the offspring's characteristics. Yes, so the answer becomes C, so C becomes the most correct answer. Which one of the following is the significance of DNA replication? We see that chromatin network condense, that's the description which happens in the during interphase, so this is out. Complementary pairing of the nitrogenous bases, yes, it happens, but that's not the function. This will lead to what? So this one goes out. Weak hydrogen bonds break, yes, they break, but it's a description of what happens in the DNA replication or in the DNA in the protein synthesis. But in this case, they're talking about DNA replication, so that's not an answer. So this one goes out. The most correct answer is the only option which is remaining, and they're saying that the genetic material is doubled. You double the genetic material so that when the cell divides, when the cell divides, each cell must have equal amount of the genetic material. So that's the function of the DNA replication, so that it can bring about the multiplication of this DNA, so that during the division of the cell, the genetic material can be equal in each cell. So the answer becomes D. So D becomes the most correct answer, and then you get those 2 marks. Thank you so much for liking and subscribing. All right, we continue to the next point. The biological evolution refers, remember when you talk about evolution, it means that the increase in the complexity of an organism. So when you increase the complexity of an organism, so we call it you're evolving. Yes, but this, because it is about life, that's why you're linking it to an organism. So evolution can happen, evolution of a fawn, evolution of the sun, evolution of the moon, evolution of the what, but because it is about the organisms which have life, then it's going to become biological, because biology means, bio means life. So biological evolution must be referred to the increase in the complexity of an organism which has life. Let's go to the objectives and then we find out. Formation of new species, that's speciation. Gradual change in the characteristics of species, that is also not gradual change in the characteristics of a species. Over time, gradual change. You see, you are changing the characteristics. So this looks to be that correct. Present day distribution of species in the world, that is distribution of organisms, that is by geography. Structures that have the same basic plan but adapt for different functions, though that is in homologous structures. So this is also out, this is also out. So the most correct answer looks to be B. So B becomes the most correct answer and you get your 2 marks. So we go to the next point. Peacock, you know them, use bright colors to attract the mating. Mating, you know the mating. No, we don't need to explain about mating. Everyone knows about mating. Don't forget to stay virgin. All right. The reproductive isolation mechanism is called breeding, hybrid infertility. No, this is not about the colors. They're saying about reproduction at different times of the year. They're saying use bright colors, attract. There is nothing related to time there. So time of the year, no, out. Species specific courtship behavior. The way how they dance is not the same way you dance when you're looking for the mate. It's not the same way. So basically here, this one becomes the species specific courtship behavior. Each species has a specific way in which it attracts the different sex, the different gender. You understand? During the time of mating. So each species, when the frog is doing it's nice for them. It's ugly for you. But for the frogs, it's nice. So that's how they do their behavior. You understand? Yes. So this is species courtship behavior. Copulation, you know that the insertion of the penis inside the vagina, that's out. It's not about that. So this is the most correct answer, which is species specific courtship behavior. Each courtship behavior is specific, a specific species. That's what it means. However, humans, some of them are ugly. You want to try everything. I didn't say that. So the answer is C, and then becomes the most correct answer. You get 2 marks. Thank you so much, guys. We continue. Life science is nice, guys. By the way. All right. Mendel, the scientist who played a role in the discovery of DNA. They are not talking about discover the DNA. They're asking the person who played a role. Whatever part you did take in the discovery of DNA, that's what they are looking for in this regard. Let's continue and find out. Mendel, and Crick. Yes, Crick, yes, but Mendel out. That is evolution. Bloom, but this evolution. Watson is yes, is correct, but Darwin, that is evolution, is out. Franklin, yes, correct, and Wilkins, yes, correct. So D becomes the most correct answer. James Watson, Francis Crick, Maurice Wilkins, and Rosalind Franklin. Those are the 4 individuals who were participating in the discovery of DNA structure based on our molecular. Yes, so that's it. We continue. Difference between genotype and phenotype is that the genotype is a genetic makeup, while phenotype is external. Genetic makeup, genotype, the gene type. Which kind of genotype do you have? Which kind of genetic composition do you have? That's the gene type. Is it a recessive? Is it dominant? Which type of gene you have? So that's what it means. And then you have the phenotype, the physical expression of an organism. How do I look? I'm dark-skinned, man. You see me. So that is physical expression of an organism. You see? So that is the phenotype. So this looks to be correct. Saying that the genotype is an allele inherited from maternal origin, while the phenotype is an allele from Norman. They're saying that the phenotype is external appearance. Norman, phenotype, gene type. So it's not about the external appearance. Genotype is the inheritance, while phenotype is not. Man, the characteristics you have, you obtain them from your father and your mother. So since it is like that, then it means that we inherit even the phenotype, because the genotype will determine the kind of the phenotype you have. And this genotype, you obtain it from the mother and the father. You understand? So So this one also out. So the most correct answer becomes A, and A, you get 2 marks in that regard. You see, by the way, life science is so nice and enjoyable. Eh? When you try to do this life science, you'll find out that life science is nice. It's nice. By the way, every day you practice life science. How can you fail life science? Let's continue. 1.1.8. The graph below shows the average height of giraffe. Giraffes in different time period used by Lamarck in the evolution theory. All right, it's a story. The average height of giraffes in different time periods. Okay, sharp. Which one of the following is shows the correct percentage increase in the height of giraffe different times of the year or periods? Remember, percentage increase, we say that it is final minus initial over initial and times 100. So I need to find out that if I get the final, is saying that from 20 to 15, yeah, 20 to 15, yeah, 20 to 15, now I mean is okay from here to here. So if you put this minus this, divide by this, times 100, do you get this out? B, 15 to 10, meaning that final, which is 3.3 minus 2.2 divided by 2.2 times 100, do you get this? If you don't get it, then it's wrong. 10 to 5. 10 is 3.4, 3.3, and the 5 it is 4.5 minus 3.3 divided by 3.3 times 100. Do you get this? If no, this is wrong. Then 5 minus 1 or until present, 5 from here until present is 5.5 minus 4.1. That is 1 divided by 4.5. Then times 100. If you get this, then that becomes the, if it's wrong, then it's wrong. If you get it, then it means that that's the correct answer. So our most correct answer here is D. So D becomes the most correct answer. You understand? Yes. We go to the next question. 1.19. All right. The ultimate purpose of artificial selection is that increase the genetic variation. Artificial selection increase the genetic variation. I don't think so. Allows the organism to survive in the natural environment. Artificial? No, man. No, man. Reduce the size population of species by removing the organisms with the undesirable characteristics. Why should we reduce the size of the population? For the organisms, yet we want more of them. I don't think so. Let me see it. Increase the organism with the specific desirable trait for human benefit. Discard acid, for example, milk, different characteristics we are looking in organism, meat, disease resistance, meat. I talk about meat because I like meat. Hey, man. Nyama. All right, we continue. 1.1.10, which organic catalyst is used to cut the plasmid, in genetic engineering. Remember, plasmid is that bacteria which, did I say bacteria? Yeah, is found in the bacteria. This plasmid is the one where the enzymes are used. We call them restriction enzymes, basically. We call them restriction enzymes. Restriction enzymes, these are enzymes which cut at a specific area, you understand, of the genetic material. So they are used to cut that plasmid's DNA so that the one which you want to multiply is inserted inside there. So basically, when they say the organic catalyst, definitely we are talking about the enzymes. They are specific, the enzymes, these ones, which we call the restriction enzymes. So they're saying enzymes, okay, looks to be correct. Stem cells, nope. Recombinant DNA technology, nope. Bacteria, nope. The plasmid is found in the bacteria, so it can't be. So the answer is A, which becomes the most correct answer. And that it means that you get 20 out of 20. So you see that, guys, it is so nice chilling, and getting 20 is very easy. Guys, make sure that in these objectives you get 20 out of 20. Yes, if you see that you have challenge, check out what you think it's not right. There are some answers which you see that this is wrong, can't be. So the remaining one could be the correct answer. So you can use what you call the elimination method. Yes. All right, we got to question terminologies. I like them. I like them. Yes. Pattern of DNA bars, a pattern of bars, black bars, bands on X-ray films where sample is put through special biotech process. So we call these ones we call them DNA profile. These are DNA profile. Yes. Then they're saying that a fluid that has free RNA nucleotides complementary to DNA strand. A fluid that has DNA. When you say that fluid, remember, when you talk about free DNA RNA nucleotides complementary to DNA strand, meaning that this they are talking about this is DNA. So where is DNA found? DNA is found in the nucleus. The what? In the nucleus. So if it is found in the nucleus, how do we call the fluid inside the nucleus? What is this fluid called? Since it's the fluid, the one which is outside here, we call it cytoplasm, the fluid of the cell. The one which is found inside, we call it nucleoplasm, the fluid of the nucleus. So the most correct answer here is gonna become, we're gonna say it is a nucleoplasm. The type of dominance that produce an intermediate phenotype. Whenever we talk about intermediate phenotypes, we're talking about incomplete dominancy. For example, none of the two is completely dominant over the other. It means that each contributes 50%. For example, I always give an example of presidents. The president of South Africa and the president of which country can I say? Zimbabwe, Malawi, Botswana, Eswatini. So all of those, if they are there, yes, they have the same power in their own countries. So when they sit on the table, they address them as presidents, meaning that if they are discussing about something, each vote will be equivalent to the same power. If this vote is 1, even the other one is going to be 1, meaning that they have equal power. None of them is completely dominant over the other if they are sitting on the same table. None of them, because all of them are presidents. So if you have red crossed with the white, you produce a pink. Red is not completely dominant over white, and white is not completely dominant over red. And then now you produce what we call an intermediate color. Yes, or an intermediate characteristic. So this is called incomplete dominancy. Then you say that the type of barrier in speciation that split the single population into subpopulations. We call it the geographical barrier. Yes, we call it geographical barrier. Yes, remember we said that there was one population of. Remember, you have to apply when it comes to explanation of the speciation. There was one population of rats. You understand? The population is divided into 2 by a rock. So a rock becomes a geographical barrier into 2 subpopulations. So it divides this population of rats into 2 subpopulations. So how do you call structure which split this population into 2 subpopulations? We call it what? We call it geographical barrier. Correct. A reproductive isolation mechanism that occurs in flowers only. Flowers only. Obviously, flowers, they do carry out what we call pollination. So it's can call it, you are adapted to different pollinating agents. Yes, that is the word I wanted to modify. Pollinating you are having a different pollinating agent. For example, the insects, you are specific to a specific insect with a specific shape of the beak. So we call it pollinating at different seasons of the year. So that is it. Then we got the next question. They are saying that the genetic disorder resulting from a mutation caused by absence of blood clotting factor. A genetic disorder resulting from absence of a genetic disorder resulting from a mutation caused by absence of blood clotting factor. If you don't have blood clotting factor, it means that when you have a hole, maybe you have got something on your skin, the blood will ooze. So when the blood oozes, then it means that we call it hemophilia. Hemophilia. Yes. This is very common in ladies. Did I say in ladies? Many they are more affected because they don't have remember, hemophilia is caused by a recessive allele. So male are more affected by hemophilia, yes, than female. So we mostly find it in the males than in the females. Why? Because males they only have one X chromosome. So you can we will talk about it. If you find a question, they can ask you that why are males affected by this sex-linked disorder than females? You understand? As a result, you can say that males have two X chromosomes if a recessive allele appears on one X. Did I say males have two X chromosomes? Yeah. Females have two X chromosome. If a recessive allele appears on one X, it can be masked by a dominant allele on the other X. You understand? And then the person is still normal, but is a carrier. But if the males, once the recessive allele appear on one X, is carried on one X, automatically the person is what? Is the hemophilic. Then they're saying that the source of variation where individuals in the population has equal chance of mating with other. The source of variation where any individual in a population has equal chances of mating with other individuals. So how do you call that source of variation? So we say that we call it random mating. We call it random mating. Then evolution theory proposed by remember that you must always say A only, B only, or A and B. Rapid periods of change, meaning that they are alternating with one another. So it is the punctuated equilibrium. So you guys, yeah, we got the next question, which is 1.1.3. They are saying that a component of nucleotide found in DNA, component of nucleotide found in DNA and RNA. They're saying DNA and ribose sugar. No, this one is found. This one is found only in RNA, but they said DNA and RNA, so the answer becomes A only. A only. A only. Yes. They're saying that segment of DNA that code for a particular trait. This one is the segment of DNA that control a particular trait. So we call it a gene. It's A only. Advantage of bipedalism, wider view of the surrounding climbing trees. A only. Okay. Remember that you must always say A only, B only, or A and B, and then both A and B or none. So please make sure that you have this word only. Yes. So we go to the second question, which is 1.4. The diagram below. So once they bring such diagram, they can ask you describe crossing over, draw the structure of a chromosome after and before. They can ask you a lot of questions here. They can talk about the karyotype. They can talk about Darwin's. They can talk about trisomy. Trisomes. They can talk about the trisomes. That is Down syndrome. Yeah. So what are they asking? So you guys, I always tell you that once you find this question, try to rotate it so that it looks like this, so that this and this, they are on the sides. Okay? So once in the side, you can see that this is the middle. So if you look at them, then it means that this is metaphase. And then once they are like this, because these are the spindle fibers, they are radiating like that. They are supposed to be inside. So once they are like that, then it is easy for you to make it simple. So since it is one chromosome per, if there was another one here, then it would have been meiosis 1. But since it is only one chromosome, then it means that is meiosis 2. So now we know that that is yeah, so now we know that that is which diagram 1 or 2 represents meiosis. Diagram 1 2 represents meiosis and then represents mitosis. So this one looks meiosis, diagram 1, and then diagram 2. Why did they bring this question? Anyway, I will say that it's diagram 2. Do you know why? Because here there are no variations. Here you see that the chromosomes, they are varying due to the crossing over. But here I don't see any variation. So it could be mitosis. So I'll conclude it like that. Name the process that occur in the cell division, diagram 1, that resulted in the you see, it is crossing over. So I will say that crossing over. Crossing over. In which phase does the process mentioned above occur? It occurs in the prophase 1. Occurs in prophase 1. Name the type of cell in which the cell division occurs. Name the type of cells in which the cell division, diagram 2. Oh, diagram 2. Remember I said this is mito. It looks to be like mitosis. And mitosis occurs, where does it occur? In the mitosis occurs in the somatic. Somatic. Use this color. Somatic cells. It occurs in the somatic cells. Then they are saying that state the diploid number of chromosomes in the diagram. Diagram 1, diploid number of chromosomes. Diploid number of chromosomes in diagram 1. Did they say it's a human cell? They didn't say anything about human cell. So it's going to be diploid number. It's going to be 4. And also this one is going to be 4. So, state the number for the cell. No, in diagram 2, remember, if this is mitosis, let's analyze it like this. If this is mitosis doesn't change the number of chromosomes. So if it is 2, they're gonna be 2 in meiosis in from the beginning until the end. So they're gonna be 2. So if they're saying diploid number, and here I'm seeing 2, so it means that this is not 4. This is gonna be 2. While this one, since this one it looks to be metaphase 2, then it means that in metaphase 1 there were 4. So there are 4 here. Like this. There are 4 here, while here there are 2. Why? Because in mitosis, the number of chromosomes don't change, while in meiosis, the number of chromosomes change. And if you look at it here, this is meiosis 2. That means that in meiosis 1, the number of chromosomes divided. So since these are 2, so in meiosis 1, then it means that there were 4. And give the number of chromatids in diagram 1. What I see. So it's 1, 2, 3, 4. So remember, each chromosome is made up of 2 chromatids. It's 1. Check. It's this. then it's that. So this one. So if I have 2 chromosomes, so it's gonna be 1, 2, 3, 4. So the answer here becomes 4. In this bird species, dark feather color, capital D, is dominant over light feather color. Okay. And then long tail is masks the expression of the short tail. Okay. They are saying that the square below shows the possible results when the gametes of the parents were crossed. Okay. So if they were crossed, they are saying that name the type of cross shown in the diagram above. So this is a dihybrid because we are considering 2 characteristics. We are considering 2 characteristics. Yes. All right. Here you are seeing 4 of them, but those are not. If you are saying that size, you have big or small. So that's one character, which is size, your body size. So that is one character. So you have to understand that concept so that you don't get it wrong. So they're saying that they name the type of cross represented in the Punnett square above. So we say that is dihybrid cross. They're saying that write the phenotype of the offspring. Right, the phenotype of the offspring. Just a moment. Write the phenotype of the just a moment. Okay, the phenotype of offspring X, it is easy. I go with this and this because that's where the cross is going to be capital D, capital L, small l. Yes. Then they are saying that the genotype, right? State the genotype of two parent 2. Oh, this is parent 2. So how do we get the genotype? So what I do, did I say genotype? They're saying genotype. So let me first identify the phenotype so that I'm able to identify the genotype. So the phenotype, I'm gonna say capital D, the first letter. Do I have any small d? Yes, I have a small d. You see? So small d. And then capital small l, and then small, even this is small l. Even this is small l, so it means that is capital D, small l, small l. Yes. So this is the genotype. Capital D means what? We come back and then find out. Capital D is dark feather. And then small l is small l, small l, small l, small l, small l. Long tail masks the expression of the short tail. So this is small l. So it means that it's going to be dark feather with short tail. Yes, so that is going to be the most correct answer. Yes. Then they are saying that the dominant, the type of dominancy, the type of dominance in tail length. Tail length. Where's tail length? One is completely dominant over the other. So it means that is complete dominancy. So there I'll go with the complete dominancy. Remember, if complete dominance, if one allele is completely dominant over the other, for example, light-skinned crossed with a dark-skinned, then you produce a kid of dark-skinned, then it means that dark-skinned is much more dominant over the light-skinned. Tall crossed with a short and then produce a short, then it means that tall is much more dominant over the short. Then you can look at any character and then you see what you get as a result could be that the other one which is not expressed was masked by the dominant allele. Yes. So which parent, 1 or 2, has the heterozygous genotype for this characteristic? Which parent, 1 or 2, has the heterozygous genotype for both characteristics. They're saying heterozygous. I see it is the current one. You know why? Let's find out. Let's see here. Because here I have capital D, I write the capital D. Do I have capital D again? Yes. So I cancel it out. Do I have a small d? Yes. I have small d, so I write small d. So I don't write the D again. So I have capital L, capital L. Do you have another capital L? Yes. But do I have a small L? Yes. So I would go with a small L. So that is heterozygous for both characteristics. But if you look at parent one, you have capital D, capital small d, yes, small d, small L. Do I have a capital L? No. So I have to go with the second L. So you see that this is homozygous, recessive, or this is heterozygous. Well, this one is heterozygous, and also this one is heterozygous, because this is a capital L and then this is a small L due to what? Then they are saying that name two Mendelian principle that describe the separation of allele during the gametes. So here we can talk about Mendel's principle of segregation, and then you can also talk about Mendel's principle of independent assortment. We talk about the segregation, and then you also talk about the Mendel's principle of independent assortment. And then that makes us to get 50 out of 50. Who got 50 out of 50, or who is willing to get 50 out of 50? And if you got them, thank you so much. You are a star. So question number 2 is saying that, okay, question number 2 is saying that the diagram below shows different processes involving DNA molecule. So you have process A, you have process A. So it's question 2. You have process A and then process B. They're saying identify process A. If you look at here, we see that process A, you're forming DNA. So this is DNA replication. While DNA replication. While process B, we see this is the protein synthesis. Yeah, basically we see that this is transcription. So which part of protein synthesis is transcription? So process B is transcription, while process A is DNA replication. We can ask you a question: what's the difference between transcription and translation? What are the similarities between transcription and DNA replication? They can ask you the similarities between DNA replication and transcription. So you look at the steps that DNA replication occurs in the nucleus. Also transcription occurs in the nucleus. DNA replication, double helix DNA unwinds. It's the same story here. It also double helix DNA unwinds. If you look at the differences, you can talk about two strands they form the act as a template, while the other side you can talk about one strand act as a template in transcription. You can talk about two new DNA molecules are formed. This side you can talk about messenger RNA is being formed. So you look at the differences and the similarities. They can ask you those questions. They can talk about the differences between nucleotide of DNA and nucleotide of RNA. So they can ask you a lot of questions concerning about that. Name the organelle where process A and B occurs. Process A, so that is the nucleus. And then they're saying that basically we only have two organelles we usually talk about. We usually talk about the nucleus, then also talk about the ribosome. They state the phase in the cell cycle in which the event takes place. State the phase in the cell cycle in which event A takes place. We say that it occurs during interphase. So done with that, describe the event that occurred during protein synthesis after R molecule attaches to the ribosome. So if you are talking about R is a messenger RNA, so they're asking you to describe the process of translation. So you describe it. You must have the distinction material book. You'll be able to answer all these questions simply. Maybe you can say that according to the RNA anticodon, the transfer RNA with the complement. According to the RNA codon, the transfer RNA with the complementary anticodons will bring the required amino acid to the ribosome. The amino acids are joined together by the peptide bond to form a required protein. So basically, that you can explain it, or specific amino acid is going to be brought to the ribosome when the anticodon of the transfer RNA matches with the codon of the messenger. You understand? The anticodon, the amino acids are joined together by the peptide bond. The bond is peptide. Don't make a mistake and say polypeptide bond. Zero will give you zero, and that zero will have ears and eyes. Please don't do that. Yes. So the bond between the amino acid is called the peptide bond. Yes. Then they're saying that to form a required protein. So you explain as long as you talk about all the marking points which we are looking for. Describe the event that occurs during. Okay, we are done with that. Explain one functional relationship between DNA and molecule R. What is the functional relationship between DNA and molecule R? Basically, molecule R contains the coded message from DNA so that the proteins can be manufactured. That's what they are looking for. It has coded information for DNA so that that thing can be processed, can be formed. Yes. The proteins. Then they are saying that explain the function. The table below shows the amino acid between DNA and the molecule R, which is the messenger RNA. They are saying that use the table below to determine the codons. When you talk about the codons, remember we have the codes which we find on DNA, we have the codons which we find on the messenger RNA, we have the anticodons which we find on the tRNA. Yes, so I've explained this before. If you check on our channel on Thundereduc, then you'll be able to get all these questions and explanations about it. Yes, make sure that you get a distinction material book so that you're able to obtain these distinctions. Yeah, nobody, if you are using the distinction material book, you only don't get a distinction because you don't want. If you want it, use the distinction material book. The distinction is in your hands. All right. Then they're saying that write the triplet bases. Okay, triplet 1, 2, 3. So if you look at 1, 2, 3. So they are saying write the codons. So if this is DNA, then it means that I'm going to have to change it into a messenger RNA. So T goes as TGC, and then you have TAC, and then you have AGG. So it's gonna be T is gonna go with A, and then C, then G, and then it's gonna be A, it's gonna be U, it's gonna be G, then it's gonna be U, it's gonna be U, it's gonna be C, it's gonna be C. So basically this is what we are looking for, and that is what you are supposed to write. Yes. So you get your 2 marks there. How do they mark? I don't know, but it's supposed to be like that. Determine the amino acid for DNA based triplet that is complementary to this. Guys, we always ask you these questions, and the mutation. Did they ask you? They didn't ask you about the mutation, but you always ask you about this. Determine the amino acids for DNA triplet that is complementary to TGC on strand 2. Determine the amino acid for DNA-based triplet that is coded to TGC. Strand 2 is DNA, and what is here is also DNA. So since they are giving you DNA and they are looking for DNA, then definitely we are not changing anything. So it's going to be TCG. Let me clear it up. TGC. So it's going to be cysteine. So that's what is going to be the answer. Let me check properly. Yes. Cysteine. Determine the amino acid for. Let me first clear it up so that I don't want to make a small mistake, and then yes, they're saying that determine the amino acid for DNA the DNA base, the amino acid for DNA base triplet Okay, that chord, that is, oh, you see, that is complementary. You see, because this thing was covered, so you see. Let me just do like this. Yeah, guys, make sure that you're always keen. They're saying complementary, so this is complementary. This is C. So what is the complement of this? T goes with what? Goes with A. G goes with what? G goes with C. C goes with what? With G. You understand? With the C, ne? This is C, goes with the G. So it means that this is still DNA. It's still DNA. It's not messenger, because what is given here is DNA. So it means that what I'm going to say, that this is just, they are looking for the complementary. You understand? So since this, they are looking for this, so I'm going to look for this. So it's going to be ACG. ACG, ACG, which is triaronine. So that is the correct answer. So you guys, you see these questions, ne? You see, even me, I was gone because I couldn't see this because of the words which are there. So make sure that you take note of these questions, ne? Don't just give the answers and then you just go like that. Please make sure that you read these questions, because a small mistake like this makes you to get out of the what? Out of, don't finish the paper and then you sleep. Doesn't make sense. It is like you are making the examiner to be a fool. Yeah, how can he set a paper for 2.5 hours when you finish it in 1 hour? You are making the examiner a fool. Yeah, that when you don't know what you are setting. So guys, don't make us fools, ne? Yeah, don't make examiners fools. Okay, we are saying that name the type of mutation that caused Jacob's syndrome. What is Jacob's syndrome? Karyotype below. Remember that karyotype is the type of the nucleus. Karyo means the nucleus, and type means the type. Which type of nucleus do we have? Of the male suffering from Jacob's syndrome. What's Jacob's syndrome? We'll find out. What is it? Name the mutation that occurred that caused Jacob's syndrome in this image. I see here on chromosome number 23. Is this a male or a female? Is a male. Why? Because I have X and 2Y. But anyway, I'm not going to talk about that too much. I'm just answering a question which they said, and then so in most cases we don't bring these questions, which we bring the one which has the trisomy is on chromosome number 21. The one which were like this, they had been removed from the examination guidelines. So if they are brought them, let's answer them the way they are. Yes, but it's not supposed to be. Let me just say it like that. It's not supposed to be in this format. Anyway, following the examination guideline, that is my opinion. Yes. However, they also had their opinions. Why did they say it? Maybe they want to say it about something. All right, we continue. However, you can use the same principles used here to answer these questions. The good thing, they're not asking you what is it. They're just application question which you can apply and then explain. So use the concept of Down syndrome, and then you are able to obtain or transfer that knowledge to this question. Maybe that's the instance for them setting such question. They're saying that for this individual, determine the number of autosomes. Remember, autosomes are the chromosomes which are not they are not sex. So I'm going to have to count out is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. Guys, always count them, because sometimes there is one where there is 3 of them. So autosomes, there are 22 pairs. Or I can say they are 44. Yes. Then gonosomes, we see them, there are 3 chromosomes. Yes, I can't say 3 pairs because they're just three. 1, 2, 3. So these are 44 chromosomes, not pairs. Yeah, 22 pairs of chromosomes or 44 chromosomes. Use letter X and Y. Write the gonozome of this individual with Jacob's syndrome. So we see that this is X, the one which is long is X. Let me clear it up so that you're able to view. This is X. This is X. This is X. And then this is Y. So basically that is it. Yes, they're saying describe the way in which the karyotype of the person with the Down syndrome will differ from the one mentioned above. Remember, Down syndrome, the trisomy is basically happening on chromosome number 21. Like the 3 chromosomes will appear on chromosome number 21, while this one it appeared on chromosome number 23. You understand? Yes. So basically the trisomy 20 Down syndrome, it occurs in the orosomes, the orosomes which are non-sex chromosomes, while this one occurs in the sexy chromosomes which we call the gonosomes. So I think that's it. Explain how the error during the gamete formation in meiosis 2 could result in the Jacob syndrome. Then it means that if you look at this, what happened is this could be for a female or it could be for a male. But what happens? It means that during anaphase, yes, the chromosomes for the chromosome pair, like 23, one pair. The 23rd chromosome is supposed to, for example, this one is supposed to split like this. But now, that is, it is in metaphase. So during anaphase, they are supposed to split so that they look to be like this. So they are moving in opposite poles. Yeah. But now what happens on this gonosome? What will happen? This chromosome, which is the gonosome, for example, it instead of this one, instead any of them, instead of splitting like this, both of them are going to go to one side, meaning that leading to this other side having nothing. So we're gonna result in this cell having 2 of the gonosome. You understand? And then this side there is nothing because nothing went there. It's only have these orosomes which are there. So it means that if this was a male, it was a male, yes, is going to have X. You understand? 2 of them, X. So if it was a male, it's going to have Y. And then this one is going to have an X. 0. Nothing is there. So there is no gender there. So if this cell fertilizes a female ovum, the ovum, yes. So what is going to happen? It means that the ovum is going to bring X, so that now this individual is going to have 1 X and 2 Y chromosome. So that is what is supposed to be. So basically here, non-disjunction occurs as the failure of the chromatids fails to separate at position 23. So that's what we talked about. And then he said that during in anaphase 2, because this is, they are saying in meiosis 2, a non-disjunction occurs in anaphase 2. During anaphase 2, the sperm cell will have an extra chromosome, you see, due to the failure of them to separate. When it fertilizes a normal ovum, yes, the individual is going to have 1 X and 2 Y chromosome. So, which is what we call the Jacob's syndrome. But I talked about it that in most cases, we don't usually set this. I think they were just setting it to link it to Down syndrome. We like it to bring it to Down syndrome. And then if you see how I was explaining it related to Down syndrome. But instead of writing 21, chromosome number 21, I used the chromosome number 23. 3. Yes, so that is it. And then we are saying that 2.3, they're saying that state the phenotype of anger. Okay, here we are having this and this. So to know the phenotype, you need to know what is the kids. How many generations? There are 2, 1, 2, 3. So there are 3 generations. How many kids are for James and Anga? Is 1, is 2, is 3. Okay. Someone is going to ask, how is it 3? Because this shows that it gave rise to that and that and also that. So this they just got married to those kids. So they're saying that the genotype of this, so if this is blood group A, they produce blood group AB. Definitely this is blood group B. You understand? So that this person, since it is blood group B, blood group, yes, they produce this. They produce this. Why not blood group AB? AB. Because if there was blood group AB, then it means that the genotype is going to be like this. There is no way how they're going to produce handle. So it means that this one is blood group A, heterozygous, and this is blood group B, heterozygous. So that this and this, they combine and they produce tandem. Yes, so it means that Anga was blood group B. Then the genotype of individual that plays codominance, which is codominance, that is blood group this AB. So the genotype, so definitely it's going to be A. B, because they're looking for the genotype. They're saying that give the number of individuals with homozygous recessive allele, that is blood group O. That is blood, but they're saying give the number. How many are they? So it's 1, it's 2. So it's gonna be in this answer. This question is gonna be 2 of them. Explain why the future children of JD and Lisa could display different blood groups compared to their current children. Lisa, JD and Lisa. Have to look for JD and Lisa. So since JD and Lisa, this is A, this is B, this is A, and then since these ones were like this, then automatically JD was a small letter. And then to produce a different blood group, yes, to produce a different blood group, then automatically even A must be heterozygous. Oh, this is B, ne? JD is B. And then so that their children could be, when this smaller and smaller, when they combine, they're able to produce blood group 4. Meaning that the JD will pass a recessive allele to the offspring, the same way Lisa is going to pass the recessive allele to the offspring to produce blood group 4. But I need to find out what is the genotype of Lisa, which is blood group B, heterozygous, and also JD is blood group B, heterozygous, and also Lisa is blood group A, heterozygous. So you see that to obtain any answer related to this, make sure that you look at the babies they have done what they have produced. Then you are able to obtain those blood groups. Yes. So we go to the next question. Use the genetic cross to show the percentage possibility of Beta and the Oneka having children with blood group O. So I have to go back and look for Beta. Where is Beta? And then Onek. What? Bela and Onek, the same genetic cross. So you have, since they are on the second generation, I will say that P2, F2. And then they are saying that the phenotype, phenotype, I told you that this 1 mark is obtained free, mahala. So genotype, I come the question, they're saying that Baila, blood group what? Blood group of blood group what? Baila is blood group AB of blood group AB crossed with Onika of blood group what? Of blood group of blood group O. Blood group O. So when you look at the genotype, blood group AB looks like this, while crossed with Onika, which is blood group O, small I. Then they form meiosis. You have to write all these points, and then you say that A gonna be B, is gonna be small I, small I. So these are gametes. So we call them gametes. Gametes. I say that don't leave them naked because they're not at the beach. So they are supposed to be dressed up. So after that, you can use the Punnett square or you can just do the crosses. So let me just use the crosses so that I'm able to be a little bit faster. That with that, it's gonna be A, small I, and then now this with that is gonna be A, small I, then this B, this with I, that's gonna be B I, then that with that is gonna be B I, small I. So these are offsprings. So we see that these two blood group A, and then two blood group B. So we don't have any chance for someone to produce blood group which is O. So it means that 0 out of 4 times 100, so it gives me 0%. So the probability to produce this kid who is like this is gonna be 0%. Then we go to the next question. This is genetic engineering. We can ask you genetic engineering questions. We can also ask you about stem cells. Yes. So the type of cell produced by meiosis that is extracted from the donor chiral. So we look at this. This is a somatic cell. This is a somatic cell, and this is the ovum. That's a gamete. That's the gamete. So somatic cells are always diploid, while this one are always haploid. So that now, because we're looking for the animal which looks like this, so I'll keep the nucleus here. Check, this looks like this. So it means that we use the genetics of this, which is the somatic cell. So it means that I'll keep the nucleus here. I throw away the shell. Here, I throw away the shell. Sorry, the nucleus, I keep the shell. That's why you see the nucleus here looks like the nucleus which came from here. You see? And then if you look at the shell, looks like that of thick and thick. So now when you have this, we use what called the mild electric shock to combine the nucleus from here and then the shell from here. So we combine them together using the mild electric shock. Then now these embryos are being developed, meaning that they have the genetics of this. But because this embryo, this cell looks like a zygote, because remember zygote, it has a sperm, and the sperm only the nucleus fuses, and then the ovum, so this nucleus of the sperm goes into the nucleus of the ovum. So outside is the ovum and the inside is diploid. So since this is a diploid, yes, and the nucleus outside here, this is the membrane we used, looks like this. So this cell looks like a zygote. So it's not a gamete. Why? Because this is 2N, it's a diploid. So now it undergoes mitosis to form a ball of cells which develops into embryos. So these are being implanted into surrogate mothers, and then each surrogate mother develop, remember these are just stem cells, the function of stem cells, then they develop to form mature babies, and these babies are being released. So that is it. So name the type, the type of cell produced by meiosis that is extracted from the donor cell. The type of cell produced by meiosis that is extracted from the donor cell. So we're going to call it a gamete. Yes, basically it is ovum because it is a female. They're saying the organ where implantation takes place, it takes place in the uterus. Uterus. They're saying the cattle counted to be 63% of 395 different cloned animals in the particular country. Calculate the total number of cattle that were cloned this country. So this one is simple: 63,3 divided by 100 times 395. So the answer you get, that is the answer. I don't have a calculator here, so you can calculate and then you find what is the answer there. Then they are saying that explain how the process of cloning will affect the variation of the cattle. Obviously, cloning result in the identical organism. So it means that the variation is going to be reduced. So it's going to be reduced variation. Suggest the different characteristics desired by human that influences the cloning of the 3 characteristics desired, like meat. You can talk about meat. You can talk about resistance of diseases. You can talk about meat, resistance of diseases. You can talk about milk. So all those can be used to clone, be reasons to clone these organisms. So this question is also 50. So if you got 50 out of 50, congratulations, you are a star. Let's go to question number 3. Go to question number 3. They are saying color blindness is a sex-linked genetic disorder that is characterized by the genetic defect of on X chromosome. The moment you say X chromosome, then it means that you want to use X and Y. They're saying that the people with color blindness cannot differentiate cannot differentiate between certain colors. The table shows, okay, according to the table, which region has the highest or has the lowest percentage of male with the color blindness. So if you look at it here, we shall see that this is the answer. So it becomes Sub-Sahara Africa. This is just a question which is an application. It can be changed. It's easy to answer. Yes, suggest one reason why the results in the region identified in the question might not be correct. Why should we? Why should do you think? Maybe there are no proper facilities which are used to test. Yes, maybe the facilities are not there. Maybe there is lack of reporting of some cases might not be reported, and then so the information could be wrong. So things which can make the results not to be right, or not everything which has been collected could not be right, or some information might be missing. Yes. So the prevalence color of color blindness in the female is 0.5 globally. Explain why female have a lower chance of being colorblind. So this one is talking about, this is what I explained, that females they have less to be affected by hemophilia. This is a sex-linked character. You understand? Yes. Why? Basically, females have two X chromosomes, so they will require two recessive allele for them to be color blind. So if one recessive allele is appear on one X, it can be masked by a dominant allele on the other X, and the female is still normal. However much is the carrier, but is still what? Normal. So females will require two recessive allele on the X chromosome for them to be color blind. You understand? Therefore, it means that even if one recessive allele appear on X, then it can be masked by a dominant allele on the other X, and still the individual is still normal. Then you draw a bar graph, you guys. This is question you must be knowing. Make sure that the bars are the same size. Make sure what you have here, this is X, this is Y, so this one goes to X, so it comes here, and then these numbers, they go here on Y. You can't have words here, never on Y axis. No, they must always be on the X axis. Then you put the caption. Make sure that the caption, you put them properly. Make sure that what you put here, you put units if the units are there. In this case, the unit is percentage, so the percentage must be there. Countries, region, is basically, these are countries, so you can put in brackets a region. So it can be something like a unit, and then it must be indicated there. And then make sure that these bars are the same, the spaces between the bars are the same. Then you'll be able to obtain all the marks, the 6 marks they have given you. Then we go to question 3.2. 3.2 is a little bit easier. They're saying the diagram below shows the scar of hominid ancestor. Yes, the hominid ancestor, and they used the tools as their evolution. So they are looking at the evolution, the tools they used as their evolution. Okay, they're saying state the line of evidence for human in evolution. In the diagram, state 2 lines of evidence for human evolution in the diagram. So in this case, we see that we have the tools, and then we have the fossils. So remember, when you're explaining, you don't say tools. Remember, the evidence is cultural evidence. Example of cultural evidence is use of tools. So the answer here is cultural evidence, and then here number 2 is fossil evidence. Then they're saying that name 2 Australopithecus africanus species found in South Africa. We talked about them: Taum Chad, Barefoot, and Mrs. Place. So this one are easy to identify. Identify the type of tool used by species with the largest cranial capacity. So if you look at the tools they use with the largest cranial capacity, so we see these are the tools they used. Then they are saying that how many genera are shown in the diagram. So if you look at the genera, we look at the first, like the surname. So we see the Homo is one, is still Homo, is still Homo, two, and then this is the three. So there are three genera which they shown in that diagram. They're saying that based on this diagram, the Handyman species was in existence 1.8 million years ago. Write the scientific name for Handyman. That is Homo habilis. So Handyman, remember, is the first man to use tools, so we call them Homo habilis. Provide a reason why species named above is regarded as handyman. It means that it's the one which first used the tools. So I've given the answer. Explain why the evolution of large cranium in human is associated with the development of tools. Why is this that the more the brain is developing is linked to the advancement of tools. Advancement of tools, these tools, they require thought. So it requires the brain which is thinking, the brain which is much more how do I call it, much more developed to think and develop these tools. So we can say the increase in the brain will lead to the increased intelligence. Actually, when you have a large brain, you're going to have more brain cells, which leads to the increased intelligence and then leading to the development of the complex tools. So increase the brain means that you have much more brain, yes, which increases the intelligence, leading to the increased intelligence, and then this means that you're going to have to develop more complex tools. 3.3, what are they asking about? 3.3. Here is an extract you're supposed to read, and then you're saying that lactose intolerance refers to the ability to digest lactose sugar into glucose, and lactose tolerance individuals do not experience comfort after drinking milk, eating dairy products. Lactose. However, some individuals struggle to digest lactose due to the replacement production of lactase enzymes, and then classified lactose intolerance. Such cannot be. So scientists investigated the influence of lactose intolerance gene mutation on the lactose tolerance in African population. Okay, this is very important. They're saying that they collected of this. They're saying they were found in Tanzania, Kenya, and Sudan. And then they're saying that divided the participants into two groups. They're saying that I'm seeing the groups gave the participant lactose sugar solution to consume daily. They ensure that participants ate the same diary during the investigation, measure the blood glucose before and after. What is the independent variable? So what determines this concept? So basically, the independent variable is the lactose intolerance gene. This, because I say investigate the influence of this on this. So it means that this is the independent, while this is the dependent. So lactose tolerance is the dependent. So this is done. Then the type of variation existed in the ability to digest lactose. So it is discontinuous. Either you digest or you can't digest. You can't try. No, you either do it or not. So it is too, you get your mark there. It is discontinuous variation. Then they're saying that state way in which the scientists ensure the ability of the investigation. Remember ability by increasing the sample size. So they said they ensured, meaning that they used 470 people. Yes, the individuals were from different ethnic groups. You understand? We see them there. We see that all different area of the what? Sorry, guys. Alhamdulillah. So we can talk about all of that. State the factor that should be kept constant when the lactose solution was consumed. Which factors? The amount of lactose which you consume must be kept constant. The concentration of lactose must be also kept constant. Why are scientists measure the blood glucose level before and after? Basically, they want to compare these results by developing what called a baseline. Yes. Okay, before it was like this, after it was like this, what caused what? That change is caused by what? So they want to find out the effect. So by developing what called a baseline. Then they're saying that describe how scientists could establish whether the gene mutation has occurred on this, whether a genetic mutation had occurred on lactose this gene. So basically, the sequence of gene of DNA or of the participant on the chromosome, yes, should be determined. And then they have to determine it using the DNA sequence, you understand? And then after that, they have to identify the mutation of this gene, and then they compare it to the one which is lactose intolerance. If they see that there is a difference, then automatically it means that there was a mutation. So they have to first identify the one which is tolerant and the one which is intolerant. And then after that, they must compare. And then they see that, oh, the one which is intolerant, maybe the mutation occurred. If there is a difference, then automatically it shows that there was a change in the sequence of the gene. So which means that it was a mutation. Then we go to the next question. Large scale overfishing. Okay, so remember much of the rate of display humans in target fishing, and port. Next one. So this 30% reduction by the size of faster maturation. Okay. According to the paragraph, which human activity has resulted in the... So when we bring such question, guys, make sure that you read the passage and then you read it at least 3 times so that you're able to obtain the information which is there. According to the passage, which human activity has resulted in the population of Atlantic codfish? Basically, if we see that there is large scale overfishing, then they are saying that explain the effect of GHR gene mutation on the survival of the chance of Atlantic cod fish. So basically, what is, explain the effect? What is the effect of this? What is the effect of this? Yes, mutation on the survival. So we see that this mutation was useful. How was it useful? Yes, because it's basically reduced the size. Ne? So if the size is reduced, it means that the fish could be a little bit faster and then increase the chances of their survival, meaning that they are able to move very fast. They are swiftly moving, and then they are hiding. They're able to hide from the predators or from the hunters so that their population can increase. Used down in theory of natural selection, explain the evolution of small body fish, small body size, and the faster maturation rate in the Atlantic codfish. So what do we explain here? Remember, this is natural selection, so you can have to tell us that there is variation among these codfish. You have to tell us that. Then you tell us the variation. Which variation is that? Some had experienced mutation of on GHR genes that resulted in the small body and then making them a little bit faster, and then than others. And the others did not have that, meaning that the one which had the mutation, yes, they were able to be faster, you understand? And then others, they didn't have that ability. So that is the variation which we are talking about. So basically, when overfishing occurred, yes, so that is now the pressure which is being exerted to these fish. The codfish without the mutation, what happened to them? Meaning that they could move less, so they were being hunted, they died in large numbers. All those ones with the mutation, they could move faster, so they survived in large numbers. You understand? So they reproduced also in large numbers. They passed the allele for this mutation to the next generation, and then after many generations, it means that the next generation will have a higher proportion of these codfish with this mutation, and then with a less proportion of those ones without that mutation. So basically, that's what you're supposed to explain. Please don't confuse this natural selection with speciation, because in most cases, students confuse this and then they end up losing the They are saying discovery. There's so many passages. Discovery of fossil in remains of 0.87 million years of scientists like Europe understand the this call found in this transition has a transitional future similar to that of Africa. Okay, name the hypothesis of human origin that is being challenged in this passage. So we call it the out of Africa hypothesis. We don't have any other hypothesis. State the hypothesis mentioned above. Modern humans, the word modern must be there. If you don't put it, remember we have different humans, homos. All homos are humans, but the modern is the Homo sapiens. So you have to be specific and say that modern humans originated from Africa and migrated the rest of the world. Yes. Then they're saying that describe three characteristics of the skull that were similar to that of these African apes. So foramen magnum at a more backward position. Yes, it is in a more backward position. A more backward position. And then you can talk about large brow ridges. You can talk about they were prognanthus, meaning that they were prognanthus, meaning that more protruding jaws. Yes, and then you can talk about the rectangular palates. So the way how these apes were. Yes, they are saying that what is the name of the birthplace where most fossils of mankind were found? So this is cradle of humanity. So basically, that is the paper, and since the paper was not that difficult, I see that you guys you will make it, and then we see that um so guys, yes, we wish you the best in your exams, and we pray that you guys you pass you very well. I've not talked about the phylogeny. Oh, the phylogeny was there. Make sure that you do all those questions which are found in the phylogenetic tree at the back of the section material book, so that you'll manage the paper. Don't stress about it. You'll manage it. You'll pass it. It has been MSID. I love you so much, guys. Thank you for loving Thundereduc. And just know that we are here to see that you guys, you become responsible citizens who are not just citizens, but those citizens who are rich. Welcome back to our class. Today we're going to be talking about the paper 2. This paper 2 was being written. Guys, thank you very much for joining our class. Let's talk about the memorandum of or suggested answers of this paper you wrote. I know that some of you smashed this paper, most especially if you were among the people who were following us. Yeah, I hope you managed to score high. And then don't be challenged by anything. For example, maybe my answer is a little bit different from your answer. Yes, it could be, but it could be different answers. I cannot discuss each and every answer which is most likely to be in this paper. So, guys, let's start without wasting time. Our paper, so that we are able to get it right. I thank everyone who shared, who were on this discussion of the prediction, the late revision, everything. Thank you very much. May God reward you, and we pray that you have a successful life. If things don't go the way you expect them to go, please still don't lose hope. If one door closes, obviously there will be another door opening for you. Just keep opening the doors. You'll be able to get one which is right. All right. 1.1.1, we are saying that which female scientist took X-ray diffraction X-ray of DNA molecule and concluded that DNA is helical shape or helix. shaped. In this case, all of them, these people, all of them were among the people who discovered the structure of DNA. They just talked about one name. James Watson is a man. Francis Crick, they were working together, is also a man. Rosalind Franklin is also a man. So the answer is, did I say Rosalind Franklin? Maurice Wilkin is a man, and then Rosalind Franklin is a lady. Is the lady who died of cancer. Yes, so the answer, the most correct answer in this case is supposed to be B. A cell with the 14 chromosomes undergo meiosis. In this case, they're saying a cell with 14 chromosomes. They didn't say that this is a diploid, homologous chromosome. They just a cell with 14 chromosomes. They didn't say that a cell with homologous chromosome. So it means that they're just talking about 14 chromosomes. If they're homologous, then automatically it means that there are 7 chromosomes. So a cell with 14 chromosomes undergo meiosis. What is the number of chromatid? They're asking for the chromatid. Remember, each chromosome is having 2 chromatids. So they're asking what is the number of chromatid in this cell at the beginning of meiosis. So they didn't say homologous, therefore it is 14 times 2, so that it gives you 28. I know that some people wrote you are 58, but they didn't say homologous. They said just chromosome. You understand? If they had said homologous, then you would have said 56, but they just said 14 chromosomes, which means that it's just a single chromosome. Yes. So the diploid of it, meaning that it is 77. Yes. All right. So the most correct answer becomes C, which makes it to be the most correct answer in this regard. They're saying that inheritance of blood group are two. Remember when we are talking about on the prediction about the blood group, I said that blood group is controlled by one gene. Yes, one gene, 3 alleles. Yes, 3 alleles, and then we say that four phenotypes, phenotypes, and 6 genotypes. 6 genotypes. One gene, that's gene, just a gene, and then gene for blood group. 3 alleles, allele for A, allele for B, and allele for O, and then 4 phenotypes, that is A, B, A, B, A, B, A, B, and O. And then 6 genotypes, we saw them. So we come here, we say that 2 phenotypes, that one goes out. 3 phenotypes, we said there are 4 phenotypes, that one goes out. 4 phenotypes, correct, and 3 alleles. You see, 3 alleles, so this one becomes the most correct answer. 4 phenotypes and 2 alleles. No, it is 3 alleles. So the most correct answer in this regard becomes C. So you get 2 marks there. Yes, guys, don't panic because you're not getting right. Yeah, there are other questions where you're gonna get them right and still you stand to score a distinction. Yes. The following statement relates to cell division. Okay, crossing over, they're saying contributes to genetic variation in the population. Okay, they're saying that produce cells with the haploid number of chromosomes produce somatic cell. Diploid number chromosome cells with the diploid number, this is very rare unless it's fertilization. Somatic cells only, not really. So okay, well, let's see what they're saying in this regard. They're saying that which one of the following combination of statements refer to meiosis. Obviously meiosis result in haploid, so not diploid. So this one, whenever there is 3, is off. So this is off, this is off, so it's off. So expect to have this and this. They're saying that produce somatic cells. Meiosis doesn't produce somatic cell. Meiosis produces gametes. So therefore, anything which has 4 is out. So automatically, we only remain with B. So B becomes the most correct answer. Then we go to 1.1.5. They're saying that some and the beds are believed to have originated from common ancestor. The shaded part of the diagram below shows the distribution of these species. So we have these shaded parts. So the distribution of the species. So in this case, what are we looking at? The type of evidence for evolution. The type of evidence for evolution that is represented that is represented in the diagram is, in this case, we are seeing that different lives are there, so therefore it is bio, because the different geography of the world therefore becomes biogeography. So A becomes the most correct answer. They're saying that the plant with yellow which is recessive, round which is dominant, is crossed with the green which is capital letter which is dominant, with wrinkled which is recessive. So meaning that you have capital Y, small y, and then capital R, small r, capital R, small r. So in this case, what are they asking? They're asking that which one of the following are possible genotypes of the parents one. So which parents resulted in the formation of this? So definitely I will need to have one which has a capital letter and one which has a small letter. Yes, so that when they combine they form this, and then this. So if you look at this one goes out, not really, because of yes, this one, it goes out also. Yes, this one goes out. So the most correct answer, because this one will come, bring it this one there, and then this one brings that there, then this one brings that there. Yes, and then this brings that there. So the most correct answer becomes A. Yes, so that is it. We go to the next two. If this is 12, definitely this is 12. They're saying 27% of guanine is strand B. If this is 27, also this is 27. All right, because they are complementary to each other. So now they're asking you, they're saying that uh, they're saying that in the segment of DNA, there will be also 31% of adenine in strand 2. If you look at strand 2, 31% of adenine doesn't look to be right. 31% of adenine. If you look at this strand 2, 31% of adenine. Adenine, no, it's not indicated in this case. So that one's out. 12% of cytosine in strand 1, 12% of cytosine, we said is 27. So that one is also out. 31% of thymine in strand 2. Thymine in strand 2 is 31. Let's see the last one, 27% of cytosine in strand 2. Cytosine in strand 2 is 12. So definitely the most correct answer of meiosis, where chromatids are pulled opposite. They are saying chromatid, they are not saying chromosomes, they are saying chromatids. So in this case, when we talk about chromatids, it must be in a meiosis 2. So therefore, where there is meiosis 1 is out. So the answer is either this or this. But they're saying chromatids. If they are chromatids, it means the chromatids are the one which are being pulled to opposite poles. This happens in anaphase. So in anaphase, if that's the anaphase, then automatically it's anaphase 2. So D becomes the most correct answer. Yes. Huh. Then we go to the next question. They are saying that the next correct question, they are saying that the section of DNA molecule that code for a specific character, the section of DNA that code for a specific character is called a gene. Is called a what? A gene. Then we go to the pattern of black bears resulting in DNA analysis. We call it DNA profile. Not DNA profiling, is DNA profile. Punctuated equilibrium. Yes, it's punctuated equilibrium. Then we go to the next, the production of genetically identical copy of organisms using biotechnology. We call it cloning. Cloning, yes. Call it what? Cloning. Then they are saying that undifferentiated cell in animal that have the potential to become any type of tissue. How do you call that undifferentiated cell? The undifferentiated cell, we call them stem cells. Stem cells. We call them stem cells. Then we are saying that a genetic disorder, genetic disorder that result in a person who cannot distinguish between colors. We call it color blindness. Color blind color blindness. Yes, the part of the plant where male gametes are produced. The part of the, I don't want to go to this like booklet distinction material to show you. Where are you? Yes, must be around here. You see, we call them what? You see, even emphasized about it. You see that this is the point, that you call them anthers, and then they say the male. If you check here on the question, they say that the male gametes. So we see here that the male gametes are produced in plants, are produced from the anthers. And how do you call those male gametes? We call them pollen grains. Yes. So guys, that's what is supposed to. Yes, then we go to the next question. The next question, they're asking you that the structure that join two chromatids together. How do you call the structure that joins two chromatids together? We call them the centromere. Centromere. So we call it a centromere. So you see, you have got 8 out of 8 in that regard. And then I think that, guys, you are achieving to that point. Then let's still go to the yes. So we call it incomplete dominancy. Yes. Then we go to the next question. The next question, they're asking you that the point where two chromatids overlap during crossing over. How do you call the point where two chromatids overlap during crossing over? So in this case, the answer here is none. It's supposed to be none. The 2 points where the two chromatids overlap during crossing over, we call it chiasma. So the answer becomes A only. Yes. Then you go back and then see that the scientists who produced the law of segregation was discovered by, was proposed by Mendel. So in this case, there is nobody. So the answer becomes none. So it is none. It is A only. It is none. You see? Yes. 1.1.4. So we go to the next question, which is involving the discussion of 2 types of nucleic acids. We have the DNA and we have the RNA. So if you are talking about DNA and RNA, this is DNA, this is RNA. Why this is DNA? We said that is DNA because of one thing. Which one thing is because of the weak hydrogen bonds, while here there is no bonds. So identify the nucleic acid in the diagram 1. So we say that is DNA. Identify A. What is A? We said it's weak hydrogen bonds. Yes. And then you're saying that structure B, this is phosphate, phosphate group, or phosphate. Even if you say phosphate, you still get it right. Name the type of sugar found in nucleic acid represented in diagram 2. The sugar found there in diagram 2, because it is ribonucleic acid, therefore the sugar is ribose sugar. Yes, they are saying that which diagram 1 or 2 represent the nucleic acid used in the tracing of human ancestry. This one is we use what we call the mitochondrial DNA, therefore it's a DNA, therefore it's diagram 1. And then they're saying that paternity test, still we go for DNA test, therefore is still diagram 1. Yes. Then you go to question, the second and the next question. Next question is talking about the diagram below shows the karyotype of a gamete of an individual. So if you look at this karyotype, all of it, type of mutation is called chromosomal aberration or chromosomal mutation. So just say that that's chromosomal mutation. So the type of gamete which is formed here, the type of gamete, because now it has two gonosomes. So, and these two gonosomes are different. We can call it a hetero because hetero means different. Heterogametic, gami, gametic, heterogametic sex, or we can call it heterogamete. Yes, it's heterogamete. Or yeah, that's the most scientific answer. Or it's a haploid gamete. Definitely, is a haploid gamete. Yeah, and this will result in a person having being a hermaphrodite. Yes, so in this case, in so I'll call a heterogamete. So if you give a different answer, it could be correct depending on the scientific view of it. In this gamete, give the number of orosomes. Number of orosomes are this. Remember, they are single. Therefore, they are 22 orosomes. 22 orosomes. Not 22 pairs, it's just 22 chromosomes. Yes. And then gonosomes, if you look at them here, XXY. Or if yes, it is X and Y, this one, and then if it fertilizes a normal gamete which has a Y, therefore is going to have XYY. So these are the two answers which we are looking for in this regard. You get a mark here, you get another mark in that regard. Yes. So 1.6, the diagram below shows, in this case, what are we looking at? We are looking at the diagram below shows the evolutionary relationship between the different species. So in this case, we are looking for different species. Identify. We talked about this phylogenetic tree, gave you all the answers, whatever they were asking you. It's basically in that distinction material book. So if you go down, yes, those questions here being even shown what we are discussing about when we are trying to predict the paper which is gonna come. So everything were given in that regard. So they're saying that let's go and then see what are they saying. Phylogenetic tree. Yes, so we get a mark there, and they are saying genus that gave rise to Australopithecus africanus species. The genus which gave rise to Australopithecus africanus, Australopithecus at A. What genus is this? Obviously, is the first name. So we talk about the Adipithe. Yes, that's the Adipithecus, which called the Ad. That's the genus which gave rise to that. Then they're saying that the letter that represents the most recent common ancestor for both Homo sapiens and Homo erectus. So you guys, this is what I was talking about, that we asked you this, what is the common ancestor of Homo sapien, Homo erectus. So we talked about all these things when we are discussing about the paper which is going to come, and then our prediction was 97% right. It's only I only saw just one small question where we couldn't spot it out. All right. Name two species that existed at the same time, meaning that they competed for resources between for Homorilla, and then we have the Pan. So basically, these are the two species which competed for resources in that regard. Then we go to the next question. They're saying that give two give one example of fossil of Australopithecus africanus that was found in South Africa. In this regard, we are looking for the species. We said, I told you that always remember three species which were discovered in South Africa, that is Australopithecus africanus species. So in this case, we are supposed to say one of it, Taum, child. Yeah, number one, we can talk about Mrs. Place. Yeah, they say that this is a male, the female. Yeah, so if you wrote misplaced, but it's better to say Mrs. Place because that's what it is still in the books. And then you can talk about the little foot. Yes, the little foot. So it's Taom child, Mrs. Place, and then number 3, little food. They're just looking for just one. Yes, they are looking for just one. Guys, if you got 50 out of 50, congratulations. It's not 50%, it's 50 out of, so that the university or to be given a job or what. Yes, as long as you get decent, what we call the decent marks which is above your age. Yes, definitely. Yeah. But don't embarrass me. You get marks below your age. Guys, don't do that to me. Yes, it's like me spending the whole night trying to show you what's coming, and then you surprise me with such. All right, guys. All right, I know you did not do that to me. I know you guys, you got good marks. All right, let's go to question number 2. The diagram below represent the stage during synthesis of certain proteins. All right. So you have this, identify the organelle 3. This is a ribosome. It's a ribosome. Describe the role of molecule 1 in the stage of protein synthesis. So they're just trying to ask you to describe the role of transferrin A. Basically, you're describing translation. Yeah, remember transferrin A carries the specific or the required amino acid to the ribosome. So it carries this amino acid and brings it to the ribosome. So it carries a required amino acid to the ribosome. So this is 2. So automatically, if this GGA, so it's gonna be CCU. So 2 is gonna be CCU. And then they're saying on the DNA molecule that code for histamine. DNA molecule, this is histamine, automatically is gonna be GTA. Yes, okay, let me show you how did we get the GTA. This is transferrin A converted to messenger, which is going to be CAU, and then convert it to DNA, which they are looking for, which is going to be G. A goes with the T, and then T goes with the A, and then gives you rise to that. So basically, that's how it's supposed to be done, because they said on DNA, and what is happening here is transferrin A. Therefore, you convert it into messenger RNA, then you convert it again into DNA. It is just a reverse. During synthesis of some proteins, there was a change in the amino acid sequence because alanine was replaced with the tryptophan. Alanine was replaced with the tryptophan. Explain the possible reason why this happened for this. Why was alanine replaced with the tryptophan? Was AA during the formation of messenger RNA. So that now if you have GG, it's supposed to be GG. Yes. Now during the transcription, instead of G having CC, then it came to CAA. Eh? No. It came to CCUU. You understand? And then the transferrin A came to be GAA. And then instead of bringing the alanine, then it has to look for what code for GAA. GAA is tryptophan, so it's tryptophan which was brought instead of alanine. So in this case, where the problem came from is it came from during transcription, the two bases CC was replaced with the UU. You see, was replaced with UU, and then the code became CUU. You understand? Yes, and then tryptophan was brought instead of alanine. So that's how we're supposed to explain. At least you're gonna have to bring at least what here is what I told you, that emphasize this before, and then yes. So 6 marks, all the red thing means the marking point. Say that it's DNA double helix unwinds, yes, weak hydrogen bond break, unwinds, hydrogen bond break. So here there was another point which they include now to form 2 separate strands. Even if you don't write it, because you have too many points we can talk about, but if you wrote it, is good, because that's the best to form two separate strands. One strand act as a template to formation of messenger RNA using free-floating nucleotides from nucleoplasm. A messenger RNA is complementary to DNA, whereby A goes with U and C goes with G. And then, yes, and then you're saying that the messenger RNA now has a coded information for protein synthesis, which moves out of the nuclear pore, out of the nucleus via the nuclear pore to the ribosome in the cytoplasm. So basically, to the next question, they're saying that the diagram below represents metaphase 1 of meiosis. Guys, this is a cell membrane. I told you that when the cell is under metaphase, anaphase, we don't have nucleus, nuclear membrane, we only have cell membrane. So the answer here is cell membrane. State one function of A. Function of A is to produce production of spindle fiber, which are used to hold the chromosomes in their position. Yes. Explain the contribution of metaphase one in natural selection. So in this case, they are looking at how is this metaphase one going to help us to bring to bring about variation. Remember, variation is the one which help us to survive in the what? In the environment. So how is that going to help us to bring about variation? Yes, that's the meaning. That's what they're trying to mean. Other side. What if this one is here? Automatically they will form different combination. So they, because they align along themselves along the equator randomly, we don't know which one is which side, so they create different different gametes. Yes, these gametes, they have different chromosomes, and then these different chromosomes, they us in forming different individual who are going to suit or who are going to adapt to the environment differently. That's why you don't resemble your sister, you don't resemble your brother, you don't resemble your mother. Why? Because of the different combination. Or you have one, two characters, you resemble your sister or your mother. Yes. Uh, and the others, they resemble their neighbors. I don't know how did you resemble your neighbor. Yeah. Don't say that you don't resemble. Haven't you ever gone somewhere and then someone say that, oh, when you resemble someone I know? How did you resemble that person? Anyway, maybe the father was a great man. Describe the diagram below shows four possible daughter cells from the at the end of this meiosis. All right, so draw the cell D, including the fourth chromosome. So here they want just to see the variations which are happening. When I was explaining to this, someone asking me yesterday that, sir, do we need to shed? I told them, yes, you need to shed because those variations, those shaded areas, they mean marks. So I know that some of you, this one gave you a headache. How did you find it? But it was just a very simple thing. Let me show you. You come here. So the way how these chromosomes move is that all these, they will go, the last one they will go in one cell, the middle one go in another cell, they go in another, because they separate by two. You understand? So now if we say that black, white, black, black, so I will look for one black, one white, three blacks, so it means that it's that cell. Then I continue, I go to black and white, black and white, black and white, black and white. So I'll look for the one which has black and white, black and white, black and white. So it's this one. It has black and white, black and white, black and white. Also this one, black and white, black and white, black and white, black and white. So you see that it's the next cell which has black and white. One, you see the carvings are for this one. Yes. This one, you see the carvings for this one. Then automatically the one which is missing is this. So it means that you are supposed to draw this cell again, and then you add a white one, and then it's supposed to show how straight it is. Everything the way it is, that's how it's supposed to be done. And then maybe you put the labels, you label them, and then you put a caption, so that 1 mark for caption, 1 mark for label, and 1 mark for drawing a correct maybe diagram. Whatever marks they will be allocated, so as long as you drew it the way I'm telling you. Yes, so guys, you see, meiosis tend to be simple but difficult when it or complicated when it comes to the paper, but you just have to keep on revising and revising. Sex linked. Do you see anything here saying the sex linked? No. So no use X and Y. Explain why both individual 5 and 6 must be heterozygous for cystic fibrosis. Why are they supposed to be heterozygous? They're supposed to be heterozygous, these 5 and 6, because they produce a kids who are uh, having the disease, they're affected. So it means that one recessive allele came from 5 and one recessive allele came from 6. Therefore, they are supposed to be heterozygous for both conditions. Now, someone can say, why not small b? Small b is because they produce again individuals who are normal. Yes, they produce both individuals who are sick and or who are having the condition, and also those who don't have the condition. Therefore, they are supposed to be heterozygous. Use the letters B and C, okay, and then do the genetic cross to show the inheritance of fibrosis when heterozygous. Guys, this is just a simple thing. It's just a walk over for you guys. They just put this question so that you get those free marks. Yes. So they're saying that you have to say P1, obviously F1, that you tell us gonna be genotype is gonna be genotype. They said it is heterozygous, so it's capital B, small b. This one capital B, small b. Then you have meiosis. Meiosis is gonna take place. It's gonna be capital B, small b is gonna be capital B, small b, and then we call these ones gametes. Gametes. So you can draw a Punnett square method there, or you can just cross. So C, B with B, capital B, capital B, B with B, capital B, small b, B with B, capital B, small b, or small b, small b. So basically we call these ones offsprings, and then we call this one fertilization. For that one, fertilization. So in this case, that is how it's supposed to be done, and could be depending on whether you are whether you are they give a compulsory mark or not. So we come here, we give a mark for phenotype, give a mark for genotype, we give a mark for gametes, we give a mark for that, and then we give a mark for you telling us P1, F1, we give a mark for fertilization. Yes, you see all those marks. Maybe the compulsory mark was saying that you tell us the phenotypes. How many phenotypes? This is 3, 3, normal, 3, normal, or without 3 normal, without fibrosis, and then one is affected with the fibrosis. And then when you go to the genotypes, you have 1 capital B, and then 2 capital B, small b, and then 1 small b, small b. So basically that is it. That's how it's supposed to be done. So this question was just open for you guys to kill it and then get marks mahal. All right, let's go to the next question, that is 2.5. And then once we finish 2.5, we'll be able to have 100 marks. 100 marks out of 150, which is not a bad mark for you guys if you are a person who is looking for. Describe how genetic engineering of BTO was done. So in this case, they brought a gene of a plant which is resistant to those insects, and then they inserted it into this bt corn. Therefore the bt corn started to be also resistant, and then they kept on reducing the amount of insecticide they are using. So they just crossed the gene for a gene which is from a plant which is resistant to those insects. Yes, remember insecticide, they kill the insects. Yes, so that's how it became to be more resistant. So describe the relationship between the land and the grow BT corn and amount of insecticide. land to grow bit acorn and amount of insecticide. So basically they're asking you to describe, they're not asking you to explain. Explanation, you require a reason why. So in this case they're just looking at, as the number, as the land size increases, yes, the amount of insecticide used decreases. Yes, as the land size increases, yes, land size used to grow Bitcoin, yes, increase the amount of insects. Two other plant characteristics that are genetically engineered to increase food security. You have two other plant characteristics. They're looking for characteristics. You can talk about the size of fruit, the size of fruit. You can talk about the shelf life. Yes, how long does it take stay in there? You can talk about the sweetness of the fruit. You can talk about the juice. You can talk about, there are so many things you can look at just to increase the food security. Then lastly on this question is draw the bar graph to represent, let me see, the percentage land used between that year and year. So they are looking for only this. They are not only this and this. Therefore, year, since year is time, always is supposed to go on the x-axis. So the years is supposed to be here, 1995, 1995, 1995, 2000, what did I say, 2000, 2005, 2005, 2010. So like that. So maybe it's 0, this is 10, or 24, then the fifth one is 60. You see, yeah, remember when you're marking, don't forget guys that we're supposed to check here the size of this and the size of this are the same, size of this and the size of this are the same. Yes, also the space here is the same. Yes, the space is the same. Then now we give you the caption. Caption must have two the variables, this variable here and a variable here. If you look at here, this variable is saying that land used to grow Bitcoin in percentage. So you tell us that land used to grow Bitcoin and then you put there in percentage. So we come and give you labeling the axis correct, and then you look at the scale. See the scale correct, then we also give you a mark for you having a correct scale. Then we come here, we give you maybe 2 marks for plotting properly. Then you give you a mark for correct type of the graph. Then we give you a mark for caption. You see, yes, that's how you accumulate marks. So you give 1, 2, 3, 4, 5, 6, yes, one, maybe any other mark depending on their marking guideline. Yes, so you see that this question was also simple. You were able to get it. I would have kept myself on Thundereduc because really we try to give you everything you need. Yes, and I repeat, thank you guys, those who came back to me and said thank yous. Mr. M. Saidi, you really helped us, and also I'm saying thank you very much for not letting me down and keeping yourself on our channel. A certain country, 20 to 25,000 individuals were suffering from hemophilia between 2012 and 2020. Hemophilia is a genetic disorder caused by mutation. Pie chart below shows the percentage of individuals with the different levels of hemophilia in this country. All right, then you're saying that the percentage of individual, okay, cool. Explain the effect of mutation on individual. The mutation basically it causes the blood group the blood not to clot. You understand? So you lose a lot of blood in in a situation. Yes. Calculate the number of individuals who were moderately affected by hemophilia in the country. Show you're working. In this case, because 1,250 people, so those are the people who were moderately affected by hemophilia. They're saying that explain why it would be expected that most of the individual affect hemophilia will be males. Basically, the reason why is like that because males contain 1X. If a recessive allele appears on X, the individual is already affected by hemophilia. Why not female? Because females have 2X. If a recessive allele appears on 1, it can be masked by a dominant allele and the individual is still normal. Yeah, that's why you see that it's very common in males. Why? Because males have 1X chromosome. If a recessive allele appears on it, the individual is automatically sick. Yes. Then we go to the next question, which is 3.2. 3.2, what are they talking about? Here they talked about the insects on island with tree are likely to be wingless because they fly dangerously for them in area with a strong wind. Yes, you see, stoneflies have undergone a natural selection. Some have wings and some don't have wings, depending on the area where they are coming from or they are found. The diagram below shows the stoneflies with and without the wings. So here is with the wings, here without wings. Scientists wanted to determine the relationship between the presence of trees and the wings on the stoneflies. The presence of tall uh, tall tree and wings on the flies. They selected 6 locations, or which 3 had tall tree plants and then without tall tree plants. Using specialized net, yes, collected thousands of stonefly. This one is going to help us when you are talking about increasing the reliability. Yes, stoneflies, the location, the sample was labeled according to the area. Yes, these samples were all collected in the morning. Yes, you see the number of stoneflies wings and without wings at each location was counted and recorded. Presence or absence of tree, yes, on the island, of tree on the island, yes. And then this one is still presence or absence of the wings. So the wings will depend whether there is a presence of trees or there is absence of trees. Yeah, this is just a general question. State 2 factors that were kept constant. In this case, um, the factors which were kept constant is the collecting time. They were collecting in the morning. Yes, collecting time, and also the method of collecting. Yes, they used the specialized nets. Yes, method of collecting, it was one, and the time of collecting was one. There are even others which you talked about. I have not talked about them. Yes, they also might be correct. State 2 ways in which the reliability of this investigation was ensured. Yes, was ensured. So standardized data collection, it is just a standard. The way how you are collecting data is the same. So that increases in reliability, and also replication of these uh, individuals. So in this case, if you look at them, you'll find out that we used thousands of. It would have expected the more stoneflies with no wings in area without trees. We expect that because of the natural selection. How did natural selection? Those flies with wings could fly and then they're being blown away by strong wind. While these ones, you see, yes, by strong wind. While those ones without wings, they were holding themselves on the ground and they could not fly. Since they are holding themselves on the ground, therefore they could hide from predators. They survive, they produce large numbers, and they are able to increase their population. Yes. So basically, those which had wings, they fly, blown away by by the wind, and also eaten by predators. Those without wings, they could not be blown away by the wind. They could hide from predators. They could survive. They will reproduce in large numbers. Basically, that is it in that regard which is natural selection. While here describe how Lamarck would have explained the evolution of stoneflies. When I was talking about Lamarck, only people know that the more used, the more frequently used the structure, the more becomes bigger and stronger. If you don't use it, becomes smaller and being applied. There was no variation among these stoneflies. All of them had wings. Yes, the stoneflies started to use the wings less frequently, and then the wings became smaller. You understand? And finally disappeared. This character of no wing was passed to the next generation. Then the next generation were born with no wings. That's it. That's how Lamarck explained. There was no variation among these flies, meaning that all flies had wings, and then these flies started to use the wings less frequently. Then the wings became smaller and then disappeared. This character was passed to the next generation, and then all the flies were born without wings. So that's how you're supposed to explain that in that regard. Yeah, you see that the paper was not that bad, just some question required you to apply. Then they're saying that describe the process of speciation through geographical. This is another free mark, which I'm not going to take too much time in that regard. Explain this and emphasize this during the scope time or during the yes. Say that yeah, this question will come. It is here. Mm, okay, it's here. It's here. Punctuated. No, 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 no. That one we explained it. Yes, you see when it's highlighted nicely, it told you that 6 marks, 6 marks, it could be 7 marks. It doesn't exceed 7 marks, but it can go to 5 marks. So describe the process of speciation. This question is asking you, describe the process of speciation by the geographical isolation. So in this regard, what are you supposed to look at? You come here, you tell us that a population of a particular species were split into two by a geographical barrier, for example, a mountain. As a result of two parts of population cannot interbreed. So when you separated it, the two parts cannot interbreed. Like your home, the left side and the right side is not the same. You have different environmental conditions. Even in your class, there is a party spot you like to sit in when it's winter, like that part where maybe next to the window where the sun is coming, you want to bask the sun. Just a class, they don't have the same environmental conditions. What about when the geographical barrier is separating them? Definitely don't have the same environmental condition. Because of the geographical barrier, there is no gene flow, they could not cross each other. Typically different. Yes, they became genotypically and phenotypically different. I don't see people asking me questions. Ne? Maybe you got it right. Ne? Yes, if you have a question, let me know because I'm on live. Ne? It's not just talking. Ne? Not just talking. Ne? Yes, I'm on live. You understand? Yes. So if you what when you say as a result the two population became genotypical, you start with the word genotypically before you go to phenotypically because it's the genotype which determines the phenotype. Yeah. Even if the two population mix later or again, they will not be able to interbreed. Yes, produce a fertile offspring. The word is fertile, not viable. So therefore, both or one or both population has new species. That's what they were looking for, so that you get your 6 marks in this regard. Mahala. I call them Mahala marks. Yes. Then we go to the next question, saying that list three. The diagram below represents the skull of hominid. All right. These three similarities between for correct judgment. Name one species in the diagram that is prognanthus. In that regard, you are talking about the gorilla. It's gorilla, which is prognanthus. Name one species. All right. Describe two structures that cause the species to become prognanthus or more prognanthus. First of all, we talk about the protruding jaws. Secondly, we talk about the flat face. The face is flat, therefore is the protruding jaw, and then the flat face. Those are the characters which we can also talk about the position of foramen magnum, but they said, yeah, two. All right. Hominid was a bipedal for most of its time or adult time. Yes. Explain how the structure of hominid skull would have assisted in bipedalism. Yes, the position of foramen magnum is a little bit forward, therefore it helped in connecting the spine vertically, and then is able to walk onto limb. Yes, the position of foramen magnum is a little bit forward, which helped to connect the spine vertically, and then it was able to walk onto limbs. So basically that's it. Describe the difference. One is short and wider, wider, and then this one is long and narrow. So that's if you go to our distinction material book, you'll find out that we highlighted this very well when it came to that. You see, it's short and wider, and then long and narrow. You see, long and narrow, and then this one less curved or C shape, and then this one more curved. Yeah, so everything was there, just that sometimes because of pressure, because of panicking, we panic and we end up forgetting our answers. Ish, that's a very bad thing, eh? Yes. Explain why gorilla species have cranial ridges. They have cranial ridges. Cranial ridges are these ones. Cranial ridges are these ones. Yes, these things. Yes, these ridges. Yeah, it's not like human. They have cranial ridges for muscle attachment. Muscle attachment because gorilla they feed on raw food. Yes, so basically is for muscle attachment because it requires a strong is something. It looks like how do they call it? It looks like the trastramonia. It looks like a eggplant. Yes, it looks like eggplant, and it is very toxic. Yes, it used to kill soldiers. This thing is there. Flower open in evening and then release a powerful fragments which attract hawk moth. The Datura produce a high they produce highly addictive nectar, which highly addictive nectar. Yes, which do what? Yeah. Which ensure that it ensures that the hawk's moth stay longer inside the flower and can only visit the trastramonia, the flowers. So they're asking you explain two ways in which yes, explain two ways in which this tetra is ensure. Explain two ways is a key point. Basically what they do is they are very important. They help in number one. They tend to open the flowers in the evening. Yes, so if they open the flowers in the evening, you see, so it means that other time of the day they are not able to do that, so they are a little bit isolated, and then they attract the moth. Yes, so basically this is they have specific pollinator attraction, which is addictive. Yes, addictive. Yes, this one which is addictive. So they try to produce a high addictive nectar, which ensure that the moth stay longer. Yes, you see. So opening at different specific time is basically issue. Yes. Set two other mechanism by which by other mechanism that would ensure reproductive isolate. It can be basically special features on the plant which do not allow another plant to carry out cross pollination, or it could be isolated geography.
For our life science grade 12 2025. So I'm a little bit happy that we started together, we have concluded together, thank God. I'm also happy that you are here to check and see how far we have gone. I thank everybody who tried to join our classes. Before we start, let's do this and we see what. I know that you have been hearing that sound. It shows that the sign that the Thundereduc has arrived. It means that we are moving like a bullet. So if you want your marks to increase like a bullet, you have to join Thundereduc. You can tell your brothers, sisters, and others. Anyway, let's start without wasting time. I hope everyone is hearing me. Before I start the paper, I'm going to use 1 or 2 minutes just to remind you something very small of what we did last time. All right. Let's start. Remember our prediction or our things we did. When I checked the paper, I've not even I've not taken all the time to check the paper versus to what I told you to read about our scope 100% project. We see that this was there, monomers of DNA, nuclear, other types of nuclear DNA, other debates of profiling this structure. It was related to that. They brought about this ribosome. They ask you even it was the same circle. They brought almost everything. When I check, they brought this, which is like this. When I was discussing, I tried to explain. They brought Down syndrome and asked you to show you how to do that. They brought about the definition of this. They brought this, but in a different angle. But I explained it. They brought all this. This one they brought exactly the same question about the same disease. They brought. Actually, when I saw the paper, I saw that people who used our discussion we did, I think you got it right. And we pray that you get all this. They brought about it. They brought about the locust. They brought about the species. They brought... what should I say? Why shouldn't I say? They brought this time. They increased the generation to 60. Then I used the one for 30th generation. They brought these one skulls. They talked about the comparing the skull of the specifically the skull, not any other part. They brought about the 3 factors about bipedalism, what causes. Yes, they brought it. What can I say? They brought these calculations. They brought about these calculations exactly. They brought about the out of Africa hypothesis, other factors which is about the fossil to use it. Actually, you guys, you did, I hope you did a great job. Let's see how we're gonna go through the question paper, and then we see how much you have got. We thank everybody who tried to join our class. And we tell you that this is not the beginning and this is not the end. And remember, not the best in class is the best in life. Not the person who gets the most marks is the one who succeeds in life. So please don't be worried about the mark. The mark do not determine the life. However, the mark can facilitate the life. So let's start, guys, without wasting time. All right, we are starting. The question is saying that, I don't read the instructions. They're saying that gene. What's a gene? A gene is a section of DNA that control a particular characteristic. That is it. Ne? So it means that set of chromosome, nope. They're saying a set of chromosome segment. That is it. Segment, but is it? Coding for a particular characteristic look to be much more correct. They're saying the haploid number, nope. They're saying triplet bases that code for an amino acid. It's not just a triplet. Yes, they are triplets, but not just one triplet. So nope, it's supposed to be a segment. A segment is made up of many triplets. So B becomes the most correct answer. We go to the next question. Let me check something here. Segment. Cool. They're saying that significance of DNA replication, we talked about it. Yes, we talked about it. In the cell formed at the end of mitosis. The significance of DNA that is formed at the end of mitosis. Remember, mitosis, the cells are the same. So genetically identical chromosomes or cells. So that looks to be the most correct answer. Half the number, nope. Genetically different, nope. Half the double the chromosome number, nope. These other things are the importance of meiosis. The remaining one is the importance of mitosis. So A looks to be the most correct answer. Why am I shaking? Don't shake, Buffett. Which one of the following statement is a feature of punctuated curium? I told you, define about the punctuated curium. Okay, remember we said that long period of no or little change with alternating with rapid period of short period of rapid change. So we have to look for that. Individuals organisms adapt to changes of environment. Nope. The inheritance of acquired characteristics. Nope. They're saying that the long period of little or no change. That's what I told you in that. Some of them I didn't even tick them because our spot was around 98. We shifted from 95. Yes, for the case of paper 1 was 99%. At least last year we were 95%. This time it was 98 paper 2 and 99 paper 1. Yes, so tell your brothers and sisters that the thunder moves like a bullet. All right, we are continuing to the next question. So this one looks to be the most correct answer, which is C. Long periods of little change and then or no change at all, but the periods are supposed to be longer. Remember that we say that it's like a ladder whereby you have long period of no change and short period of rapid change. Long period of no change or little change with rapid period of with the short period of rapid change. So that is punctuated equilibrium. We talked about hemophilia. That was also there. That the disease characterized by lack the disease which is characterized by the disease which or disorder which is as a result of failure of the blood to clot is regarded as what? And then we also brought it as a question that hemophilia is a sex-linked disorder. Don't want to talk about it, but I just want to show you something here. They said hemophilia is a sex-linked disorder, and then here they said a mutation caused by a mutation on X chromosome, meaning that it's a sex-linked disorder. Yes, sex-linked disorder. So B looks to be characterized by inability or distinguish between red and nope. That is color blindness caused by non-disjunction. Nope. That is Down syndrome caused by allele carried on autosome, not autosomes, sex. So meaning that B looks to be the most correct answer. Autosomes are body cells. We go to 1.5. Which one of the following is an example of heterozygous? Hetero means different. Different may be X and X. X. Okay, D and small d, something like that. Okay, they're saying that if you look at hetero means different, so it look at this, it's the same. So this is homozygous, this is also homozygous, this is sex linked, this is non-sex linked, so it's out. This is homozygous recessive, this is out. So C looks to be the most correct answer. RW, this is heterozygous, so that is to be the most correct answer. We go to the next question. We brought this question but in a different version. Did you pass it, guys? Let's go and then you find out what is it all about. Which one of the following is the combination? Which one of the following is a combination correctly? Okay, here we say that this diagram below shows what? DNA replication. So okay, now I know that this DNA replication. So they're asking you that which one of the following combination correctly identifies molecule 1, 2, and 5? 1, 2, 1, 4, and 5. Okay. Let's go to molecule 1. What is molecule 1? That molecule 1 is this molecule. That's the reason why I'm giving you molecule W. Done. All right. Life is good, man. Don't worry. Thundereduc, Said. Can't cook and then the food doesn't become nice. The food must become nice. When I cook. When I cook for you guys, my students. All right. We are saying that's it. So it's a nucleotide. So I know that it's a nucleotide. So whenever I see a nucleotide, I mark nucleotide. So this one, so B is out. So if you wrote B, you are gone already. Number 4. Where is number 4? Number 4 is, since they say it's DNA replication. See, they say DNA replication. So what is number 4? Number 4 is deoxyribose sugar. Deoxyribose sugar is the same question here. I asked you that what is Y. All right, we continue. Then they're asking you the next question, the number 5. Number 5 obviously is the phosphate group, and is the same question here, which is XC. They just changed the numbers, and then so A looks to be the most correct answer. We go here. Which one of the following combination is correct for number 2 and number 3? Number 2 and number 3. So let's go to number 2. Where is number 2? You see here they brought it. Okay, number 2. I gave you an idea of using this. By using this, you guys, you remember that what is this thymine? What is this? It said it's adenine, but you have to look at, because here is thymine, so you look at it and then you are able to identify what is V. So basically that's what they're asking you here. So what is this? So you need to understand the concept that, this. If this is 2, what about, okay, now what about this? So if I can know this, remember this thing we are together. So this is the first nucleotide, this is the first nucleotide. So if this is A, automatically this is A. The other strand must be T. And if this is T, automatically number 2 is A. So I'll come back here and I say that number 2 is A. So I know that, number 2 must be A. Okay, then I go to the second one, which is number 3. Number 3 is the same story, is the same idea which I gave you to use. Hey, I told you. We are saying that, Akara, I need to show you how did we get to the answer. Hey, don't rush bafethu. Yeah. Then we say that we continue, and then we say that number 3. Where is number 3? Okay, number 3 molecule. Number 3, they say that number 3 is 1, 2, 3. So I count 1, 2, 3. So if this is thymine, automatically this is gonna be adenine. Therefore, you're gonna be adenine. That's the answer. So B becomes the most correct answer. All right, we go to the next one. Question 1.8 to 1.9 refer to the information in the dihybrid cross. Okay, dihybrid cross. Did I finish the objectives? Not yet. Let's talk about the dihybrid cross. Actually, even this one was almost similar to the question which I gave you in the previous. All right, let's continue. So you guys, sometimes I have to a little bit to explain, because there are some other people gonna be watching this video. For example, those who are upgrading, those people who are going to be using it for next year. That's why I don't just need to rush. However much I'm rushing, but I don't need to rush. I need to show you where is the answer coming from. Yeah, it's not just for guessing. All right, in certain plants species, on one gene controls stem color and another controls height. This is the height, and then the other one controls the height. Okay, they're saying the stem can be brown. Brown is capital B and red for small b, while the plant with the height, tall is capital T, short is a small t. All right, when plant 1 was crossed with plant 2, plant 1 is brown, so we see that brown is small b, and then brown is dominant over what? Brown is capital B. It's gonna be capital B. It can be capital B. All right, let's look at it like that. Or capital B small b, capital B small b. Did I say it's homozygous? Okay, we shall see. And then they're saying that red means that this is small b. And then they're saying that this is tall, it can be capital B, capital T, or capital T small t. And then short, automatically small t. All right, let's continue and find out which one of the following represents the genotype of parent of plant 1. Plant 1 is here. Okay, it must be having small t. I need to look for small t. So small t. So these one are out. Oh, all right. And then plant 2, I can look at red is small b. So I have to look for small b. So this one, remember this one I said is correct, but it's not small b. This one I said is correct, small b. So this one looks to be correct. So this one is already, yes, is correct, but is already out from this side. So this one is also out. So the most correct answer looks to be B. So B becomes the most correct answer. All right, they're saying that the number of possible different genotype expected in the offspring in cross this. Okay, when you cross them, what do we expect it to be? When you cross them, what are we expecting it to be? So it's gonna be let me scoop out the genotypes. Is B, small t, and then this one is gonna be small t, small b. I'm reading it from here. Yeah, I'm reading it from there. So it's small b, capital T, small t. So since this one are the same, so I'm gonna take one. And then this one is gonna be, okay, so it's gonna either be, capital B, small t, yes, or it's gonna be small b, small t when I combine. Okay, some of you didn't get how did it come about. I'm saying this is how it was. This goes with this, it's gonna be capital B, small t. This goes with that, it's gonna be capital B, small t. This is gonna go with that, it's gonna be small b, small t. This one is gonna go with that, it's gonna be small b, small t. So automatically I'm gonna say capital B. You see, they said different because the key point is saying different. So this one and this one is the same. So I'm going to take capital B, small t is different. And then this one also is going to be small b, small t. You understand? And then now this one is going to be, it's the same story. This is going to be small b, capital T. This with this is going to be small b, small t. This one is going to be, you see, small b, capital T. This one is that, small b, small t. So you see that this and this, they are the same. So I cancel out one. This and this are the same. So I cancel out one. So automatically, my genotypes are going to be capital B, yes, small t, yes, this one. And then small b, small t. You understand? And then now, let me clear up this one. Then this one is gonna be small b, capital T, and then small b, small t, like that. So basically, when I cross, you'll find how many spaces do I have? 1, 2, 3, 4. So there are 4. So the answer is A. So if you cross them, you're gonna form 4 different individuals. You understand? When I cross, that's because they said from the cross. You understand? When you do, they're gonna be 4. So the correct answer becomes A. So that is it. Then we go the next question. Let me clear up here. They're saying that a rare form of rickets. Rare form of rickets is caused by a dominant allele on the X chromosomes. I explained this yesterday very well. Which one of the following statements is true about the inheritance of this form of rickets? Which one of the following is true? If the father is affected, all the sons never, because the father will, to the sons, he will give you the recessive, the Y chromosome. So this is out. If it will be expressed more frequently in males than females. Not dominant allele, that is only for recessive allele. Out. If the father is affected, all the daughters will be affected. Yes. Because it's caused by dominant allele. Look, guys, if this is capital D, and this is why, because the father is gonna be giving to the girls an X. So I don't care what is on these ones. Yes. So if this goes with this, it's gonna be X, capital D. Whatever is coming from this side, I don't care. Then this one to this is gonna be X, capital D. You see, and another X. Whatever is here, I don't care. So you see that the females. Because now if I cross this, it's going to be a male. You see? So the father will not give any allele to the male. It's only the mother. So basically all the daughters will be affected with the disorder. Therefore, C looks to be the most correct answer. If it will be expressed in a female, since they have two, it will only be expressed in females, since they have two. No, even males, it will be there. Even males, they will have the dominant allele. Who got 20 out of 20? Okay, who get 18 out of 20? Who got 18 out of 20? Oh, I see some people getting the yes. Thank you so much, guys. Let's go to terminologies. Terminologies. Where are you? Who got all the marks in terminologies? Let's start. Bond formed between two amino acids. Peptide bond. Remember, we don't call them polypeptide bond. Peptide bond. I told you yesterday. An organelle that contain DNA, which is used to trace the female ancestry. We said it's a mitochondrion DNA. Mitochondria is a mitochondria. There's an undifferentiated cell that have the potential to become any type of cell division. I don't want to go back to show you these things. I don't want to go back because it's going to be taking a lot of time, because you saw those people already. This question also was there. Even this one was there. Okay, let me just continue. We call them stem cells. Not cells of the stem, stem cells. All right. A change in the sequence of nitrogenous bases resulting in the formation of different proteins. How do you call that? We talked about it. We call it a mutation. Mutation, but the same sequence of nitrogenous bases. So it's better to say gene mutation. Yeah. I hope they will mark gene mutation or they will mark mutation. Yes. All right. Natural shape of DNA, we have been singing a song on this. Double helix. Double helix. Helix. Okay. Permanent disappearance of species from Earth. Even a grade 10 can answer this one. Extinction. The type of variation which intermediate. We talked about it and give an example of a head and skin. Yes, we call it continuous variation. Continuous variation. Yes. They're saying an allele that is expressed in the phenotype in heterozygous. We told you yesterday the same thing. Is expressed in the phenotype of heterozygous state. We call it a dominant allele. And then someone was saying, no, it's not allele, it's allele. And I say, okay, it's allele, whatever. Dominant allele because it expresses itself in the heterozygous state. All right. Position of a gene on a chromosome. Ay, bafetu. Hi. Hey, how do you call that? We said you call it a locus. So what I did is I say, what is a locus? So that you know what is the position of a gene on a chromosome, position of an allele on a chromosome. We call it a locus. Locus. Good. I see people, they are bringing a lot of locus, buffet. All right, I see people are hot. Yeah. It means that you are on our discussion when you're busy talking about. Guys, don't mess up. Always watch Thundereduc with M. Saidi. You will never regret. Who got 9 out of 9? 9 out of 9. All right, we continue. Guys, we are counting distinctions, and I told you have to be better than me. That's the idea behind this. You have to be better than me. All right, we continue. They're saying that the phase of meiosis during which non-disjunction occur, we said it. Non-disjunction, the phase of meiosis during which non-disjunction occur. It occurs in both anaphase 1 and anaphase 2. So that's why I said that the failure of the chromosome to separate during anaphase. They don't say during anaphase 1 or anaphase 2. Or they don't say during anaphase 1. Or they don't say during anaphase 2. No, they say during anaphase. Or during anaphase 1 and 2. So both A and B. Both A and B. Alright, we continue. Representation of number, shape, arrangement of all chromosomes in the nucleus. How do we call that? We talked about it. It's a karyotype. And someone is asking me, why bring the karyotype? I knew. Alright. Alright. Don't ask me how did I know. Unless I don't have brain. Alright, we continue. The allele of genes separate into different gametes. I even ask you, state Mendel's law of segregation. I didn't want to talk about Mendel's law of independent assortment. Me, I wanted the Mendel's law of segregation. Because I knew. So state it. Alright, the answer is B only. And even this one is B only. Alright, we continue. Hey guys, hey, life is good. I know that you are killing this paper. Who got 6 out of 6? Life is good. Who got 6 out of 6? Yes. Who got 6 out of 6? You see? I told you. He's here. State Mendel's law of segregation. The locus is here. The dominance is here. The dominant. Let's continue, man. Let's continue. Let's kill it. We continue. I brought this for you to label. I know this one you killed it. I told you that once it's not in a prophase, you can't see the nuclear membrane. So A is a cell membrane. B is a this is a spindle fibers. Yes, Buffet. Yes, Buffet. I had a reason, and even I told you answer that. I had a reason. Okay, we continue. What is C? Spindle, centrioles. Centrioles. Those are centuries. I had a reason. I told you to answer this. I had a reason. Not to. We continue. Which one of the following W, V, W, X, Y, and Z represent a cell during prophase? During prophase. We continue. Prophase. For that reason, have to drink some sip of water. All right, which one shows I got you here. Prophase of mitosis. Now here, do you know what you're supposed to do? Someone said nuclear membrane. No. I told you this is not nuclear membrane, Buffett. I said this is the nuclear membrane. That's the nuclear membrane. This is the cell membrane. It's a cell membrane. This is the nuclear membrane. I explained very well that after prophase, that's why it's dotted, so that it's gonna disappear. After prophase, you don't have nuclear membrane. Where were you starting from yesterday? All right, we continue. Which diagram represents the cell during prophase of mitosis? Now here, let me clear it up. This is the part which made me lose the 99, the 100%. Yeah, they brought this question. I didn't think about it. Okay, this question, this one I'm going to answer. They're saying prophase of mitosis. In this case, remember in meiosis, now we need to identify. But I asked you to do the difference between mitosis and meiosis. So if you had that, then you are able to identify it. Okay, remember in meiosis, chromosomes align in homologous pairs. They're in homologous pairs. While in mitosis, the chromosomes are single. You understand? All right. Now let's check. They are pair. You see? This shows his prophase. Even this one shows his prophase. But one is prophase in meiosis and one is prophase in mitosis. Check this one. Is alone. 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 You see? So this is mitosis. This is meiosis. Because they have to align along themselves so that crossing over can take place. So, in this case, we're gonna say W is mitosis. W. We call it as W. W is mitosis. Metaphase 1. Metaphase 1, they are arranged along the equator. Along the equator. This. All right. That question, this one was there. Even as you draw a cell after that. So it's gonna be Y. Prophase 1. Remember, mitosis doesn't have prophase 1 anaphase 1. No. So this is prophase 1. So it's gonna be V. V here. W H Y V. How many chromosomes will there be in each cell at the end? Did I say this is a human cell? I told you, don't talk about human cells. How many cells will there be at the end of mitosis? The way they are, the way they will be. It's gonna be 1, 2, 3, 4, 5, 6. They're gonna be 6. 6 chromosomes. 6 chromosomes. So the answer here is 6. We continue the next one. Just a moment. Yeah. All right. We continue the next one. Just a moment. Yep. Okay, we continue next one. How many chromosomes would there be in each cell at the end of meiosis? Meiosis, remember, there are how many? It's 1, it's 2, it's 3, it's 4, it's 5, it's 6. At the end of meiosis, there is nuclear division. So it means that the number of chromosomes, they become half. So it's going to be 3. Hey, someone is asking me what is Z. They didn't ask you about Z. Did they ask you about Z? Why do you bother about Z? But Z looks to be prophase 2, because they already have the variations, you see, and the chromosomes are single, and because of this shows that is prophase. So that's prophase 2. Yeah, prophase 2. Yeah, and since it's 1, if there were 2, maybe it would have been hey, who got 8 out of 8? 8 out of 8. All right. Oh, who would have got 8 out of 8? All right, we continue to the next question. You guys, I hope you didn't disappoint me. Let's continue, guys. They're saying that the diagram below shows represents the stage of protein synthesis. Each amino acid and PQR is are coded by different codons on this messenger RNA. All right. The stage, identify the stage of protein synthesis shown above. You see it here, and then you ask, you say ribosome, and then you ask, you say ribosome, and then you ask. They're asking, they only brought this part. You see it there, and then you ask. So it is translation. 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 What is molecule 1? What is molecule 1? What is molecule 1? T. You see it's a T. Say T. So it's tRNA. It's tRNA. tRNA. What is molecule Z? We have been talking about it. Is a ribosome. Yes, I told you it. Yeah, when you were busy complaining about this, I said which molecule 6. I had a reason, Bafeto. We continue. All right. Then they are saying that state the sequence of nitrogenous based on DNA molecule for codon. This the same things on that question. I don't want to go back there because it will be wasting our time more. State the sequence on number 3. Okay, number 3. Where is number 3? Number 3 is here. Number 3. They are saying that state the sequence of nitrogen bases on molecule for codon. Okay, number 3, DNA. So if this is messenger, so I'm just gonna change it. It's gonna be C, it's gonna be G, it's gonna be T, because it's DNA, so it's a T, not U. Done. We go to the next one, number 4. Number 4 is not there. Let me check. Okay, now you know what you're gonna do? We don't know what's number 4. We don't have any idea. They didn't label it here. We don't have any idea. So they want, what do they want? They want write down the sequence of DNA from number 4. So I need to find out what is number 4. So you know what I'm going to do? I'm going to say CGU is a transfer RNA, anticodon. So CGU goes with what? C is going to go G, C, A. So what is CGA? So it's this one. So this one becomes 3. So that one is out. Then I'm going to go GCU. GCU going to go CGA. CGA is here. So this is number 5. Okay, let me go to this one now. GCUA is going to go with GAU. GA. It's going to go GAU. Where is it? GAU. This looks to be number 4. Okay, let me go to this one. AUG. A goes with U. U, A, C. Ah, yeah, this is number 2 because this is this one. So automatically this is number 4. Number 4 goes with this. But they want the one for DNA. So automatically it means that if this is CUA, it's going to be GAU. Then on DNA, it's going to be CTA. So this is what is going to be on DNA. So it's going to be CTA. CTA. So that is the answer. Someone is saying it's GU. It's going to be GTA. Why do you say GTA? No. Is it GTA? No. Okay, let me check it again. It's gonna be this one. Is gonna be C because this is anticodon. I told you it's anticodon. You change it to codon, and then you change it to code. That's what I told you yesterday. Then I say that if this is C, it's gonna go with the G. If this is U, it's gonna go with A. If this is A, it's gonna go with U. Wabona? Now I have to convert it into code, which is the DNA, because they want it on DNA. So it's C, G is gonna go with C, A is gonna go with T because it's DNA, and then U is gonna go with A. So it becomes CTA. All right. CTA is the most correct answer. You got 7 out of 7. Oh, which amino acid PQRS is coded by number 5? Which amino acid PQRS is coded by number 5? So we go back and find out what is number 5. Amino acid coded by number 5. Yeah? PQRS is coded by number 5. Oh, so we saw that this is 5 is here. So it's going to be P. Because this is CGA. So when I transfer it, it's going to be GCU, which is this. So it's P. So the answer becomes P. Hey, but guys, the paper was fair, ne? Yeah, the paper was fair. Someone saying 45 out of 50. Congratulations. Who got 50 out of 50? 50 out of, okay, 48 out of 50. Okay, 47. Okay, 46. I only see 45. Oh, I got someone for Lungelo Koza 46. Chayani say 46. 31, 44, 45. Thank you guys, ne? Thank you. Sipesihle. Is a specific. We're just laughing at. Oh, I have Ontile 48. Oh, Ichum 50. Congratulations, guys, ne? Yo. I know that you guys were here at Thundereduc. Thank you so much, guys. Let's go to the second question and then we smash it. I told you, here at Thundereduc, we smash the paper. We don't play. Have you ever seen a thunder playing? No. We smash. Our slogan is smash it. All right. All right. Sai is the best. Lolo is the best too, Priscilla. All right, guys. The diagram below shows. I gave you this diagram, but it was in a different angle. I explained it, but it's in a different angle. That one. I had a reason for that. All right. Let's continue. All right. Let's start. Identify the structure labeled A. Structure labeled A. Yes. Structure labeled A. That is a centromere. The central body. Centromere. Biological name for sex chromosome. We said they are called what? Gonosomes. They're saying that describe how the chromosome would differ in the somatic cell and of a male. Describe how these chromosomes will differ in the the gonosomes will differ from that of a somatic cell or of a somatic cell of a male. The somatic cell, then they will be identical, they will be homologous. homologous. You understand? Well, this one, it will be for gonosome, for this cell, it will be X and Y. It will be X and Y. So basically, the chromosomes in this individual describe how this chromosome will differ. Yeah, there will be this one. There will be X and Y, which are not homologous. So the other ones, in the somatic cell, they will be homologous. So you are supposed to talk about X and Y. All right. An exchange of genetic material takes place at point P. Okay, we are listening. Describe the process. That's the process we talked about. Crossing over. Identify the phase during which this process takes place. Guys, we say this what? Prophase 1. They're saying that. Give one reason why this process mentioned above is important. Guys, it's the same question Z I asked you yesterday when I gave you my scope. We kill two. We propose the scope for reality. All right, give one reason for it. Brings about genetic variation, and I told you it must be genetic variation, not just variation. Genetic variation. We continue the next point. Genetic variation. Explain how the structure of chromosome would differ at the end of meiosis. Explain how the structure of chromosomes will differ at the end of meiosis. Basically, each chromosome will consist of single chromatids which are non-identical. Yes. It means that, remember, they will be like this. Like this. And all of them, they will be non-identical. They will be single chromatids. They won't be two chromatids, no? Single chromatids, and these chromatids will be non-identical. Each chromosome will consist of single chromatids which are non-identical. This one I also give you the question. Non-disjunction of these chromosomes occur during meiosis and the gamete with an extra chromosome is formed. This is if this gamete is fertilized by normal gamete, by resulting zygote will not be normal. Okay, let's continue. Explain the genetic composition of the zygote. So basically, in this case, since there are 3 marks, there's a non-disjunction of these chromosomes occurred during meiosis, and the gametes result in extra chromosome. So it means that I'm going to have a gamete which has an extra chromosome. Okay, cool. And it's formed this if the gamete is fertilized by normal gamete. So I'm going to have another gamete with the normal number. Okay. Result in the zygote will not be normal. Explain the genetic composition of the zygote. It means that the zygote, remember we say that the normal gamete, you need to tell us how many chromosomes will it be. It will have, the one which has extra chromosome will have 23 chromosomes. The normal gamete will have how many? Plus 1, which makes them to be 24. And then the normal gamete is going to have 23 chromosomes. When the two are fused, the resultant zygote will have 47 chromosomes instead of 46. So the Down syndrome, they're asking you, they just brought it in a short term, but I explained it was there in the questions I gave you, and I explained it, and I told you, take a screenshot. Take a screen what? Shit. All right. We continue the next question. All right. I hope, guys, you got all the marks there. 11 out of 11. We continue the next point. They are saying that, is there any question I skipped? Nope. They're saying that the diagram below shows. This one, I just used it from Distinction Material book, and then I told you what to do. The diagram below shows analysis of crime, okay, sample from the victim, sample, okay, from the suspect in their investigation. All right, in their investigation. In their investigation. Okay. They are saying that in their investigation, the police conducted the comparison of DNA to identify the criminal. All right. Identify the criminal. All right. Identify the technique which is being used. DNA profiling. DNA profiling. The technique. DNA profiling. Which suspect 1 or 2 was at the crime scene? So we see that this one matches with this, this 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 matches with this. Check, 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 man, check, man, check, man, check, man, check, man, check, man, check, man. So it's suspect 1. Suspect 1. Suspect 1. Give one observable reason for that. You you have to tell us that the DNA bands of suspect 1 matches with the DNA bands of that found on the crime scene. You understand? So that one of the bands matches with that of the crime scene. That's what it means. Therefore, it means that suspect 1 is the what? Is the problem, is the one who caused the problem. They're saying that give the possible reason why this suspect identified and 2 above might not be convicted if this evidence is used. Hey, don't forget that DNA is unique, but is only unique if a large sample is used. If a large sample is what? Is used. DNA is only used, you see, here is the question. You see it? There is the question I told you. Yes, suspect 1, suspect 2, suspect 3. And the answer was the middle one. This one and this one. You see, suspect 1. And I asked you the question Z, which followed there. So they just brought it. Alrighty. Okay, we continue. And then Alright. We're saying that basically DNA is not unique if a small sample is used. DNA is unique, yes. But this person might not be convicted because if a small sample is used, it might not be unique. That's why some parts of you can be resembling your father, you can be resembling your grandfather, you can be resembling your neighbor, you can be resembling your grandfather. You see, it doesn't necessarily mean that, your neighbor is your... I didn't say that. Yes, okay, if a small sample is used, so the DNA might not be unique. Then number 2, we call them the debates or the disadvantages. Then number 2, the person might be framed. What if someone collected his blood or somewhere and then went and then sprinkled it on the crime scene? It can be framed. Yes. So you can look at any other. Wabon. Human error. Someone is telling me even that one. Human error. Maybe someone who was drunk over the weekend has babalaza, and then in the morning on Monday, and then is doing the investigation, still even having babalaza. You think this person is gonna give you correct results? Okay. So it means that human errors can happen. Even I might not analyze properly. Oh, there is some contamination. There is a lot of things. Here is not like this. The answer. There are a lot of answers you can talk about. Then they're saying that give 2 possible reasons why the suspect identified above. Okay, we're done with that. Explain how the technique. Yes, explain how do I use this technique to identify, to bring about the determination of this individual. How am I going to use this technique to identify the individuals? So basically, the DNA, we know that DNA evidence might be explain how the technique is used in identifying the paternity test. So basically here, what you need to do is to first identify the the sample of the child. You understand? Then you identify the sample of the individual. Then you have to compare it with the what? With the individual. If you're talking about the parents, if it's a father, all the bars which correspond with the mother, the remaining bars must correspond with the father, and then that will confirm that that is a father. Let me show you here. Yes, it's just here. Okay, it's just here. Paternity test. Yes, paternity test. Yes, DNA profiling. A child received DNA from both parents, and then they're saying that the profile of the mother, child, and the possible father are determined, meaning that we collect them. Ne? And then after that, the comparison of DNA bands of the mother and the child is made. The remaining bands, all the remaining bands, this is the key point. All the remaining bands are compared to that of the father. If all the remaining bands of the child matches with that of the possible father, then it will be a biological father. If all the remaining bands does not match, then it will not be the possible father. You understand? Yes. So that person will not be the biological father. So that is what you're supposed to explain. Life is good, man. Oh, this one, they brought it. They brought it. Kasi this, and I say, I don't care how do you read it. Kadasi whatever. I'm saying the same thing. Whatever, I don't care. Is an autosomal genetic disorder caused by a dominant allele, and it is as a result of thickening of the walls of blood vessels. The diagram below shows. And we are bringing, we don't just bring, we bring. They are saying that the type of dominance, the type of diagram which is being shown there, this is what called a pedigree. How many offsprings for individual 1 and 4? Individual 1 and 4. Did I say 1 and 4? Did I read 3 and 4, man? 3 and 4 is 1. It's only one individual. That's individual 8. This question is the same question they asked. Individual is 1. The phenotype of individual 10. The phenotype. This person is normal. From the condition. No. This person, individual 10, is shaded, caused by a dominant allele. So if this person is suffering from CADICIL, whatever, I don't care. And yes, it's called this by. So this person has CADICIL, and the genotype of 5. This one, you remember what we said. So if this is 5, and then this person is normal, and it's caused by dominant allele, automatically this person is gonna be small d, because the moment a capital D is there, the individual is gonna be affected with CADICIL. You understand? So it means that all these people, they are small d. All these people small d. Why? Because this condition is caused by a dominant allele. Therefore, I will say that the genotype of this individual is D, small d. We continue the next question. Using the evidence from the diagram, explain why 1 and 2 are both. Using the evidence from the diagram, explain why the individuals 1 and 2 are both heterozygous. Individuals 1 and 2. How many marks? 4 marks. Oh yeah, I see now. They want you to prove that why is this person. Remember, this person is affected by the CADICIL, ne? So, and it's caused by dominant allele. So this person can be capital D, or capital D, small d. That's what I did even yesterday. Capital D, capital D, small d. All right. So now, but they produce a kid who is small d, small d, small d, small d. So if this is small d, this can't be. If this is small d also, you see, this one also can't be. So it means that one came from the father, one came from the mother, and one came from the father. So automatically is individual D is small d, and individual this one number 2 is small d. So they want you to put it in the writings. Then you're gonna tell us individual 5 and 6 are normal from the condition. Therefore, they are recessive. They contain 2 recessive alleles. That is, you are saying that these individuals, they are homozygous recessive. Still is the same thing as saying 2 recessive alleles. Then you tell us that this both of them, they obtain 1 recessive allele from either of the parent and the second recessive allele from another parent. You don't say that they obtain the recessive allele from the parent. They obtain 1 recessive allele from either from this person or from this person, and then the remaining recessive allele was obtained. If they got it from here, then the second one must be obtained from another parent. That's what they wanted. They wanted to show us that these two, they wanted the genotype of them, which is homozygous recessive, or they contain 2 recessive allele, and they wanted you to show us how did they become homozygous recessive. They inherited this recessive allele from the parents, meaning that they got 1 recessive allele from either of the parent and the second recessive allele from another parent. Hey, someone saying that this question was classwork. Yeah. Alright, bafethu. Hey, this one was your classwork. This one was our meal yesterday. If it was your classwork, it was our meal yesterday. It was our scope yesterday. Alright. Okay, we continue. What is the percentage chance of parent 7 and 8 having another child with Card cell? What is the percentage chance of having another child with 7 and 8 to have another child of having Card cell? So automatically this one is, since they have someone who is normal, is the capital D, small d, and then this one, since it's normal, is small d. So it's gonna be 50%, because if I say that this goes with that, is gonna be capital D, small d. This goes with that, is gonna be capital D, small d. This goes with this, is gonna be small d. Then this goes with this, is gonna be small d. So if it's 2 out of 4 for either this or this, and then times 100, so it's gonna be 50% chance. So the answer here is gonna become 50% chance. 50% chance. Someone said 100%. 100%, eh? How did it be 100% when this one is already sick? And then because they already produced someone who is small d, automatically this can't be like that. You understand? So if this was homozygous dominant, then automatically it would have been 100%. But it was not homozygous dominant, because of the individual 9 they produce, who is what? Who is normal. Yeah, is homozygous recessive. Okay, who got 9 out of 9? 9 out of 9. All right, we continue the next question. The next question is saying that, okay, blood group. Yes, we asked you a lot of questions on blood group, and they brought this in June, not June, in prelims of Gauteng. All right. The graph below shows the distribution of blood groups for 2 ethnic groups in certain countries during 2021. The percentage of people with each blood group. Which blood group? Which blood group is most common? Which blood group is most common in both ethnic groups? Most common. The percentage of with the blood group. So automatically it's going to be blood group blood group O, because if I add this and this, it's gonna be something. This one is a little bit lower, so the answer is gonna be blood group O. Has the highest percentage in ethnic group 1 than ethnic group 2. This one is C. 1 is lower than 2 is equal, lower than 2 is higher than 2, so it means that it's going to be blood group A. It's going to be blood group A. State the possible genotype of people in ethnic group 1. Guys, just a moment, a small moment here. I need to check up on the power. Okay, so we are saying that how is it not AB? They say the most common. How is it not AB? Which question is that? How is it not AB? The higher percentage, common. What did I say? Yeah. yes. Let me see the question. What is saying? They're saying that is the most common in both ethnic groups. Oh yeah, man. Yes. They're saying that the most common, meaning that they have almost the same amount, which is 4 out of 4. So it's B. Thank you so much. Is A, B. Yes. Then they are saying that which blood group has the highest percentage in group 1 than in ethnic group 2. Group 1, where is group 1? Yes, we say that. We said it is A. Yes, it's A. It's A. So I was rushing, ne? Yeah. If you said phylogenetic tree, no man, it's not phylogenetic tree. Even size confused. No man. All right, we continue. Wait, now how can you say that? Okay. Akira, you are rushing me. They are saying that set all the possible genotypes of state all the possible genotypes of blood group of state all the possible genotypes of 11% of people in ethnic group. 11%. Where is 11%? Oh, 11%. This 11%, it shows that it is blood group B. But this blood group B can be homozygous or can be heterozygous. It can be homozygous or it can be heterozygous. So it means that I'm gonna say blood group A, blood group B, heterozygous, or blood group B, homozygous, or blood group B heterozygous. So that is the meaning. So we got the next question. So it means that they want you to write the genotype of blood group B, of which it can be blood group B. If you wrote it like this, 0, we're gonna give you 0 with the eyes. If you wrote it like this, 0. If you wrote it like this, 0. there is no such. So I told you that yesterday. Each group, ethnic group 1 had a population of 360,000 people in 2021. Which blood group A B or O? A B or O had this? Okay, now I don't know. I need to check. Okay, so I need to find out. So I'm gonna say 39,600, yes, divide by out of 360,000 times 100. Ish, guys. I don't have a calculator here. You know, I'm just doing the question paper just like you. Just like you, I don't know. Guys, I don't know. How much did you get there? I don't have a calculator here. I have to calculate. Let me check and use my phone and see. Now I have to find out what is the answer, because if I don't... Okay, they're saying 39600 divide by 36000 0,11, yes, times 100, which is 11%, which is times 100, which is 11%. So I need to look at which one is 11%. Is blood group B. So the answer becomes blood group B. Blood group B. Wow, thank you so much, guys. You gave me 11%. Yeah, I didn't have the calculator. So yeah, I also got 11% here. Thank you so much. Yes, it's 11%. All right. Then they're saying that, you know, guys, don't make me rush, because when you rush me, I end up reading the question wrongly and then give you wrong answers, and I can't do that. All right. Explain the important. Explain. By the way, if you see me that you have read something wrongly, you tell me on the live, so that I'm able to check it before I continue. Explain in the inheritance of blood group AB. Explain the inheritance of blood group AB. Oh, okay, now you need to understand. You need to understand what is blood group AB. What is blood group AB? Blood group AB is codominance. Wabon. Blood group AB is codominance. So is blood group AB. So you're gonna tell us that blood group AB is codominance, isn't it? Whereby it's A and B. It has both allele for A and allele for B. You understand? One allele. Let me say, yes, one allele will come from the father, i.e. A or B, and the other allele will be inherited from the father, and the other allele or allele will be inherited from the mother, i.e. A or B. Why do I say A or B the same thing? Because I don't know which one is going to come from the father, which one is going to come from the mother. I don't know which is going to come from the mother. I don't know which one is going to come from the father. So one allele, yes, the person is codominant. Yes, they're going to hope to give you a mark there. And giving me the genotype of the person is AB, which is not the phenotype. The phenotype is given, but you want the genotype, which is AB. Then you have to show us where is this A coming from and where is this B coming from. It means that the A, one allele of either A or B will come from the father, and the remaining allele, either A or B, will come from the mother. That's how it's being inherited. Yes. Explain. So we continue the next question. Oh, they're asking you a question here. Name the type of dominance displayed in the inheritance of Palomino. Yeah, we did the horse. Yeah, Palomino are bred by horse breeder for their coat color. And then the Palomino has golden coat. Golden, okay. They're saying it is an offspring of the horse with the cream. It is an offspring. It is an offspring. Oh, I'm getting the question now. It is an offspring. This Palomino, whatever, I don't care. Palomino is an offspring of cream-colored coat. Okay, and the horse, which is AA, meaning that this one is AA. All right. And since they are both of them are capital letters, it means that it is co-dominance, or it is incomplete dominancy. Okay. They're saying that horse with chestnut colored coat. All right. It means that this one is gonna be small capital G, capital G. All right. Meaning that if it is a combination of the two from the offspring, it means it's going to be A and G. All right. Name the type of dominance which is being displayed. This is not a mixture. They say that Palomino is horse golden. Something golden. It's not a mixture of color. It's an intermediate of a color. Cream-like, and so since it is an intermediate, it is incomplete dominancy. That's why I gave you the differentiate between complete dominance and incomplete dominance on our scope. Yes. Explain the answer. So the answer, why do you say is incomplete dominance? It's because the Palomino, the offspring, yes, is heterozygous. Heterozygous. You understand? With an intermediate color, that is golden color, with an intermediate color. That's what you are supposed to look at. You need to show us that is heterozygous, and you also need to show us that it is incomplete. It is incomplete. Is incomplete dominancy. How? Heterozygous with an intermediate color. All right, meaning that it is a what? Is a middle color. All right, we are saying that breeding of horse with the desirable characteristics, okay, is a type of biotechnology. Breeding of horse with the desirable character, oh, I told you that we only give. Okay, this question, it was breeding of horse. The question which I brought, it was about cows with milk, but I also brought the question of horse. All right, cow with milk. And then I say that breeding of horse is a type of biotechnology. Name the type of biotechnology which is being shown there. It is selective breeding. Yes, because the question is saying that explain selective breeding for the cow to produce more milk. Okay, selective breeding. So this one is going to be selective breeding. Then they are saying that if you say neither of the allele is dominant, neither of the allele is dominant. If you say neither of the allele is dominant, even the codominance is neither of the allele is dominant. They need to talk about intermediate in that question. In this question, you need to talk about the intermediate color and the heterozygous. Maybe that one they will correct you related to that, but also maybe they will give you 1 mark. Yes. All right. We continue to the next question. Name the 2 phenotypes of the horse that must be bred. That must be. Name the 2 phenotypes of horse that must be bred. Interbred for 100% Palomino offspring. Remember, Palomino offspring is a combination of the two. Then automatically, I'm going to cross this and this. Then automatically, I'm going to get the, if I cross A with A and G with G like this, automatically, all of them is going to be GA or AG, whatever. GA, GA, GA. You see, all of them 100% is going to be Palomino. And then you see that we say that Palomino is AG or GA. It's the same thing. GA. You see, it's the same thing. You see, so it means that I have to cross. Your answer was supposed to be you have to cross cream colored coated horse, and then also with the also chestnut colored coat horse. We continue the next question. Use an cross to show the ratio of offsprings. Show the ratio of offsprings. But they say the horse of cream color. Okay, they say that cream colored coat was crossed with Palomino. Palomino is the second generation because you can't get it from the first generation. Maybe they will create accordingly. I don't know. But to me, I'll say P2, F2 because Palomino is the second generation. Or even those who said P1, F1, they could create you also. So phenotype. I told you, you get it from the question. They say, what did they say? Cream colored coat horse crossed with Palomino horse. Cool. Then we go to the next point. What do you say? You say genotype. Remember, Palomino is GA. And then this one, cream, we say that when you go back to cream is AA. Cream is AA. So now you must say meiosis must take place so that the gametes are separated. A, G, A. Then they say that this is called the it's right, but just not in the right position. This is the genotype. This is the meiosis, and then these are gametes, and then fertilization. Now fertilization must take place so that I form the offsprings. So in this case, you can draw a Punnett square, or you can do what I told you yesterday. Put a dot, put a dot, put a dot, put a dot, so that everything is in order. This goes with that. I'm gonna say GA, then this with that. It's gonna say AA, then this with that. It's gonna be GA, and then this with that is gonna be AA. So these are offsprings, F2 or F1 offsprings. And then now we come back here. What are they saying? They want show the phenotypic ratio. They want the phenotypic ratio only. So it's gonna be 2 GA, and then 2 AA. What is GA? GA is Palomino. Palomino, whatever. And then the other one, 2 Palomino, and then the other one is what is coated, what cream coated is cream. The other one 2 is cream. But what is the question? What is the question saying? What do they want? What do they want? They said ratio. So it's gonna be 2 to 2. But you can't write it like this because this is not the simplest. So it's gonna be 1 to 1. So that is the perfect answer. 1 to 1. Who got it right? We got the next question. Yeah, I wrote AG. It's the same thing. It's the same thing. The order doesn't matter. Yes, the order doesn't matter because they didn't give you the order to use. Either AG or GA is still fine. If you wrote AG or GA, you wrote AG, is fine. It's fine. 100% fine. Even here, if you wrote AG, you wrote AG is still fine. 100% fine. All right. Hey, the question is done. Guys, at least who got 48? 48. 50 out of 50. Oh, I have someone who got 48. Ah, I see you got 48. Someone is saying now I have 100. 86 out of 100. Hey, we are shifting. But guys, the paper was fair, ne? 49 out of 50. Ducky. You guys, 98 out of 150 so far. Thank you so much, guys. So it means that you only lost 2. Wow. That's so good, ne? You are so impressing me. You are impressive. You are impressing me so much. Yeah, I think I must be there when they're giving awards for life science, ne? Yes, they are in Midranda. See here. Thank you so much, guys, for following us on YouTube channel and TikTok. Facebook is there, but they always hack it. So there are some people even asking money from people, yet it's not us. We will never ask you money. We will never ask you money. And if we ask you money, you buy something, you buy it online, or it must be the account for Thunder. If they are books, that's the only money. If you are requesting for the books, nothing else. We don't send money once WhatsApp. We don't. Please don't do that. All right. Theory of human evolution are based on similarities between human and African apes. We brought this question. Give 2 characteristics related to vision. Okay, you can talk about any binocular vision, eyes in front. Yes, they can talk about whatever. You can see color. They can see color. All that. Okay, they're saying that describe 2 differences between the jaw of a human ape. Jaw. They're saying jaw. So for humans, it's like this. C shape, U shape, C shape. And then African ape, rectangular shape. Palate, the palate. The jaw for human is less protruding. The jaw for African ape, more protruding. Actually, African apes are prognanthus, if you wrote prognanthus. Well, this one you can talk about is non-prognanthus. Done. The jaw, I think even the canines can be there. For the case of humans, small canines, they just look at the jaw. Basically, they are looking at the jaw, and then you give the answers based on what? On the jaw. All right. We got the next question. Explain the significance of position of foramen magnum, the shape of the spine, okay, wait, the shape of the spine, okay. Okay, they're saying that explain the significance of position of foramen magnum, the shape of the spine, and the pelvis. Guys, this is the question which I asked you yesterday. This one. Where is it here, by the way? It's here. This one, the factors which affect 3 factors. The way they asked you them, and then I showed you where to get them from the distinction material book. Distinction material book, it will never let you down. So these are the questions we're asking you there. Following, you see that all this you are talking about, they're asking you, and then I told you, take a screenshot. So you're saying that foramen magnum is in a more forward position to allow the spine connect or enter vertically. Pelvic girdle is short and wider to support the upper body weight. Spine is S-shape to act as shock absorber to allow flexibility during movement. That's it. Nothing else. We got the next question. They're saying that. So, that is 6 marks. Mahala. This one, it was also there. How many genera are represented diagram? I said 1, 2, 3. 3 genera. 3. Give 2 examples of Australopithecus africanus species found in South Africa. Give 2. Give 2. Give 2. There is the question I brought. Give 2 Australopithecus. Give 2 Australopithecus africanus found in South Africa. And then, 2. I asked for 2, not 3. I know there are 3. I wanted 2. 2. All right. They're saying that Australopithecus africanus, there is Taom child, Littlefoot, or Mrs. Place. Okay, sharp. Give the average brain of species that was first used tools. The average. Remember, it's Homo habilis. That's what we said. This one, the average volume. So you're gonna say 609 milliliters. Calculate the percentage increase. This one I explained it even in paper 1. Say that I'm not going to talk about paper 2 because you know. He said that final minus initial over initial times 100. Okay, final. Where is final? For Homo habilis and the Homo sapiens. Homo habilis. Where is Homo habilis? Homo habilis 609. 609. 6. Aibo. Final. Where is our final? Final is 1,333. 1,330 minus 609 over 609 because initial times 100. So what is the answer? I don't have a calculator, I told you. Hey, why am I the only one seeing your scope after the paper? But I nailed it. It's because maybe you don't keep yourself on Thundereduc. You must keep yourself on Thundereduc with these scopes from grade 8. Yeah, grade 8. Yeah, so basically, sorry if you just saw it, but I know that at least you have been on Thunder. They're saying that I don't have a calculator. You guys, who got it? I will never skip your video. Okay, guys, let me calculate. I don't have a calculator, guys. If you have a calculator, okay. How much did you get, you guys? How much did you get? You say get 180. 118.39. Oh, so I see everyone is getting that. Yes, remember last time when I telling you that the percentage increase, it can be more than 100%. Yes, let me see what is the double of this. If I say this is the double of this, yeah, obviously is 1,218. 18. This is more than this, so automatically it doubled and then went up. So it's supposed to be more than 100%. So from the chart they're saying it is 118.39. Since everyone is getting that, then automatically it could be the answer. So if you calculate with the calculator, guys, I don't have a calculator. I'm also doing the question the way you are doing it. When they open, then you start doing. All right. Thank you so much, guys. Let's go to the next question. Is it the last question? Still have too many questions. All right. They are saying that the extract and diagram below based on the rat tails, whatever. Breeding month of ratitis, whatever. I don't know. Okay, these are laying flightless birds. The eggs laying month are influenced by the month factor such as temperature, length of the day, availability of food. Ostrich normally lay their eggs in September and renas, whatever, between October and March. While imps lay their eggs. I remember when they brought this question some time back. Yeah, it killed the people. I thought, I hope it's not gonna kill you, this one. Then they're saying that state what is meant by biological species. Remember we said what's a species is a group of organisms that have similar characteristics that are able to interbreed naturally. Naturally to produce, they're able to interbreed naturally because of the biological to produce fertile offspring. Yes. Then they're saying that use the information in the extract explain why ostrich and emus considered to be different species. So they consider to be different species because they are supposed to be they are supposed to be able to interbreed. So in this case, one breeds in September, another one breeds in November. Automatically they can't breed, so they cannot breed. So they are different species. So they cannot interbreed to produce a fertile offspring because they are breeding at different time of the year. Yes. So that could be, yes, that could be the answer, that basically they lay their eggs normally in different times of the year. One is in September and then another one is in November. Lays its eggs in November. We got the next question. They're saying that you skipped a question. Sir, you skipped a question. Oh, guys, tell me which question did I skip? So this one is done. And also this one is done. Hey, guys, tell me which question did I skip? Oh, thank you so much, guys. I've seen it. Okay. Should I first finish this? I go back, or I should first... Okay, let me go back. Explain the relationship of average brain volume and the cultural evidence in human evolution. Remember, as the brain volume increases, there will be in advancements in the what? In the cultural evidence. You understand? Yes, I brought this question yesterday from our scope. Say that and they also brought it in this year in May, June. Say that explain the relationship of the average brain volume. As the average brain volume of an individual increases, yes, it increases the intelligence. You don't just link it direct because the brain volume doesn't mean anything to the technology you're doing. As the brain volume increases, the intelligence also increases. So it gives the individual more thought, hence an advancement in the tools which are being used. Yes, so you were supposed to explain it that side, that as the brain volume increases, still I skipped another one. Basically, I skipped the whole question. As the brain volume increases, okay, I'm gonna go back to it. As the brain volume increases, all right, there is more brain cells, and then this will increase the intelligence of an individual. Yes, hence advancements in the tools which are being used by human. Which shows that the advancements in tools will show that there is an increase in the brain, in the intelligence of an individual. You understand? So it is the brain which determines the intelligence and which determines the tools, not the tools determining the brain. Nope. Yes. The brain decomposes after it dies. Okay. Explain why scientists are able to determine the brain volume of the fossils. Explain how the scientists are able to determine the brain volume. Yes, I brought this question. They're asking you, but I brought it. How is the cranium related to? So the more, it means that when it decomposes, it leaves the skull, it leaves the cranium now, because we're talking about the brain. It leaves the cranium. So the scientists explain how scientists are able to determine the volume of the brain. It means that they have to determine the space in the cranium. So as the cranium increases, yes, it means that the number of brain cells to be accommodated also increases. You understand? So let me show you what here is the answer. Here is the answer. Ne? But now check that if the cranium, now this one is out, it's not there, and also this one is out, the brain is not there. You understand? So but you see that the skull, the cranium, not the skull, the cranium, the space of the cranium, the volume of the cranium will determine how much brain the individual is having, because we cannot trace the brain of 2 million years ago individual. No, but we'll trace the skull because the bones will be still there. So the scientists use the volume of the skull, and then to determine the volume, to determine the size of the brain. You understand? Therefore, now they can link it to how big the brain was. I think now, yes, I hope I have not skipped any other question. Give to average was first used tools. This I answered that. I also answered that. Okay, thank you. We continue the next question, guys. I stopped here. State 2 other factor of reproductive isolation that would have prevented this and this from interbreeding if they were the same habitat. Oh, if they were the same habitat. First of all, breeding at different time of their different seasons. Obviously, even if they were the same habitat, they wouldn't have breed in the same time of their. And then we can also talk about they could have been maybe, did I say from the extract? Nope, they didn't say from extract. Different courtship behavior. Different courtship behavior. The infertile offspring. Yes, this. Prevention of fertilization. Different courtship behavior. Breeding at different times of their. Did I say other than? Let me say. Give to other reproductive isolation mechanism. Okay, so you don't talk about different season, because here they talked about different season, so you don't talk about that. So it means that you're gonna just talk about different courtship behavior and then prevention of fertilization or resulting in or infertile offsprings. We cannot talk about pollinating agent. Are they flies? They're not flies, so you don't talk about different pollinating agent. Yes, so you only talk about this, you talk about this and this. That's it. Then we are saying that identify the evidence from evolution represented in the diagram above. Identify the evidence for evolution represented in the diagram above. Oh, evidence. Let me read again. This is a geographical isolation mechanism, distribution of organisms in different parts of the world. Geographical isolation. Yes. This is geographical isolation. That is the one evidence of evolution. Yes. Distribution of organisms to different parts of the world. Okay. You're saying that what if you said geographically? What if you said geographically? I don't understand what you're saying there, but maybe they will give you. But it's a geographical isolation. Oh, geographical, they are being separate geographical. I don't know. Okay. They're saying that identify the evidence. Okay, we're done. Describe how the geographical isolation. How is this geographic isolation support the theory of evolution? So automatically, now here you have to explain to us how did they distribute themselves, and then to become these different organisms. Yes. It's there. It's there in our hey, it's there. I wrote it here. Where is it? Yes, I wrote it there. Yes. So, starvation resistance refers to the period or for which organisms can go without food before it dies. An investigation was conducted to determine the effect of food the availability of food on the starvation resistance in the fruit flies. They're saying that the procedure was as follow: 500 fruit flies of the same age. Okay, let me just read just fast so that I'm able to understand what is here. They were all placed in the container of growth. Were given in cellulose. You can read yourself, ne? Me, I'm reading to understand. The food source was removed after an hour. Okay, they took 8% of the fruits. Okay, to die from starvation, and recorded when did you record? Okay, the flies were removed from the container. Okay, so that they clear up. Food sources were returned, the remaining, so they repeated. Okay, you have transferred to the new container. The first fruit flies were allowed to grow in the same age of the first generation. The food source were the time taken for the fruit fly to die starvation was recorded. Yeah, someone is okay. Let me first finish up by procedure represent. Yeah, someone is asking me if I wrote by geography here, identify the evidence shown in. Yes, by geography is the same thing. Geographical isolation or by geography is the same thing. They will still give you. Yeah, by geography still is fine. All right. State the independent variable in this scenario. Okay, I'm seeing it. State the independent variable for this scenario. So in this scenario, the independent variable, yeah, they said it. They talked about the aim here. An investigation was conducted to determine the effect of availability of food on the starvation resistance in the fruits, meaning the availability of food will be the independent variable. Then they are saying that describe how the dependent variable was measured. How did they measure the dependent variable? How did they measure the dependent variable? They said it here. The time taken, the time it took for 80% of the fruit flies to die from starvation was recorded. You see? Time taken. That's how they measured it. We got the next question. State one control variable that was considered when selecting the fruit flies. What variable did they talk? They talked about. They talked about the age, same age. They used the same age, so it was this the control variable usually they used. We got the next one. Harvard here. I come. They're saying that explain why the investigator gave the fruit the same food. Why did they give them throughout? Basically to minimize the interference from the other food, because the other food could be having different content and might result in different effects. Yes, so basically they wanted to bring about the validity of the results. Yes, these are questions where the answer might not be one. You understand? The answer might not be one. Then they got the next question. It's almost done. Then they are saying that the graph below shows show the results obtained in the first generation and then 60th generation. Okay, describe the result obtained in the investigation. Oh, they're saying describe. Just tell us what is happening. So basically, you see that in the first generation, we see that there are many died, many flies died between 0 and 40 what? 40 hours. Between 0 and 40 hours. In the second, in the 60th generation, many flies died. Yes, number of, let me check here, hours until death from starvation. Meaning that here, all this time they didn't die. Here they didn't die, they only started to die here. Here, all this time they didn't die. So here it means that many died between maybe 140 to 180. Yes, after between 140 to 180 hours of starvation. Here many died between 0. Is it between 0? Maybe between 20 and 40. Does it touches to the no, it's not 0. Between maybe 20, between 10 and 40. Yes, many of them died after those hours of starvation. But here you just have to describe your English. Then they're saying that use the Darwin theory, explain the increase in the starvation. Okay, here they want you to use natural selection, the principle of natural selection. Still, is the same story here which I gave you here. Yes, here the principle of natural selection. So the other one was speciation. Now this one is natural selection. So they brought both of them. I know that you guys, you got it, so I'm gonna try to explain it. So those who explain by geography here, how did those new species form, you will get your marks. Yes, 5 marks using speciation. And then here, those who used their evolution by natural selection, they also going to get their marks. But here you have to tell us that you have to apply. Even there you have to apply using basically flies produce a large number of offsprings. There is variation among these flies. You understand? Those which are resistant to starvation. Yes, they said they're resistant to starvation. Yes, they said they're resistant to starvation. You see, those which are resistant to starvation and those which are non-resistant to starvation. In the first generation, after exposed to starvation, yes, those which are non-resistant to starvation, they died in large numbers. While those which were resistant to starvation, they survived. Those which are resistant to starvation, they pass the allele of resistance to starvation to the next generation. You understand? After many generation, there was a high proportion of flies which are resistant to starvation. You understand? Than those which are not resistant to starvation. So basically, that is it. I think, guys, that is it. I hope you guys, you got all marks. Thank you so much, guys. Let me see people from the comment section. What are they saying? Is speciation in the speciation? I forgot to include no gene flow. Yes, you lose a mark. Then there's someone geographic isolation and speciation are the same. Yes, it is the same. Basically, speciation is divided into two. We have speciation by geographic isolation and speciation by sympatric, which is it could be distance or it could be seasons, distance, like that. So in this case, we only describe geographical isolation, which results in by geography. the organisms deciding to differ. Oh, Mr. Saidi, will you always be famous, my dear? Thank you so much, guys. Thank you so much. Thank you so much. Who is this one? Spesihle Butelezi. Spesihle Butelezi, you have been with me for so long. For so long. Spesihle, where do you stay? I think I should send you something from Thunder. Where do you stay? Or try to access my numbers for Thunder. Any number from Thunder, then you ask for Mr. Saidi. Then maybe we will send you something, because I've seen you for so many since January. You are with us. Thank you so much, guys. You guys, you rock. You guys, you rock. I see the Max Day happiness I'm having here. I got a distinction because of you. Lolo. Thank you so much, Priscilla. 1-4 out of 150. Guys, I wish you get all of you distinctions. You remember it was 100% distinction. We want distinction project, 100% distinction. Thank you so much, guys. I got 90%. That's good. It has been 2 years, sir. I'm from Gauteng. Tetilo Secondary School. I think, I don't know. Let me just take a screenshot of it. I will try to follow it up, but try to check upon me so that I'm able to maybe send you something from Thunder. Anything which is written as a proud subscriber from Thunder. Yes. See you in another life, guys. See you too, guys. This is my prayer for you. I pray that you guys, all of you, you pass. I pray that when the markers are marking, they see correct answers only. I pray that you get a lot of money, which is blessed money. I pray that you enjoy your life. I pray that you get a healthy life, you and your family. And I pray that you become happy when the marks come back. See you until you see me again. See you when I'm driving as you are driving. Amen, 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 amen. Guys, I think I'm done for today. I'm done for there. Yeah. So it means that from now, I need to prepare for my holiday trip. So that yeah. All right, guys, it has been a wonderful year. Our prediction or our scope was nice. For paper 1 it was nice. For paper 2 it was nice. If you have sisters and brothers, you can also refer to them so that they also be able to benefit. So guys, we are going back to geography. Thank you so much, guys.
For at least everyone gave a chance to everyone to pass. So if it was me writing this paper, these are some of the answers maybe I would have written if I was the one sitting in this exam. So please take note that this is not the final memorandum, as the memorandum will be released officially. So let's go without wasting time. It is MSID, so that we are able to discuss about it. They're saying that change in the characteristic of species over time is known as what? So when you say change in the characteristic over time, you have the biological species, there's no change there. They're saying that population, this is a group of organisms these organisms which are similar, capable of interbreeding, produce a fertile offspring, living together in a particular time. So there is nothing related to change there. They're saying natural selection, this is not related also change in the characteristic over time. So if you look at evolution, remember evolution is a change, gradual change in the complexity of an organism. So since we are saying that characteristic of species which is a living organism, therefore it is biological. So D looks to be the most correct answer, and you get the 2 marks there. Which one of the following option is correct combination of sources of variation? So we see that crossing over random arrangement of chromosome, crossing over correct random arrangement of chromosome when, okay, they have not specified. Mutations also correct. But if you talk about mitosis, that makes it to be wrong. So we go to the next one, crossing over correct random arrangement of chromosome, correct. Continuous variation is not a factor which causes variation, but random fertilization is correct. So this one makes it to be wrong. We go to next one. They're saying random mating, correct. Random fertilization, correct. Mutation, correct. And meiosis, correct. Remember meiosis? We have not talked about random arrangement of chromosomes during metaphase, and we have not talked about crossing over. So those two are being generalized here. So C looks to be the most correct answer. They're saying that meiosis, mutation, discontinuous variations out. So C looks to be the most correct answer, and you get 2 marks there. They're saying that evolution involving long period of time during species undergoing little or no change followed by a short period of rapid. Guys, we discussed all of these things before you wrote the paper. Yes, we discussed everything. So let's just continue and find out where species undergo rapid change through natural selection. Lamarckism, no. Punctuated equilibrium, we say that is long period of no change and rapid period of a short period of rapid change. Long period of no change, rapid short period of rapid change. So it keeps on going like that, like that. So it's like a stair. So this looks to be the most correct answer. Artificial selection, not really. Gradualism, you see it is saying it's gradual, so it's out. So B looks to be the most correct answer. So we go to the next question, 1.1.4. Which one of the following fossil does not belong to Australopithecus africanus? Mrs. Place is correct. Lucy is Australopithecus afarensis, little foot and home child. So B looks to be the most correct answer in this case. You get your 2 marks. 1.5, which one of the following is only found in RNA nucleotide? RNA nucleotide, when you say RNA nucleotide, here we are looking at adenine nitrogenous bases. We are finding it here. The key point here is only and RNA nucleotide. So they're saying uracil nitrogenous base, correct. This one, adenine, we find it in RNA and DNA, so it's not. So it makes this one to be wrong. DNA, deoxyribose only found in DNA. Phosphate, it can find it we can find it in RNA and DNA, so it's out because of the word only. So B looks to be the most correct. We go to the next question. You're saying that 1.6 to 1.7 are based on the diagram below. Okay, you have a black hen. I don't know whether it's a hen or a cock. And then a white one. Then you cross them, you get gray. So you're getting an intermediate. So that's the key point there, that you are getting an intermediate. So we are saying that which type of dominance result in intermediate phenotype that's shown in the diagram above. So that is incomplete dominancy. Ne? Yes, it is incomplete dominancy. So the answer becomes C, because for dominancy you have a mixture of the two. Yes. So if you look at this, it looks to be a mixture of black and white, but that's as a result of not the perfect picture. But since they specified it here and they said gray, so it makes it to be more sensible. So C becomes the most correct answer. 1.7, the genotype of gray of chicken is, so since it is a combination of the two, the combination of the what? Of the two, so it's going to become capital B, small b, capital W. Why do we say that? Because of the B, capital B is here, and then you have a capital W, so that when you mix them together, you form capital B, capital W, which is incomplete dominancy. However, even the codominancy is the same type of genotype. We go to 1.8. The diagram below shows the formation of the sperm cells of fruit fly. Okay, sperm cells of what? Fruit fly. Fruit fly, this is what called a Drosophila melanogaster. Yes, scientifically we call it what? Drosophila melanogaster. So this Drosophila melanogaster, we're saying that it has 20 chromosomes in the nucleus, and then you have after mitosis, meaning that the number of chromosomes remain the same. The number of chromosomes remain the same. But now what happens? After meiosis. You see, this is after meiosis, and meaning that now you have produced the what? The gametes. So it means that this becomes 1N. Into N, it's 1N. How many chromosomes are present in W and D? So it means that we're going to find out that in W, we're going to have W, this one, we say it's 2N, meaning that is 20. And then this one must be 1N, which means it is 10. So we have to look for that, which is this and this. So D looks to be the most correct answer. Then we go to the next question, 1.9. The diagram below represents the different phases of meiosis, saying that which one of the following correct sequence of the phase. So we see that X, Y, Z. So you see that the correct, which one of the following represents the correct sequence of the what? The correct sequence of the, so we see that this is metaphase. See, they're in the middle, yes, and then this is anaphase 1. Why? Because they are still chromosomes. Well, this is anaphase 2. You understand? So because the anaphase 2, they are chromatids. You see, they are chromatids. So it means that I'm gonna start with 1. I go to 2. I go to 3. So Y, it must start with Y. So whatever is not Y is out. So it's gonna be either this or this. So we go to the next one, which is 2, which is X. So it's gonna be YX. This is YZ. So this one out. So this one looks to be the most correct answer. And then you see that is Z. So automatically that makes it to be the most correct answer. Then we go to question 1.1.10. The diagram below shows the DNA molecule on the left and a pool of few nucleotides on the right. Original. So you have the original DNA molecule, then the new DNA molecule. If replication of original DNA molecule takes place using new nucleotides, what would the product of replication look like? They say they are using this. So it means that the original must look like this, and then the new must look like that. You understand? So it means that, remember, in the DNA replication, this structure is supposed to unwind so that each is forming a template. You understand? So it means that this one must be on one side, Also, this one must be on the other side, then it opens. You understand? So even this one must be like that. So let me just use a different color so that now this one is the red one. So then you're going to have to be with another strand here, form another structure, and then also another strand here to form another strand. So that now it means that the red one becomes the new, and then this blue one becomes the old. So we have to look for something like that. So we see that this one is out, this one is all new. Then you see it has the old one, which is the blue, and then the black one also, which is the blue, and then you have the red one, which is this one, which is the new one, and then this one, which is the new one, because this and this, they became template for formation of the new strand. So C looks to be the most correct answer. And then I will say that C, you get 2 marks, and this marks our objectives, who got 20 out of 20. And if you got that, congratulations for this achievement. Then we go to the terminologies. We are saying the diagram below used to show the evolution relationship between the species. How do we call that diagram? We call it phylogenetic tree. Then we are saying that ability of chimpanzee to walk on 4 limbs. So we say that is quadrupedalism. So we get a mark there. The part of the skull where that houses the brain, they brought this one even in paper 1. So it is the cranium. They are saying the structure in the animal cell that give rise to the spindle fibers or spindle threads. We call it centrioles. So those are 2 marks. Sorry, 1 mark saying that sex-linked genetic disorder where blood fails to clot or does not clot. So we call it hemophilia. So also another mark. They're saying that the genetic state, the genetic state of having 2 identical alleles for a particular characteristic. So how do we call that? Genetic disorder having 2 identical, that is homozygous. So it becomes homozygous. Then they are saying that an organelle containing DNA found in plant cell and not in animal cell. How do you call that organelle which has DNA found in plant cell only, but you cannot find it in animal cell? So we say that is a chloroplast. Yes. So that is it, guys. I hope you got all the 7 out of 7. If you got 7 out of 7, congratulations. If you didn't get that, 5, 6, still is fine. All right, let's go to law of use and disuse. Who said that? Basically, it is Lamaka who said that. So the answer is B, only. The word only must be there. I've been emphasizing that. They're saying that because this one they talked about punctuated equilibrium. So they're saying that chromosome complete complement of women with Down syndrome. Remember, Down syndrome is chromosome number 21, but here they're saying 44. So since they're saying 44, they are supposed to be 45, and then XX. Yes, but since this is 44, so this is a normal person. And then this one, it is not XXX because Down syndrome doesn't happen on a sexy chromosome. So the answer looks to be none. Then they're saying that the father of genetics, we call it Mendel. Mendel is the father of genetics. So we get also there 2 marks. So it is A only. So basically it is B only, none, and then A only in that regard. So I hope that one was also a little bit simpler. You got 10 out of 10. Then we go to the next question. They're saying that Mr. Forbes has had a baked graduation cake for her sister and then left in the container in the cupboard. When she returned from work, all remained in the container were crammed from the cake and trace of blood droplets where the thief had grazed his or her finger on the sharp part of the container. It's the DNA sample of her 2 children along with the DNA sample from the blood found in the container. All right, DNA sample of child A and DNA sample of child B. So they're saying that name the technique used to compare these results. Remember the definition, they're no longer asking it, but now they're asking what is this technique. So we say that it's called the DNA profiling. There's a difference between DNA profile and DNA profiling. DNA profiling is the technique, while DNA profile is this what you get after using the what? After using the technique. So give the letter of the child who most likely ate the cake. So in this case, we're gonna have to look at, because they say that this goes with that, goes with that, this goes with that, this goes with that, this goes with that. So looks to be child number B, child B. Then they're saying that From the list below, write down the letter of two correct uses of the technique mentioned above. So they are saying that from the list below, write down the letter of two correct uses of the technique technique use. Oh, they gave you what is here. So basically, cloning, no. Identification of genetic disorder, correct. Establishment of family, what? Family relations, yes. Genetic engineering, no. So the answer is B and C. Yeah, B and C. Yeah, I see, guys. The paper is still moving well. It's not that bad. It's not that bad. At least it could be a recovery from paper one for those who are crying for paper one. How do you see? Tell me in the comment section, and then I find out how do you see this paper, or how do you see this paper was. They're saying that diagrams A and B show the skull of two hominids. Skull of two hominids, and then you have skull A, and then you have skull B. So if you're saying that you have skull A and skull B, large cranium. So where is the large cranium? Which diagram, or if you look at it, you see that here it has a big space of the what? Of the skull. So it looks that B has the biggest cranium. Skull of a chimpanzee, remember skull of a chimpanzee. Chimpanzee walks almost on two limbs, so the foramen magnum must be in a more backward position. So B, and also if you look at the teeth and the shape of the palate, you see that is rectangular. So I will say that skull A is Z for chimpanzee. Then they're saying that name the opening to which the arrow is pointing. So how do you call that opening? The opening is called foramen magnum. Foramen magnum. Yes, so that's what we call. State the shape of the jaw. How do you call that shape of the jaw of A? So you see that we say that is a rectangular. It's a rectangular shape. Yes, and then they're saying that give a letter of the diagram A and B, A or B, representing the bipedal hominid. Bipedal, why do we have to say A or B? In this case, I will say B. Why? Because of the position of foramen magnum is in a more forward position. If you look, yes, it is in a more forward position. But they did ask you to give a reason, so I will use that to identify as. We shall say that this is B. They're saying which skull one or two better adapted to chew raw uncooked food. I like the way they have asked these questions. Yes, because we have been asking them the other way around. Yes, the person who said this paper, yeah, you are good. So now we are saying that I will say in this case, I will say that which skull in diagram is better adapted for chewing raw and cooked food. So I will say in this case, I will say A. Why am I saying A? Because of the teeth, the kind of teeth this species is having. Look at the kind of teeth this species is having. It shows that it was feeding on raw, tough food. So because it was feeding on raw, tough food, so I will say that that is the answer in that regard. So give the scientific name of modern humans. Remember, I told you that whenever you are writing the scientific name, you have to write the first name, which I call the surname, which I call in science the genus name. The genus name must start with a capital letter, and the species name, or what I can call your names, it must start with a small letter. You don't stop there, you underline. Homo sapiens. Homo sapiens. So, since you are not a computer, you can't write it in italics. So it is Homo, yes, not Homo. Homo, then sapiens. Yes. So after that, you underline. Make sure that you underline. But make sure that this letter is a capital letter, and make sure that this one is a small letter. If you see that mine is big, then you can write it small. Sa Homo sapiens. Yes. So please, you must always write it like that. Yeah, you see, this question was not that bad. You could get all the 7 marks here. Yes. Okay. We got to question 1.6. Let's go to question 1.6. We are saying that, what are they saying in question 1.6? They're saying that in humans, one gene controls hairline and another controls freckles. The hairline can be curved, forming a point down, meaning this, pointing down the forehead, called Windows what? Peak, H, capital H. And then that is straight like this. I think it is small. I think even this one has the same thing. Freckles, which are tiny brown spots on the face of the what? Present, these ones. They're saying that the diagram below shows a cross between a male and a female who are heterozygous for both genes, meaning that this is heterozygous, okay? It's heterozygous, and this is heterozygous. And then if you look at this, we shall see that capital F, small f, when I do the gametes, this is gonna go with this, is gonna be capital H, capital F, then this is gonna go with this, capital H, small f, and then this with this is gonna be small h, and capital F, and then this with this is gonna be small f, sorry, small h, and then small f. All right, let's see, confirm, and then it's even the same story here. It's gonna be the same. So if I have these ones, correct, I have it, correct. Then I have it. Huh? What's happening here? Is there... this looks to be a small mistake. I don't know how they're gonna mark it, but basically when you look at it, it is supposed to be small h, small f, because if you have these gametes, so these gametes, they came, these gametes are supposed to be transferred here. You understand? Yeah, the same way these ones, because it is the same genotype, this one has this, which are not resembling this. So there was a small mistake there. But let me see if them writing it like this will influence your answers. Yes. All right. Name the type of genetic cross represented above. It is a dihybrid cross. Okay, correct. State the dominant characteristic of hairline. Dominant characteristic of hairline. They said it here. It is the Windows peak. This one, Windows peak, they said capital H. Windows peak hairline. All right. So we have not yet influenced that. Name the process in the reproduction mentioned above. Name the process in reproduction indicated by above. Remember, this is basically showing us that, it could be fertilization, because this is fertilization, but also this one should be this to form the gametes. It is meiosis. You understand? And then still I can also see, because I see a sperm here, so it can also be gametogenesis. All right. Because I'm seeing a sperm here and seeing a sperm, so it can't be oogenesis and gametogenesis. Sorry, and spermatogenesis. So I'll maybe have combined it, and then I say gametogenesis. Or how do gametes form? They form through meiosis. All right. Let's find out what they're saying. Arrow. Where is the arrow? Arrow. Oh, these arrows. What am I forming here? So basically, it looks to be much more accepted as gametogenesis. And then they are saying that Pinnet square. The Pinnet square is basically fertilization. Remember that we always write here that fertilization. So that is it, fertilization. They're saying that the genotype of offspring X. So now this is going to affect us. The genotype of, so I'm going to continue the way it is there. I'm going to say capital H, capital H, capital H, capital H, small f. Oh, wait. I need to see properly. It's gonna be this with this. So it's gonna be capital H, capital F, small f. And then this one is gonna be capital H, small f, small f. You see? Yes. But if someone had written, if we were supposed to take it the way it was supposed to be, remember, it's supposed small h, so it would have been capital H, small h, capital F, small f. So instead of capital H here, it would have been small h. If we have taken it, if like the proper way, the way how it's supposed to be, but it will depend on how they have marked. But obviously, this, if we continue the way it is here, if you did not maybe see it, so it could be the mark there. Yes. So basically, it is gonna be like that. So just cross this with this so that I'm able to get this. And then to get this, I have to cross this and then with this. So where they meet, and then that is why. Or the allele for the start, as long as it is allele of the same character. So if you look at this, capital small h and then capital F, because these are two different alleles. That's why